Oh, there's some more fishing line there.
All right, Colleen, you're live.
All right. Hi, everybody. We're back. We're now in a new location. If you've been following us the last couple days, we are now on the southwest part of Puerto Rico in a region called La Praguera. And we just got down to the bottom. We're at about 340 meters. And we're just kind of transiting to the east a little bit, and then we're going to start going up a really steep wall that should take us into some mesophotic reefs. So it should be really exciting. I forgot to introduce myself. I'm Colleen Hansel, and I'm a scientist at Woods Hole Oceanographic Institution. So we're just seeing some sponges now, a lot of sand, a bit of sargassum here and there, and what looks to be fishing line. In the southwest area of Puerto Rico, it's a very steep wall, and right at the crest is a region that's well known for having just beautiful mesophotic reefs. So reefs that span about 50 meters to about 150 meters to 200 meters in some areas. What's really exciting about these reefs is that the corals transition from being in sunlight, so in the photic region of the ocean, to the mesophotic, which is really much lower sunlight, into the deep, or the aphotic region. So we should see a nice progression of different types of corals during through that transition from the upper mesophotic to the deeper mesophotic. We won't be able to get up into the full photic region because we're in an ROV, so we're already kind of pushing the limits here, getting up to the upper mesophotic. So in the upper area, this is where we can scuba dive up about, to about 30 meters or so. So we're just going to be below that and exploring that 50 to 150 meter range. So unlike the dives we had the last few days, we're going to see a lot more stony corals, so scleractinians, ones that form kind of dense calcium carbonate aragonitic skeletons. And we'll also see a lot of the corals we've grown to love over the last couple days, some octocorals. And that will transition quite a bit as we're moving over that 150 meter range. So it's going to be very sharp transitions due to the penetration of light, or lack of penetration of light, as you get deeper. Seeing a lot of sargassum over here.
Saw a lot on the sea surface.
Yeah, today, moving to the southwest coast here, we're seeing huge patches of sargassum in the waters around the ship. We know that the sargassum belt is moving west, so we're hoping that our luck holds out and that it doesn't get here until our cruise is over. Otherwise, that could make diving a little complicated. So you see some sponges.
Is that green on the map our target path?
It was. It's since we're now going to go a little bit to the east of that and then north.
Yeah.
Oh, is there something?
There's a mooring ball out there.
Oh, I see.
Yeah, we're moving along the bottom here because as we got to the station, we saw that there was a mooring buoy on the surface, so we're wanting to be safe and move far to the east of that. Don't want to get tangled up in in a line.
There's an anchor right there.
Is that an anchor?
Former mooring ball,
Oh, yeah.
Look at that. Huh.
chain, road.
So this may be an area where people have come up to a mooring and tie up their boats to go diving in the shallower regions along the coast here, or fishing.
Also, just a shout out to our listeners. You should know that our narrator has a birthday today. She's spending her birthday narrating this exciting dive.
I was told I'm gonna get a big discovery today.
Yeah, it's in the cards.
We might find Atlantis today.
We're gonna find Atlantis.
The lost city. of Atlantis.
What depth, 200 meters? Never spawned in Atlantis, that's what we're calling.
Sponge Lannis.
Isn't there some story that Puerto Rico used to be Atlantis and it came up?
Is there?
I heard that from a Puerto Rican that.
Didn't Atlantis go
I
down,
haven't-
not up?
I don't mean, no, you've never heard that? Never mind. Little magical thinking.
Search that.
You're just gonna squash that one.
Puerto Rico Atlantis
Oops.
theory.
Is the red circle on the map the ship? Oh, yeah.
What are we doing?
The blue is. What's the red?
So we try to avoid getting in this area with the RV unless we're really near the surface, because it
okay.
can cause some problems with ground over.
I see. And what's the pale blue dot?
This is where we've, it's the waypoint that we give the vessel to try to
Oh.
move to.
Oh, okay.
So I'll move this and then they follow.
Oh, okay. Is it the same as the pink on the?
Pumps look happy. Yeah.
Yes.
Solaris is bathing, looking good.
Yes, during our transit last night, we had the opportunity to spend some quality time with our sensor, which was nice. Instead of when we bring the ROV up, we typically want to get it back in the water as fast as we can. So we've been just kind of troubleshooting quickly. But during the transit, we had a nice long chunk of time to be able to kind of pull the components apart, make sure things were working right. We found a problem that we have been kind of struggling with, and we think we've got that all figured out.
To the south, correct? Southeast.
People in chat are
These
wishing
things
you a
start
happy
moving
birthday.
north from what you
Oh.
can see now.
Thank you, everybody. At my age, I stopped celebrating birthdays.
Once you get to this, waypoint, I'll start moving this north.
Except I got a bunch of ice cream for lunch today, so that was a bonus.
Today's my niece's birthday also.
Oh, it is? How old is she?
I don't remember. I got 5 nieces and nephews.
Just gotta keep them straight.
I don't keep their ages straight.
Are we talking teen?
Not uncle.
Yeah, lower. Almost 20.
Once they get to this waypoint,
Okay, so just hang on here.
yeah, then the next waypoint I'm gonna do is north.
What grade is she in?
Would it be an inappropriate time to grab a sargassum, or would this be?
No, it's fun.
Time flies.
It's your birthday.
Ah, it's my birthday. I would love to grab a couple of those sargassum
Sure, bike ride.
Samples.
You want them in a quiver? or
in
you
a
want them-
slurp would be
in a
fine.
slurp?
Yeah.
I'll get it stopped before.
Don't.
Is this SARS 6?
This would be Sarge.
I don't remember my own age.
Let's see. Let me look at my sargassum log here. This would be. This would be Sarge 5.
Oh, Sarge
So
5.
once you reach that waypoint, it might just be minutes before I start moving you north.
So we're at about 340 meters here, and we see a lot of patches of sargassum.
Do the white balance real quick, Deb? Yeah. So you've done this before, right? Just look at the arm, press the back, zoom in as much as you can. It's okay if it's not super focused.
I wanna get it really light, right?
So we're doing a white balance right now.
Okay?
We're focusing in on something that's white on the arm here. Oh, there's a fish.
Trying to figure out where I was going.
A lot of that's not really. Looks like grab the shirt where you can speak up on it. Kind of skittish up here, but you. All right, prepare to start.
So I don't know if this helps, but yesterday what I think J-Rod was doing was, well, I don't remember how he did it. Didn't he like pick it up first or something?
Picked it up, set it in front of the slurp.
Yeah,
And then
yeah,
slurped it
do
in.
it with that, they just, they left that on the porch, they grabbed the piece, and then I just started sucking in, so I'm just trying to straighten it out, if that makes sense.
Yeah, so it's not all bunched.
Yeah.
All right,
That seemed to keep it from clogging.
so put this back in here then.
Yeah.
Yeah.
Alright. Try and grab that piece right there.
Well, I can log the SARS sample. Or you restarted it?
I haven't started it yet.
And
Okay,
I'll get to
Start
crazy.
5, right?
What you call it, 'cause they're in
For what?
the, biopump.
That's pretty much what I'm gonna do.
Okay.
Probably it's a bigger one.
Yeah, does it matter what size it is?
If we could grab a couple of them, that would be great. Like maybe this one, and then there was another one up. Well, there was quite a bit of them. Whatever is easiest. They seem to be all over the slope. Oh, there's another little fish.
Is it a tripod fish? Get long fins.
Is this not I know we're all having trouble pronouncing it.
It's okay.
Very nice to meet you.
Nor do you.
Ah,
That's why, yeah, we're all like, we all keep going, like.
La Paguera.
Paguera.
So do you ignore the R in front of the G as well?
Yeah, kind of.
Paguera.
There you go.
Oh, that was good.
Yeah.
Let that sand fall off there first.
Yeah.
And then this way, I'll turn on the, I'll turn on.
Oh, doing a little shaky,
suction
shaky.
sampler.
Look at all that sand.
What's it say on the bio box?
Oh,
Oh, it's.
What could that be?
It's related to
It's actually leftover from previous dives.
Yeah.
Somebody have a birthday today?
Partyville?
What's stove on
So what, yeah, what does Paguera mean? No?
It's like snapper's place
That's-
because yeah, like the fish.
Fish, yeah.
Because barnacle is snappers is barnacle.
Okay.
Yeah.
So it means like the snappers?
It's
Okay,
snappers
that makes
place or
sense.
something
Oh,
like
it's
that.
yeah. Cool.
It's the snappers on fish?
No, like,
Yeah.
like red snapper.
Oh,
white
red snapper. Oh, I
white
see.
snapper.
Do is, yeah, I can, if you wanna pick up like a porch out. Yep.
It's like a fishing spot.
Yeah, look ready.
Mhm.
Around.
Sort of like in California, Los Gatos, the cats.
Yep.
Hey, look at that on.
Happy birthday, Colleen! Woo!
What about, can you rack out at all, or what's on the rack out?
Maybe you can see better now.
Is that permanent Sharpie? That'll be there
Permanent.
forever.
It is there.
Absolutely.
It'll never come off until the next birthday.
Yeah.
Oh, I just realized that with the transparent lid to the bio box, a lot of the bio boxes are not transparent.
Yeah.
It really helps to see what's going on.
Yeah.
And, yeah, right.
yeah.
So you can
These are
see
the
what's
best
going
bio
on.
boxes on
But
the
also when you
market.
go, oh, is there anything in that one?
Before you open it, let it out, yeah.
Right.
I think this looking for it showing up.
Come on, get in there. Oh, there it is! Yay!
Canisters.
Which
Stop
chamber?
it.
One?
Chamber 1,
Didn't get you
yes.
nothing.
Your birthday? Or is that chamber 8?
I think that's 8.
Oh,
Chamber
okay,
8.
Chamber 8. Thank you.
Happy birthday, Colleen.
If there's another one nearby, that would be wonderful.
Sorry.
Best birthday present
Best birthday
ever.
present ever. Rotting seaweed on the seafloor.
Yeah.
There's an absolutely huge one.
A gift to give to another
I
researcher.
don't want to fly that.
Yeah. Yeah, that you're not even getting to yourself.
Oh, there's a couple right below you there.
Yeah.
Colleen, would you consider this a separate sargassum sample or just the same?
Do you want it in the
These two?
same jar?
I think in the same jar is fine, yes, thank you.
So they're both.
They'll just be both Sarge 5, yeah.
Because we got them yesterday, we got 2 search boards.
Nicely done, Zach.
Definitely good flying.
Surprised it's holding up so well. Perhaps without.
Alright. Is the suction on or off?
It's on, it's on now. Just trying to get you up out of this, all this trash, so we can see.
Somebody's throwing sand at
I
it.
know. Not cool. Nope. See. Come ahead. Somebody's shaking sand.
Pulling the lowers might help a little too, I think. Yeah, there we go.
I think you're there.
Paul?
There it goes.
Did it get in there?
Yeah, I haven't seen it yet.
There it is. All right.
You need the
Alright,
slurping
Zach.
sound effects.
Yeah, you can go
The
ahead.
slurping sound effects,
Shut
definitely.
that off. Go ahead and switch the jar.
I am
Those
still
are both in the
ish.
same chamber. Both in the
Switch
They
the
are
jar.
both in the same.
Thanks.
Yeah, that's fine. So slurp 8. Wonderful, thank you for the sidetrack here.
Are you guys prepared to start moving north to Atlantis?
Yes.
To Atlantis.
There is a
Upward.
There is a theory.
Onward and upward.
Puerto Rico and Hispaniola were Atlantis.
Perfect.
See?
All right, we're starting to move north.
There's another theory.
So
that's
just to
also
remind
very
everybody, we're
regular.
collecting the sargassum for some colleagues at Hui and beyond who are interested in understanding the fate of sargassum.
Because it, was out of the water, much of it.
So we
was
know
out of
it
the
moves
water.
across the Atlantic. We don't really know where it all ends up. Some gets all the way to the coast of
I
Florida
always thought that.
and washes up and degrades on the beach, but then there's a lot that doesn't, and we don't really understand yet where it ends up and how long it persists at the bottom of the ocean. So we're trying to figure out how old this sargassum is and try to ultimately understand where its origins were as well.
The sunken harbor in Carthage used to be this had like this
So I'll bring these samples back to
that
a colleague of mine at Hui, and he will be sharing with the colleagues that are part of a bigger project or bigger group that are studying kind of this large sargassum belt.
Basically, the only thing I really want to know is it basically is solid
trying to
water.
interpret what it
So when we go on oceanographic cruises, we try to help colleagues out as much as we can. So collect samples for other people if there's interest. You probably heard us over the last couple days grabbing samples for colleagues and friends at other institutions who couldn't come on the cruise. Colleagues at the Smithsonian, for instance. Also for museums, if we find a species of coral or sponge that we don't quite know what it is, we can collect some and they can identify it and then have it as part of their collection. So it's a wonderful community where we're all kind of in it together. We want to help each other out as much as we can and really leverage, these great opportunities where we get to be at sea.
Four?
Fine.
Can I do three so I can catch up?
Oh, yeah. Chris, did you get up?
Yeah.
Okay.
Are you just trying to get it out of the way there?
Yes.
Yeah.
You need to come all the way to the left. then.
When I go left, it sticks out more.
Well, okay, so typically what they were doing was like wrapping it up like this. Kind of took it in.
This way?
Yeah, down, yeah, like pitch in. Yeah, pitch in, and then roll it, the way you were rolling it.
Like that?
Yep, and then just bring it like across through the yaw as much as you can.
Yeah, that's all I got on that.
Okay, might have to roll it down a little bit more. Maybe I just gotta stick that out more, because they were like off to the side a good ways.
Yeah, okay. How's that?
Alright.
Those? Fine.
Just gonna pull this ram out. Alright.
There's a lot of,
I just did a screen grab just to show that like the sargassum isn't actually forming like big mats on the bottom. It seems to be like kind of individual little clumps. There's might tell us a little bit something about how it sinks. If it's not sinking as a big, like on the surface, it's a large mat of sargassum all together, but it seems like we have these little clumps all over the place.
Maybe it sinks one bit at a time.
Yeah. All right, we're starting to head up. We're at about 320 meters. How is the current?
I don't really feel much of it right now.
Good.
yeah, I think.
Current bad.
Y'all were saying it was a lot last year, huh?
There was a lot of current the last couple days. Oh, look at that.
You were saying that technical divers have been here,
Not down. Yeah, to some of the upper mesophotic regions. Yep.
so they wouldn't like to dive if the currents were that bad, would they?
True. Yeah.
I wouldn't think so.
Oh, drift dive. Oh,
A drift dive. Yeah.
no. It wouldn't take
Those
you,
scare
wouldn't
me.
take you along the ridge, right? It would take you off it.
Down and off.
So it's like a drift drop dive.
This might be scared of land. Who we here? Because I feel like we just like easily get swept into the current and we can go over to like the ferry and look at this big.
Have you been diving?
No, I'm
What
really
is this?
scared.
That snapper?
Oh, really?
More sargassum. Big chunks of sargassum here. Wow.
I bet we'll see more and more as we move up.
Yeah.
That's a snapper.
Oh, looks like a snapper there,
La
huh?
paquera. La
Pargo.
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 La paquera.
That's a nice.
Eric?
So as we continue to move up, we're expecting, at some point we're going to start seeing more corals. We're going to hit the lower mesophotic reefs. We'll likely see more kind of octocorals in the bottom, transition more to scleractinians as we move up. Of course, sponges. We should change this to the sponge and sargassum cruise.
Yeah.
Those little tiny ones, little tiny sponges all over.
Well, I wonder if Jen would be interested in doing the trace metals on the sargassum. I don't know.
Yeah, we're gonna look at the arsenic in there, but the trace metals would be cool too. And we can do some imaging. So where is the arsenic? Is it getting into vacuoles or
Like the bladders and
what's that?
those bladder things.
Yeah, or like in the vacuoles in the tissues.
Oh.
So yeah,
Where is
like
the
are they
ar-
being stored? Is the arsenic stored?
Where does the arsenic originate from?
There's arsenic in the seawater, but it's really low. But
But the sargassum concentrates?
yeah, they bioaccumulate. So they just, because they, in areas where there's low phosphate, the arsenate anion looks just like phosphate. So they'll start pulling it up and then they accumulate it. Whereas a lot of other organisms will kick it back out. Some organisms bioaccumulate.
Is it harmful for them?
They seem to be okay, so they must have. I don't know enough about it, but a lot of times if there's some sort of harmful metal that gets taken up, it'll get sequestered into a vacuole. So that could be the case here. But I am definitely not a sargassum expert. But
So like when there's low phosphate, they accumulate more arsenate.
yeah, so and that happens for lots of other organisms, plants, phytoplankton, because the transporters on the membranes that bring phosphate into the cell will get tricked into thinking that arsenate is phosphate. And so if they start getting starved for phosphate, the arsenic will just kind of get brought in instead. And so it can start accumulating.
Actually, I don't know if you answered this question already. There's someone saying, have you seen any organism eating or interacting with the sargassum?
That's a good question.
Oh, we haven't seen anything eating the sargassum here, but
Not at depth, yeah. But when it's floating on the surface,
I-
there's usually a lot of fish and things associated with
I
it.
bet they would.
Cover? Is that just because it's covered?
Yeah,
Not
yeah.
because they're not eating it.
I don't know. They might.
Yeah.
I think there were like bryozoans growing on the sargassum. I looked at some of the sargassum they had brought up. It looked like
Yeah.
there were bryozoans on.
bryozoans on it.
Yeah, crushed.
Yeah.
So I don't know if that happened after it sank.
Yeah, I was just gonna ask whether that was at the surface.
Or that happens at the surface too.
Yeah, I have a colleague who has an aquacultural farm where they're growing seaweed, and they believe a manatee came and ate all the seaweed that had been hanging on their
Went to the
lines.
salad bar.
Definitely went to the salad bar. So if the I imagine the manatees would similarly just eat the sargassum. I don't know if they're picky about their type of seaweed, but
I bet they have some tastes.
Well, I heard a story about a manatee that came in the quite far when there were floods, and they're just munching up the residuals.
oh yeah,
We got our first coral.
oh a coral,
Soft coral.
little soft
Right.
coral. All right, we are now at 280 meters. We're still cruising up.
This is so steep, we're pretty much just recovering
We should just going straight up.
Just go right back up.
And if anything eats sargassum, they have a field day this year.
Oh
The
yeah,
past few years.
Yeah.
little fish on the left there.
Fish look like little geckos.
Yeah.
These house geckos.
fish and sea urchins eat sargassum.
Oh, okay. Is that in the chat?
No, I just looked at it.
Oh. More little sponges. We're now at about 270 meters.
Oh, some people use sargassum in tea.
Tea?
Yeah.
Really?
Sargassum.
Huh.
Salted tea. There's sassafras tea that would take sargassum.
Like a herbal remedy.
Okay.
0.5 grams dissolved in warm water.
Interesting.
Oh, there's amphipods, shrimp, crabs. These are a bunch of listed things you can find living in the
In sargassum?
Sea spiders, shrimps,
Really?
amphipods, crabs, amphipods,
Okay. So it's a nice little habitat
Yeah.
for.
flatworms, annelids, nudibranchs, snails, squid, a bunch of kinds of fish. There's something called sargassum fish,
Sargassum.
Sargassum.
Oh, is that a fishing line there?
triple fin, eels, sea turtles, sargassum. Oh my gosh. Oh.
Well, we knew that it was not going to be very exciting until we hit the lower mesophotic region. So,
What's the depth of that?
well, it varies depending on where we are on the wall, but starting about 200 meters or so, maybe 250 in some areas, but it could be shallower too. There's quite a bit of diversity along the wall.
There's another species of sargassum that they eat in Japan for hundreds of years called hijiki.
Oh. Hmm.
$2.54.
254. So we're just almost going just straight up. Starting to get a little rockier, maybe. Or maybe that's just wishful thinking. Is that a little anemone there?
Yeah, that's what it
We're starting to see some life here.
Yeah.
might be like a
Woohoo! Anemone.
Is it anemone or there's some weird corals that are like one giant polyp with like a big calcium carbonate plate underneath? They kind of look like that.
This looks like a different version of the one we see in the Gulf. There's
Is that puff
purple
of
one.
sand coming out of that hole?
Huh.
Above and underneath? Something small,
Little
Oh,
crab.
yeah.
Oh, yeah, right there.
yeah,
Something living in there.
not so under the bright light. There's a little shrimpier crab.
What was that cup coral above the anemone? I knew cup coral. Solid.
Yeah, there's a little soft coral, a little cup coral right there. All right, we're getting there. Ooh, what is that?
Oh, that's cool.
That looks like one of those little balls that you play with.
Or a sponge? Yeah, it does.
That's a laundry ball.
Yeah, laundry ball.
Like a dog chew toy.
That is so
I
cool.
think it's an echinoderm, right? It does look like tentacles.
Look
Nice
at
and
that.
I don't know if it's an anemone, weird anemone.
Very weird one.
Or,
Wow.
yeah.
Yeah, it looks like an anemone.
Did we discover a new species? Can I? Oh, come on. Birthday
Colleen
species.
ansilensis.
Insolensis.
That's Hershey's.
That's interesting.
That's really pretty.
Tentacles.
Yeah, neat.
You thought that schmutz stuck to his tentacles, or
Or symbionts in there?
sometimes, yeah, that's kind of what symbionts would look like if they're in there.
Oh,
Oh, it's pigmentation.
Oh, look at that.
definitely an anemone.
Neat.
Yeah.
Woo!
Okay, paying me, I don't think it. Yeah. 'Cause it's really, it's tough.
You can brace yourself on the wall if you want.
Still worried about
That's
that.
okay. We can keep going. We're just like we ooh and ah.
We got birthday mission today to get to the.
Birthday mission.
Birthday mission.
That's climb to the top. Climb to the top. That's a good title for today's stream. What was that?
Virtual level.
Turn off the live stream.
Excuse me.
240.
I got a little yellow sponge there. 240. This is so steep, huh?
Santiago played a trick on us again.
Yeah. You can actually go to Outback. Yeah. It's really nice. Really nice.
What's mud in this? What's mud in this?
Don't lose faith. It's gonna get nice.
Nah, this is gonna be good. 2:30.
We're just having to show some patience. Wait for it.
That's funny. 'cause
Wait
while
for
I'm
it.
writing, I'm like, one of the things I'm like, it requires understanding fluids, engineering, optics, chemistry, and patience.
And patience. Yeah.
Resistance.
That's not one of my strong suits. I want the corals now. It's like the Willy Wonka, whatever.
What did what?
Yeah.
Is that Veruca Salt?
Yeah.
That's the name of a band, right?
Yeah.
Yeah. That's cool. I still think Dirty Marshmallow would be a great band name. Scott, you should pitch that to your band.
I'll try.
Bird marshmallow.
Or
I like dirty
I like Dirty Marshmallow.
I don't like where that probe is. It's gotta be inside
Ooh,
first.
triggered a little fall.
Uh-oh, what happened there?
It's just the thrusters.
That close.
Oh.
Prop wash.
They got nothing to move for.
Is the background rising?
It's not pumping at all right now, we're just inserting
Oh, there's another one of those cool anemones.
Ice is just,
ambiently, yeah.
Probably
Let me
temperature.
look at this
well, yeah, none of
guy.
those weird.
Yeah.
Pitching.
I want to get like a little screen grab of that.
But PMT is on, right? Those are actually
No,
counts.
that's.
Yeah, PMT is on.
Towards you.
It just tends to drift it slightly
There you
over
go.
time.
There you go.
It's
Pitching.
happening.
Be efficient. Yeah. Then you come down when you're out there.
Things are suspicious, because they have like, really white towards the center, almost like they have
And then go ahead.
skeletons on the inside.
Yaw down. Give me the wrong direction. Yaw. Not a problem.
Well, we're at about 220 now. Moving up.
Version of which should be safe, like scallop shells or scallop tentacles. Just like, you know, they had their out front. They also have
Still,
they
the
had
iron still was out front. Brushing it.
Blogging.
put eyes on the edge.
Oh, working on it. Nice.
At least have an outline.
No, no, there's the tentacles. You know, eyes
So
on
from
the
the
edge.
400 meter start, we're about halfway. If it's the same over here as it is to the left.
You wouldn't even imagine something that weird.
Can we go that direction, or are we just
That's right here.
going towards the left towards the finish part there, or do we
Right now,
need to
we're just
avoid
gonna
that?
be north.
Yeah. Okay. And,
Is that,
no, that's fine.
Yeah, okay.
is that second green dot north, how deep is that?
50 meters.
Oh, okay, so we're like halfway to the,
Yeah, we're like 100 meters laterally from the 50 meters vertical.
okay, cool.
And we're at 220, so it's gonna be super steep this whole way.
So I guess we don't have an issue colony with sampling corals deeper and bringing them up where it's warmer.
Yeah. Right.
Oh, what's that?
Those little ones?
Big hermit crab coming up?
Oh, yeah.
Oh yeah. Look at that.
Oh, wow.
Wow. He's just like hanging on.
Yeah.
Don't slide
It's like
down.
those goats on the mountainside.
Yeah.
Can they swim or?
Oh jeez, he's gonna tumble down.
Yeah.
Yes,
Wow. He is living on the edge, literally.
Kicking the sand down. He's, oh, don't slip.
Oh, hang on. Hang on.
Can we rescue him?
Rescue.
Yes. Him for a ride.
Good luck to
That's
you.
a stretch for that word.
He's living his best life on the edge.
I was just gonna say, he's like, I've worked my entire life to get down here. This is so amazing.
A little adrenaline junkie.
You really love
Yeah,
the
I'm pretty sure if we got here and with that claw, we just would jump off.
Yeah.
Yeah.
But he would retract.
Remember the little tiny hermit crab that was climbing there?
So at the end of
What was he
one of
climbing the other day? Just like the edge of the claw? Or
Oh,
2
yeah.
days ago? It was like Mission Impossible.
And he jumps.
It's funny how slow it can fell. I was surprised about that. Sometimes when I have like snails in Aquaria, and they can, I didn't realize sometimes how buoyant they can be to like keep gas in their shell.
really?
To keep them like nearly mutually buoyant.
Yeah.
So like they'll climb on the bottom surface of the water, just hold on to the, using the surface tension of the water. And they'll just go like this. But like I've seen them like drop off something, and just kind of like really slowly float down.
Yeah, I've
Like,
seen that, yeah.
seen them like fold
You
their
think,
foot
do
and
you
just,
think they should like just go?
yeah.
They just like. You see them like use their foot to like glide.
No, I haven't seen that. They're like surfing. I've seen them like hanging from like the thinnest thread of algae. Like they walk to the edge of a plant in my aquarium, like hanging on like the thinnest thread of algae, and they like kind of fold their foot in half like a taco and just barely pinch onto it.
Oh, wow.
Yeah, I think I got a picture of it.
So we're at about 200 meters now. Getting steeper.
190. Wow.
Yeah.
This looks like it's one.
We're getting close to that, crest of the ridge. Well, it's definitely getting rockier.
Listen, this guy's one of the times I caught him just barely hanging on.
Great, great job.
I really like that. I could do flames, probably more.
180 meters.
Starting to see a bunch of little whatever.
See
Are those
fish.
fish?
Yeah. Yeah.
Getting alive.
Starting to
I'm gonna say
get a little different
140.
here.
140.
So we're at 180 now. Little tiny fishies.
On first coral,
First big sclerectine.
how much money you put?
Oh, are we gonna make a little pole here?
What is that?
A pool?
Betting pool.
A betting pool?
What?
What is that little thing moving right there? Is that a
Oh,
lobster-y thing?
Caribbean lobster.
Right there?
Yeah. What's that called? A Florida lobster?
One of those weird round ones.
Yeah.
Round ones?
That don't have like big claws.
Oh, a slipper lobster?
Is that what they're
Slipper
called?
lobster.
Oh yeah, that looks like a slipper.
A slipper lobster?
Slipper lobster, yeah, they're like really blunt faces.
Yeah.
Gonna be
Huh.
slipping.
Oh
Skunk
yeah,
with
look at that.
slipper?
Yep.
Cool.
You look up
Yep.
the slipper.
Yep. Slipper lobster.
Yeah, the ones with the blunt faces without claws are slipper lobsters.
Oh,
The
not true
ones-
lobsters, but are
the ones
closely
without
related to
claws that
spiny
look
lobsters.
otherwise like a lobster, but with big long antennae
Yeah.
are
Very cool.
spiny lobsters.
Spiny. Okay.
Huh.
Deca.
Alright, that could be a band name too.
Slipper lobster.
Slipper lobster.
Sure.
The Boston version.
I think it must be that. That's cool.
Oh, he's trying to get through this terrain as well. That's a pretty cool shell there.
Wow.
Yeah, very neat looking.
There's two teeny little.
He's filthy.
He's filthy.
He's
He's trying his best.
Jeez, Sam.
Tough crowd today.
Yeah, exactly. Clean yourself, will you?
Birthday party.
Trying his best.
Have some respect.
Look at his eyes.
Oh, look at
Those
that.
eyes.
Yeah, those are nice.
Yeah, cool.
He's just vibing.
Oh, so neat. Man, that's tough. Look at this terrain. It's got those little legs.
It's like a tank. It's like a tank lobster.
That's a
Or
real
like a
life.
really toy lobster.
That's a real life Pokemon.
For like Pokemon. It's a real life Pokemon.
That's a real life Pokemon.
Do you load Pokeballs up on the ROV? Throw it out?
Fantastic idea.
Like what?
Look at his eyes. Those are crazy. Oh, he just tripped. You're all right.
That'd be cool.
This like matches this,
Wait, would we
you know?
Would we get a cease and desist letter from Nintendo?
Probably.
Oh.
Uh-oh. Oh, don't fall. Don't fall.
We're going into dangerous category.
Before he falls.
Yes.
That's so cool.
Wow. Very neat.
He's like twitching the antenna, like the front.
He's waving to us. Hello.
slipper.
Oh.
Spanish lobster. That's what I've heard.
That's what it's called then.
Oh, it's
Yeah.
a Spanish lobster?
Yeah.
It is.
Yeah.
Yeah, that's how I've
Ah.
heard.
You can tell by the
Oh,
accent.
Spanish slipper lobster.
Spanish
It must be a type of slipper lobster.
Well, our J-Rock loves them.
Oh no, no, don't fall. Ah!
He knows what he's doing.
All right.
Okay.
It's a very steep slope.
Slipper lobster. Oh,
Oh.
he's leading us to the corals. Right there.
Oh, look at the little corals.
No, the cool.
What a nice guy. That's why the front of him is so dirty.
I don't know
Nature's
if
just
the
like, you know what, we don't have a bowl
Nature's like, we need to pull this. With two pans on each side.
Slipper lobster is the common name for the family. Spanish lobster is, I guess, the one we find out here.
Did we get a screen grab of this guy?
Yes.
Yeah?
Move
Awesome.
on?
Yes, thank you. We could watch that guy all day though. He's pretty neat.
Pretty cool.
We need a dedicated slipper lobster live stream.
He could be our ship pet.
Should probably-
There are plenty of coral pets.
So at some point this afternoon.
That's a good idea. He does give me
Look those
Frodo
guys.
vibes.
It looks like he's on
I
it.
think that's gonna be the easiest thing for us to do.
All right, we're moving on.
Like,
It's getting rockier. I'm starting to see what looks to be like crustose coral and algae there, right? The pink, possibly.
Where are we at?
We're at 170,
Yeah, there's a little patch.
so we're starting.
Here.
Do you see the
Oh yeah, I see it.
see patches here?
170 meters.
So we may be hitting to where there's a little light getting down here.
Whip coral. Oh, we're starting to see some whip corals.
Whip coral starting.
Oh, and some
Can you tell that we're
something else right
excited?
there. Coral or something. Yeah,
Is that one of those sponges?
What is that a sponge down here, hanging here?
I'm not sure. It might be a coral.
Or there's some corals here.
Oh, those look like maybe black corals. You zoom in on some of those
Maybe
twisty,
this-
branchy things on the edges of the rock.
Like this here.
Yeah, I think those are probably black corals.
Let's see, so we're at 1828 UTC. Starting to see some more corals. About 2:30 local time.
Oh, there's a couple.
Oh, cup corals.
Yeah, I think those are probably black corals.
Nice.
You guys wanna move in on it?
Yeah,
Just- Just
Is that
kinda, do you want to get a closer look? Or are you happy with that?
I'm okay with that.
Okay. There's this yellow,
Yeah.
yellow
There's also
coral up
black
here
coral.
too.
Started seeing them at like 160 meters.
All right, so we're starting to
Yellow.
get a little more activity.
They whip coral.
About 170 now.
All right, happy to carry on?
Yes, please.
All right, I'll leave that work.
Couple of little bit.
Yeah, that's a nice one.
Really like air. Remind melting you around. So.
Wow, that's a pretty big change in topography.
Yeah.
Look at all those apartments though.
Yeah.
Yeah.
This looks like this
Little
looks like it was
caves.
once an old coral reef that's like slowly
Yeah.
eroded.
This, yeah. At lower sea level stand, this looks like bioerosion at the shoreline.
Yeah.
You see this along the Gulf.
This is really neat looking.
At this
Some-
depth, it's a
definitely
little
some
bit-
corals
It's a little bit
un-
deeper than the last sea level low and, but.
It's interesting how they're all on the underside, right?
Subsided.
Avoiding the sediment.
Whatever comes over the top, yeah.
Yeah.
The coral, oh yeah.
The
underneath.
corals are all on the underside, like
Yeah, black corals. Yeah, it's
Hmm.
And then carried black coral like they hang over the
I guess that's a
over the
void being sedimented. Yeah.
Or the ones
Or
that are-
stepped on by
or the
scuba
ones that
divers.
grow up- the ones that grow on top don't last.
Don't last, yeah.
Yeah, this looks a lot like shoreline, you know,
You see some daylight.
subaerial erosion.
Yeah.
Ah, okay.
Carbon
So, all right. So we're starting to see.
Looks like that.
We're at
160.
160.
Interesting we see some daylight, but not quite a lot of CCA yet.
Yeah.
Maybe it gets this too much sedimentation in CCA.
Is that daylight right there? Is that what you're talking about? Yeah.
Should we be able to put, that camera that's just blue water in the hangar door over there? So we can see if the floods come up.
More whip corals.
Yeah, the curly Q ones are black corals, and the ones that are just kind of irregular lines are aqua corals.
Ah.
I think we see both right there.
What is? Oh, is that a shell here? There.
Yeah, look at that.
Snail? What did you call the SOP?
Wow, look at that. Thank you.
There's quite a bit of CCA starting to pop up here.
Yeah, a bit of it, yeah.
He's an
Yeah,
icon.
that pink crustose coralline algae. It's a nice substrate for coral larvae to settle on and grow. Starting to lose our sponges. Well, there's a yellow sponge, I think, over there, but not.
Are those bars like something blocking the light?
Yeah, that's when we turn on our upper floods looking up.
Bar gets.
Oh, I see.
So it's just if we're looking up, you kind of have to turn them on, but they make some weird shadows.
155.
155.
Would it be like when the MCLA pump on first, then background water, so we get one bolus?
That one, too, that one, that one.
Is that yellow kind of an encrusting sponge, you think, Scott?
I was wondering about that.
Yeah.
Yeah, it kind of looks like it, right?
Oh, is the Chick-fil-A sauce a sponge? On the arms?
Chick-fil-A sauce, I think, was a colonial city.
Yeah, you can even put a couple
It
of
could've-
parts just so
it
we can
could've
see it,
been-
we get a shallow spot up.
Chick-fil-A sauce, not
Some of these things that are colorful, just like, I guess, some acidics too. Sponges are acidic.
Starting to see more color. This is getting exciting. Really cool structure here.
150.
Rocky structure. Looks like a
It's
old
invasive in this.
shoreline.
This is?
Down here, or
Yellow
It's
The-
a
what's
white.
that?
It's
Are you
white.
talking this
Yeah.
yellow? There's a lot of like what looks to be encrusting.
If you want to zoom in, you might be able to tell.
What did you want to look at?
On
Oh.
that yellow.
If you find a good chunk of yellow and you want to know what it is.
Do you mind if we zoom in on some of this yellow?
This one over here.
Oh, there we
There's
go.
one right there.
Sorry. Ooh.
Sponge, probably.
This looks really irregular. Which one seems that? Maybe sponge. It kind of comes up and has maybe some little oscula. So I would say sponge.
Yeah. There's a coral over here.
Oh yeah, look at this coral, Sam.
Sam, there's a
Chicken ya.
Is it coral?
coral right
Yeah.
here.
It looks like it's
We're-
probably black coral.
We're
Can you get
right directly on it.
it?
There's no current to knock it over like we're normally seeing.
Yeah.
Look at that.
You might-
It's like the black coral, like handicap things. Like if you want.
So that's a nice little
Push my stuff to the dryer.
a black coral of some sort.
Black coral, maybe
You can put
a fan of like an antipathy slot.
Do we want to stop and measure or anything?
No, let's just keep
Okay.
going up. I think it's gonna get more.
Yeah.
All those really cool like curly Q whips here. Those are neat.
143.
This would be really fun to like explore, you know? Crawl around in all these holes. If it wasn't underwater.
It's so eerie, like see the sunlight coming down.
Yeah.
It's almost like we're in a cave.
somebody asked this was limestone.
There, in this region, yeah, I would guess so, just by how it's eroding, but I'm not positive.
Ooh, what's that on the top here? Is that some
Looks spongy.
spongy?
See some coral just down there.
Yeah, some sort of coral on the bottom left there, Nicole. Do you see that one, the white one?
That looks nice to see. Looks maybe like a square chicken coral.
Oh, okay. There's a black coral here right next to that spongy thing.
That looks like the cram patties we saw yesterday. Maybe a little less bushy. Maybe there's a cantopathes. Yeah.
Wow, cool. Yeah, the
Definitely.
ground cover's getting more active.
Still a lot of sediment here, huh?
There is,
I
yeah.
think once we get over the lip.
Yeah, once we get onto that crest,
Yeah.
you know. Really neat looking terrain. This is cool.
Just yell at me if you want me to stop. If you
No,
can.
you're good.
Just slap him.
J-Rod would just go, stop. Let me fly it around. Huh?
Yeah.
He was like, it's just 'cause I was panning as he was trying to move, and he was like, wait, you can't, sometimes I guess you can't, I don't know, see exactly.
It does kind of make the thing crash.
Right.
Yeah.
So it was just for a particular moment. I
As
think
long
he
as
was-
we remember to use that one, you're good.
He was like, don't pan, just zoom in and out, don't pan. And I was panning. He was like, stop.
And I'm like, okay,
Because he'll chase you.
Yeah, exactly.
He thinks he's moving the vehicle.
Exactly.
You're moving the camera.
Right. So that's what I was saying. You can just tell me, quit, stop, Scott.
back up my mind whether there was a made up feature.
Colleen,
Yes.
you may have already answered this question. I was
Look at that.
It's a big spine. What kind of rock it is?
We don't. I would assume carbonate.
Yeah, nice big sponges.
Probably a limestone rock, just the way that it's eroding.
Yeah, that's
This
what
is very, very kind of common in how limestones erode. Whole features, kind of really sharp erosion patterns. Well, that's a crazy sponge. So that would be my guess that this was maybe like a old shoreline that eroded. Now I can see the chat. Got it. Yes, this does look like caves, doesn't it?
Yeah, which is often often limestone, right?
Yeah,
Usually.
it's very, very cool. A lot more CCA here. A lot of growth like underneath the overhangs, just kind of.
It's like survival of the fittest, right? It's like whoever decides to grow in the best place
Yeah,
doesn't get wiped out when the sediment comes down.
right.
Sponges lime. Oh, lime or CCA.
Yeah. A lot of color now coming in.
A lot of little things.
This is upward looking?
Correct.
Yeah. Kind of pitched up at an angle.
Yeah, it's like that, so I
Yeah.
can see if I'm gonna.
Bang your head.
Yeah, basically.
What What kind of scleractinians do you think we might see? Agaricea?
Yeah.
I think Agaricea is going to be a big one, and this is going to be a scleractinian coral we'll see very common in this region, and it has a really large kind of depth range that it can exist. But what it'll do is when it, oh, there's a nice coral right there.
That looks like a black coral. Orange tissue. Pretty cool.
With the Agaricea, what you'll hopefully see today is that in the deeper waters, it forms more like plates. So it's kind of increasing its surface area so it can acquire more light in these kind of lower light regions. And then as you get higher into the mesophotic, it'll become more like bouldery kind of shapes. So really cool how corals adapt to their environment.
That's actually an octocoral.
Is it?
An octocoral here.
No, I guess a plexaurid. I don't know. One of the two.
Me throwing around the jargon as if I have.
But it doesn't want anything? This little?
That's really cool.
It's all been like a big cliff. Yeah,
Just like hanging off the cliff there. All right. It's wild. This is definitely more irregular than I was expecting. I thought it was just going to be kind of a sheer wall. This has a lot more kind of rugosity to it. So we're now at about 120 meters. Oh, could we go over and look at this right here?
Oh.
That's starting to look more like,
Like a Porites?
If that's a Porites, I will be
Crazy.
happy.
Or is it a
happy. But it
sponge?
might be a sponge, and then I'm going to be sad.
Oh
No, maybe we can move on.
no. Don't be sad. Yeah, I think that's a sponge.
You think?
Yeah.
Oh wow, that's playing tricks.
That's a tricky sponge.
The encrusting one or the rope?
No, the one on the bottom here has a morphology that's very similar to Porites asteroides, which is
It's like sharpened.
one of my favorite corals.
Oh, these big tubes.
Yeah, a lot of those are different looking sponges than we've seen.
Yeah, they're like hanging off the rocks.
They are, yeah.
Again, it's just hard to say, like,
Oh.
Wow, look at that one.
That's the Aplicina?
It's like facing right at us. That is really cool.
Oh, this one looks like that. Yeah. I guess this might be the same kind of different colors.
Fake news.
a lot more colorful in 2 seconds.
Yeah, it's changing quickly
It's like
now.
Wolverine's claws back there.
So we're at about 110.
That's gorgeous.
110. Bottom of these mesophotos.
We're starting to get these bigger kind of tube sponges, a lot more color.
Really close to the dock.
Drawing in all the time. Bridge, RV, we got one more short move for you.
Wow, that marine snow is looking beautiful.
Isn't that pretty? Yeah, all that green snow off to the right here.
Guys, let us know if you see our floats on the surface as well.
Okay.
Look at all those whips here.
Oh, yeah.
all the way around.
That sponge for clicking his eyeballs.
Yeah.
Oh wow, it's a lot more light going
This is the town
I
highway.
think we're getting over the edge here.
We should be coming up upon the crest. Wow, there's a lot going on this rock right now. There's a lot of things growing. There's a fish here. Oh, we got some. Those
Yeah,
look
there's-
like stonies,
I think those are
right?
still
You
sponges.
think those are sponges?
It's look pretty deep for something like Friday's estuary, 100 meters.
God, look at that though.
Yeah, I think it's a sponge. Well, are those
I don't-
polyps?
I don't know. Well, this, yeah, I think this is a sponge.
Okay, yeah, those are sponges.
But
yeah.
are we sure that these are not? That's a kind of crazy sponge form. Like this, with these
Mm.
little nubbins looks like,
Oh,
you know,
wow.
the Porites to me, the mustard
That
coral.
coming up. See that?
Yeah.
It's just,
Which
just
one
above
is
us.
it? If it was Fridays, you think it would be kind of more exposed to the light, don't you think?
Yeah.
It's kind of under.
Here it comes.
I see him there, gotta look at
Fine.
Okay.
Whoa, that is a
sponge.
huge sponge. Wow, neat.
Oh, oh my god.
That's neat.
That's volcano sponge.
Volcano sponge.
Look at that.
Yeah, that's neat.
Although our other calibrations get eaten up. Feel great about
Let's see, we're at 100 meters now.
Can we screenshot that,
it. Good.
that
I'm
sponge?
trying to get you a good
Alright.
spot.
Do you want to measure it, or do you want to keep going?
Might be fun to squirt some fluorescein around it.
Is this a hard spot to sit down? I know it's pretty.
Find out here in a second.
That's pretty.
Oh, look at the ridge, you can see above the.
Oh,
Oh, there's the ridge.
fish, all the fish up there.
That's so cool.
So we're just over the lip.
So I'm settled here.
Wow, that's really cool looking.
Looks like a candle.
Yeah, it
Those,
does.
those kind of candles that.
Can we tell if it's pumping? Do you see anything?
I can't see anything, but
What do you guys think? Do you think we can sample it really quick, or
yeah.
yeah? No, we're at a weird spot.
Is it possible to turn off the lights for a second and see what the ambient light will look like?
Yeah.
Ooh, we're gonna turn off the lights.
Oh.
Wow.
Ah, neat.
That's still light. Yeah. That's cool.
That's neat.
The difference.
All
Try
right.
putting some fluorescein around that guy.
Sure.
The dye.
Are you gonna want to put him inside of the?
Not inside, on the outside. And it should, in theory, if it's pumping, draw it in and we'll see it coming out the top.
So we're at 100 meters right now. We just turned the lights off. Hopefully you saw that on the camera.
By the way, the handful of squeezers that were in sponges, Don measured ammonium and nitrite in their elevated.
Oh,
So
cool.
there's some cycling happening.
Yeah, yeah, neat.
Yeah.
Okay, great.
Happy birthday, Colleen.
We got these good calibrations.
I'm just happy they didn't say which birthday it was.
What is this? Dive 51
Or is?
0?
Hey,
Is this dive 51?
510.
510?
you are treading on very thin ice, my
What?
man.
I just asked you what dive number it is when you mentioned how old, what birthday it was.
A lady never talks about her age. Right, Lena?
That's correct, I never talk about it.
You You never talk about calling days?
She knows better.
It's a common.
Oh, there's our fluorescein dye.
Oh, there's the dye.
No.
For those of you who haven't seen this before,
So we do have a little bit of current coming wafting.
there is
back
a little
at us.
current, yeah.
So you probably want to, put it, if you can, put it kind of on the upstream side.
So if you haven't seen this already on one of our other dives, this is a non-toxic dye that we can put in the water. It's called fluorescein, and it's used frequently to kind of look at how waters are moving. So you can see, right now you can see it's moving along the screen, so that's the way the current's going. But the cool thing about sponges is they pull in water, particularly like a big barrel sponge like this. They'll pull it in at the base, out on the outside, and then filter all that water, pull out the nutrients, transform the carbon and the nitrogen, and then they pump it out through the hole called the osculum. And so sponges aren't always pumping. They only pump on and off throughout the day and when they're feeding. So sometimes you get lucky and you hit it while they're pumping. Other times, it'll just not really do anything.
That's good. Awesome.
But what you can typically see is if it was pumping, it would pull that in, and then out the hole, we would start
Get some
seeing.
more of it out? Do you want to
Yeah,
pressurize
we
it a
can.
little?
I'm not sure I'm quite in the right position yet, though. I need to get a little more, I think.
Is it you moving the
That's
camera?
me, sorry.
Okay, it's all right. Just thought it was ROV.
Oh, sorry.
All right, that's
Like I said.
way back there.
All
You
right.
happy there? You wanna squeeze some?
Stop touching the camera, Scott.
Yes.
Yeah, I think so.
Yes.
Let's squeeze some.
So we're gonna squeeze a little bit and then watch to see if it comes out the osculum here.
I'm gonna bring this camera back.
Can we zoom in on this top view a little bit so we can see the opening if it's white or green?
I'm gonna try and freeze here too.
Wow, that view goes
Sorry,
straight
it'll
down.
go down, but
Even if I exit.
try.
That's a great position where you guys have right here. If it
Yeah.
was pumping, it would be really pulling it in. So,
It is.
oh, there it
It
is!
is. Look at it coming out.
Yay!
Look at that.
So you can see that streaming out.
It really is
It's
a
a
volcano.
it's a birthday candle.
So
Can
neat.
we do a video highlight?
Yeah, that's really cool.
What time is it?
Yeah.
It is 2:53 local time.
We got confirmation of pumping, so we're gonna do a squeezer here too.
So yeah, so that sponge right now is pulling
can
that
stop.
water
We
in
can-
from the base
Ooh, look at all that.
and then kicking it out. It's really neat.
That's awesome.
So that guy is pumping, actively pumping.
The physical sample.
I would say let's back the let's unsqueeze.
I did.
Okay. And then we can just gently move the wand back and stow the dye.
I'm coming back.
That's awesome. How spectacular.
It's a fun party trick.
A cool party trick.
Yeah.
It is a birthday party today.
Yeah, so that's a little candle there. So yeah, this one is,
Happy birthday,
is pumping.
It's in like all the shots. Happy birthday, Colleen.
Yes, it does look like Ghostbusters. Someone saying the color, the green color. It's
Perfect
kind of,
illustration.
You know when you're happy with the line.
That's really neat. I don't know if there are any other videos of this happening in mesophotocorals. If you look on the internet, you'll see people have done this before in shallow, not coral sponges, and it's really amazing to see.
That's so cool.
The handle that hit the button.
Okay, since
So what
they
that
want
tells
to
us is
sample
that
this, I'm giving this
that
to you.
sponge is feeding right now.
First we want to squeak her.
number.
Squeak her and
We want to
squeeze
squeak
her.
her.
All right, so what we're gonna do now is then try to collect a sample using the squeezers. For those of you who've watched us use these,
Sorry.
Just a laptop, that's all.
Oh my god.
we're
Heart just dropped.
I'm gonna go change my suit real quick.
Here, why don't we put it flat over there?
so we're just trying to put the tube away now. It's on a little T handle. We can kind of holster it, and then it will stop pumping. It's just kind of some back pressure.
With a lid.
$18,675.
Beauty.
So Colleen, since Kate wants a sample for this, we're gonna give it an SH number. It's gonna be 18675.
18675. Got it.
I'm calling it volcano sponge.
Volcano sponge.
Is that what we're calling it?
I'm gonna call it volcano
Oh,
sponge.
I'll save a little screenshot and I'll send it to the chat.
Science group.
Volcano demo sponge.
So yeah, so now what we can do is we can collect a water sample on the outside, and then we can collect a water sample from the inside.
Okay,
What?
Nothing. I'll just stop. It's hard to.
so don't get too comfortable. I think we're gonna do a squeezer here.
Oh, okay. So we're doing the squeezer, that's right now.
So,
matter which one.
what number would you like?
Whichever one's
Take number
easiest.
6 in the front there.
Number 6, alright.
We're
Um,
gonna do it, they skin outside and
I
then,
want. Yeah, I think.
This thing is still needs my
oh, they should
Okay.
shove it in more.
I know 4 of them have the tube in.
Right.
I want one of those, and I don't remember. I'm sorry.
I don't know.
What's the best strategy to have?
Oh, you logging system.
Yeah.
Reorienting?
I will log the squeezer
Yeah.
sample
Okay.
I say
right
we
now.
get the
So
samples
you wanna
first.
try 6, or you're pretty sure 6 doesn't have
We
the
call
Well,
it
when you
SH
pick it
number
up, if it
is
doesn't have the tube, you'll have to put it back where it is.
675.
Try and find one that has the tube. You won't be positive.
Oh my god, I found a paper and I don't have access to it.
Grass.
It's Jeremy's.
That's
It's
a good
interesting, the
graph.
previous attempts were all at night, right?
Yeah.
So,
Describe some
That's what Colleen was explaining on the.
Yep.
What's that?
Yeah.
Oh, we got the
They
pink
don't-
tube on the front.
Pink tube.
Your pink tube,
oh, the weird
okay.
pink tube.
Alright, so that's
We
good.
can try that. So that one has a little, a 90 degree on it, so we're.
If you want, or we could use a different one that doesn't have that, it's up to you guys.
I feel like we'll be able to with the.
I feel like we don't need the 90.
Because there we're
Yeah.
gonna have to try to
Exactly.
position it
Yeah.
exactly
Yeah,
right.
let's
I
do
think
a
it would be
different
easier
one,
not
sorry.
Let's use the
not to
different
do
one,
that.
sorry.
Okay, so numbers. I'm gonna make a note that number 6
Is that 9 or 6?
has
Now you have to try and.
Experimental squeezer.
90 degree elbow.
I feel like it just depends on the position if it's helpful or not.
Easy, elbow.
Yeah.
We also might want
That's
to
the
try to
trick.
close the dye before sampling the water.
Does it not close?
It's pinched off, I can't close it any more than that.
Oh, really?
Yeah. So it's
Is it
unsqueezed
actually
as much as I
pinched?
can.
It's unsqueezed.
It's pinched.
Oh, now that's kind of
With the
stuck in the
ram.
basket there. Yeah, there we go.
Super, super high tech. Pitched off flows there.
Yeah.
So you're stuck on the edge of the front side. So,
Yeah.
alright, so you gotta kind
That's
of
all right. Just
take it up a little bit and drag it back.
Real slow.
There you go.
Cool.
Now work your way around where the
Now
arm
the,
is
90's
directly in
kinda
the way.
The what?
It's just the arm gets in the way when you're trying to look at the top, so then you gotta switch to this view again.
I think the 90's free now. Now I'm trying to rotate it over
There
there.
you go. There you go. Doing all good there. Almost. Might be lined up really close. Nice. Just trying to push it out just a touch. Push out.
I think if I drop it now, I'll check it out.
Okay. Doesn't go, it'll fall forward, so.
It will fall
It won't go,
forward.
it won't go over the side is what I mean. It'll
Yeah.
lean this way,
I think
that's
you can
all.
drop it.
It'll lean into the basket. Alright.
Ding.
Nice one.
Nice shot. You win a prize.
Alright, so,
All right.
I don't know
Which one you want to try next?
You want to try the other one in the front left corner?
Sure.
The numberings come off the top. We'll see when you pick it up.
Nice screen grab, Nicole. Did you do that?
Yeah.
You got it when it was pumping out. It was
Yeah,
perfect.
there's constant screen, like, stills being
A-snaps,
taken that
yeah.
are actually, I think, better quality than we taken.
Well, that's pretty hard to get.
They're really, are they? They're
Yeah.
pretty good. And there's also constant video, so really we're just kind of like logging a section of video.
That's great.
Or,
Two?
straight.
Has a tube? Yeah.
Yeah,
Yep.
'cause we can
Cool.
calculate this velocity of pumping video.
yeah, so I would, I can show you where to access the
So now I'm
video.
gonna hang this in this,
I know,
you
I
gotta
saw
hang it in
that.
there.
I just realized too that we're over like a 60 fathom cliff.
Yeah.
Doing this, so.
Yes, you guys are
When
doing
you drop it,
amazing.
Such as like,
If we dropped it,
We are,
it's not.
This just made me a little more uncomfortable.
Number 2.
I have all the faith in you.
yeah.
so what we're gonna do here now is try to use these squeezers to collect a water sample from inside the sponge in that osculum or the mouth there. So that will be the water that's coming out. And then we will collect another sample just using a Niskin bottle, which is on the back of the RV that you can't really see right now, but it's really easy to trigger, and it'll collect a water sample in the background water. So then we can see what it looked like outside, and then how it's different, what the sponge is pumping after it goes through the inside of the sponge. So we can see how it's transforming the water. A big part of that will be it's changing the carbon. So sponges are known to be huge filter feeders in reef ecosystems. They filter hundreds and liters of water every day, just pulling that water in, changing it, and pumping it back out. So they really change the carbon cycle. They change nutrients, trace metals.
That's what they're designed for, I guess.
So you can see how far away you are and sit.
But because of the difficulty in studying anything really in the mesophotic, we don't really have a lot of information about how much mesophotic sponges are transforming these ecosystems.
Squeeze it a little bit.
Just a little bit?
Yep.
One more? Or that's it?
How's that look?
Yeah.
Is it?
We need the vessel away from us. We should close to so I can take a look at
Okay. And then when you come up,
Try to level it out.
yeah, try to level it out. If it looks like it's falling forward, you have to, just read it a little bit. Whoop. That might be good right there.
Yeah.
Yes, then just keep that jaw.
Perfect.
Keep that jaw like that. Don't, yeah. And then just bring it straight out where you're going. Give you some good views here.
All right, so that's like a syringe sampler. So
Essentially just, I'm gonna stick this into the top.
where the clamp is for the manipulator, called a squeezer, because that when we squeeze up on that topper part, it'll trigger the sponge or the syringe, and that piston in the bottom will pull up. And then on the bottom in the tube, it should pull water up then into the squeezer. These are really tricky to operate because, of course,
Just don't want to get pulled off.
you don't want to
doing this, that's all.
trigger it before you get it into the sponge. So, of course, these pilots are masters.
On the back edge? Yeah.
On the back edge, I can tilt a little.
Okay. Yeah, right there is great. Great.
Squeeze?
Now squeeze.
Come on, squeezer.
We have to do it like the second one.
Come on, squeezer.
You want me to chew on it?
Do another open or not open, close, but
Just slowly start moving.
Come on, squeezer. These have given us a little bit of a hard time. They're really tricky.
try this one. Yeah, number two is not gonna fire.
Number 2 did not fire.
Sorry, team.
So for this one, can I just put it back in the hole?
If you can.
Or does it?
If you can.
You can just set it in the box. Yeah, they are. It's whenever they talk is when it gives a echo. I don't know if they got the microphone close to the speaker or what. What it looks like.
Yeah, we see the fluorescein is still going. We have it pinched and it's still kind of leaking, so we're hoping it's just a little back pressure that will eventually stop.
Full disclosure, I was trying to put a picture from there over there, and then it suddenly turned off. I blame somebody else.
Yeah, you can just hit it in the back. Drop it where.
I mean, I can put it back in that green thing if you want.
So these squeezers are really tricky because what we want is, that's a glass barrel that's holding that piston, and it's really tight because you don't want it to release and pull in water really fast, because then you entrain a lot of the background seawater. So you want it to kind of release slowly. So these were made to be really tight pistons. Yes. Yes.
Can we Solari this one?
We will definitely Solari this one.
Solari.
Solaris.
Good morning.
Thank you.
I think Solari is
Solarising? You don't like Solarising?
Oh my god.
That is so beautiful. Oh wait, we have another birthday thing for coming.
Yeah, so
Oh my gosh, birthday gifts.
Wait, I have to get the iPhone on.
I got them moving 20 meters now. For a RV.
Already great.
My 0 meters due south. I'm doing it 0.2 knots at the moment, that's 50.
Yeah.
It's after 3, so if you want to switch out, once you get that put back in, right?
Yeah, let's get those things.
Wow.
Is that, I was kind of paying attention to, did you say keep them about 150 meters away?
It's that look.
Okay.
It's not looking. I fixed it. I just took it off and turned it back on again.
Okay.
Big bucks.
You know, about 75% of the time that works, right?
And size too close.
Tried.
So if it starts to pull, so once it back to
So we're trying another squeezer. We test these out in the lab before we deploy the ROV. But things don't always behave the same in the lab as they do once you get down into the water.
To be fair, we know that they are an imperfect technology.
It's a tricky technology, yes.
Actually, they're
But we were very lucky that a colleague of ours lent these to us for this cruise.
He actually emailed me this morning and said he usually gets about 4 of the 6 to fire.
Ah, okay. So typically
that's
to
about the odds.
4 out of 6 firing success rate.
Nice.
That's fine. It'll fall over if it needs to. It's squared, so you're good.
All right. Grab either of the 2 in the second row, I think, should have tubes on them. Switch it real quick.
Which one has been successful in the past?
Squeeze it 3 has done well. We can do that one.
Okay. I mean, we're just like positioned over this cliff.
I know.
So,
So squeezer 3, yeah, squeezer 3 misfired in the basket once and it grabbed a sample on the last dot. So
All right.
6 and 7 have also both consistently fired.
well, 6 has the weird thing on it, so we're going to
Yeah.
ignore that one.
Yeah. Yeah, squeezer 3 seems to have been done. It seems like it will trigger. Huh? 3.
3. We're going for 3. Lucky number 3.
Was there a squeezer last night?
Actually, I don't have a fist in this one. course, you might not be able to fish that one out, but try. Yeah, just watching the shoulder.
Did you see the sponge pumping?
I did, yes.
It was pretty cool.
Alright, this is gonna bless the squeezer.
Oh,
We can put it on your chair.
that would be lovely.
I think she should wear it.
We don't need to
Nice.
force that. But either way, we thought a crown could be like uncomfortable for a long period of time. So
Sash is much more comfortable.
Nice. Well, thank
Happy
you.
birthday.
Oh, it's like it almost be like a hula skirt.
You can wear it like that.
We thought it was great in the PR footage, you know.
Yes. Thank you.
welcome.
And it's goldy.
It's like you have to put it on high temp or else it won't fry.
All right, here we go. We're going to try lucky number 3, squeezer. This one has behaved for us in the past, so we have our fingers crossed.
Luke might be right with the test of spongy food.
We're just like dangling over the edge of a cliff here. These pilots are amazing and have us, you can see those 2 arms out, maybe for the basket. Hopefully you have that view. I'm not sure which view you have. Just kind of holding us on here while they
Trying to get rid of those?
do these amazing feats with the manipulator.
Still trying to get rid of
Trying to put it into that,
or whatever.
that little holster.
I'll get rid of them.
It's like those like pop rocks.
Looks great. Wow.
Yes. I don't tougher than that. You want to estimate my eyes? Make sure to pop
And
Do
then,
not do that.
Yes.
after we successfully get this sample, let the pop in this one.
Papaniskin and then do a Solaris.
Yeah. And then
All right.
I think last Kate would want the tissue.
Oh, man, this
A little
The
bit
tissue.
bigger.
Yep. Okay.
I feel like I can breathe in all the balls around the ship.
Yeah.
Back as much as you can. There you go. Just need that. There you go. You might need to just a little bit. There you go.
This is a much better place for
There's an ID online
keep it
that
level.
looks, yeah, this could be another system sponge, you know.
There you go, you got it.
I agree.
All right, here we go.
Yeah, so the opening of the sponge is kind of this way.
Don't go any more tighter than
Well, that was the
I think it is, yeah.
That was
Yeah,
freeze,
no.
okay.
No, when you unfreeze it, it's gonna loosen. So just watch yourself.
That looks great.
Yep.
So a little cock just like that.
Straight on.
Sneak the tube in.
Use your sit camera to see if you're over it.
So again, we're trying to use this squeezer to get water that's coming out or being pumped out actually by the sponge into the surrounding waters and changing the biogeochemistry. So these big barrel sponges are kind of really important. Come on, squeezer.
Breathe, not fire.
Oh,
Okay,
easy, easy, don't drop it.
That was
No,
Okay,
that one's
sorry.
not working.
Breathe, not fire.
Okay, put that one away.
Does it come? Does it
You want to try one more? All right,
Please.
we're gonna try another one.
come with squeezers too?
Yeah, apparently the guy that works, who owns them or whatever works with them, he said he usually gets on average four out of the 6 to fire.
Oh, you didn't come up with that?
I did not. No, they did not. They are cool. I know they're designed by Bari. I've seen some of the drawings.
Do you think we should try the angled one, Scott? Because they could grip it more in the middle. Do you think the location of the squeeze could help? Or?
I'm actually worried about tipping these on their side.
Oh, just location, yeah.
Dropping them 400 meters down.
Fair. Yeah,
And then we have to spend an hour going to look
Yep,
for it.
yep. 100%.
So,
Yeah. I love it.
Great, we can stop anywhere in this area here.
Have to get on next one, we'll get it.
Yeah, you can stop on the other side already.
Still being carried. Yeah, still being here. Would you like us to get to the website again, or we'll just ignore the website for now?
Yeah, you want to wait for it to stay here, please.
Staying here, please.
Need to get a little video clip of that sponge pumping. And
Did we-
you want it right now?
did we hit the video
Do you
highlight
have a
button?
video clip of it?
I can, let me see if I can, how do we do this?
Or would
Right, I
just
can
be
run
cool
into
to,
the next
like,
room.
I did it for you. It would be cool to share that. That would be a Twitter.
Oh, that's a great tweet, yeah.
Mm-hmm.
What's that?
All my followers.
Yes.
Yeah, just trying to roll it up. Yeah. There you go.
Are you passing me notes? Is this like check yes or no?
You might need to,
Do you
yeah.
want to go steady?
Yeah,
You guys are so weird.
you might have to bring it out, like push it against the other one. That's how we turn.
Fly out on the 7th, right? Yes. Who are those you guys?
She is in the live stream.
Just about lining up.
Does she think we're leaving on the 6th?
Well, she emailed all of us
Drag
before
her
we
with you.
tried to confirm, which they really it was
No,
3 days
right
ago.
there.
Yeah.
Just come down a little bit. Come down. To your left, just outboard. There you go. Right there.
Nice job.
It'll be alright. You just need to wiggle it.
Yeah.
There you go.
I'm leaving
Lucky number 7? What's the one actually right next to 3?
I don't know what the number is.
Oh, I see
don't
4,
really see
7.
it, but
Oh, that's probably East District.
yeah, you'll have to use the other camera to grab that
I'm trying to read the.
the number. Oh, I didn't see the writing on it. Oh, can you see it down there? I can't really see it.
No, I can tell that there was something written on it.
Oh, okay.
But I can't tell you what number it is. Number 7.
Is that, you want number 7?
7 and 5 have been successful for us in the past.
You're grabbing 7.
So either of the 2 on the left.
All right, we're going to give this a third try. And we're feeling lucky. This is going to work. 3. But then again, it was nighttime. And sponges are known to only pump at certain times of the day, primarily during the daytime. So it could have just been that it was nighttime when we did those other tests. Right now it's 3 o'clock local time. So maybe it was feeding time for the sponge. It'd be really great to get this sample so we could see how it's changing the water. So we're gonna try again. Hopefully the squeezer will behave this time. Send good vibes our way. All right, so it's holstered now, and now the manipulator can grab up on the top where the trigger is. The trick is if you squeeze too hard, it'll trigger early. So it's not an easy process. All right, everybody, send good vibes.
Sir Thomas John.
This is, here we go again. We're gonna move it over to the sponge. Right in that osculum. And try to get the bottom of the trigger right inside there so we can get the water that's being pumped out. Now if we give a squeeze, come on.
Yes.
Yes!
Well done. Lucky number 7.
Woo!
Lucky number 7.
Yay! Thank you guys.
What kind of depth would you say that was? 75%?
What's that?
The plunger depth?
Oh, full.
All right, squeezer number
Did that
7.
C-log. We want
Phew.
now when they fire, they either do or don't.
it on the sheet because there's no squeezer 7?
Hold on 1 second.
There is a squeezer button.
Hold on. I have it in C-Log. Okay, so you're looking at the basket map?
Oh, the basket map.
Just cross 2 off and put 7 maybe?
Oh boy,
Okay.
here we go.
That's fine. I'll tell them what I got.
Yeah.
So we tried number 2 and number 3. Didn't work.
I'm trying to get back in there.
Squeezer number 7 worked. So we collected that sample at 3:25 p.m. local time. And that's 1925 UTC.
Are we gonna get a sample of the sponge as well?
Yep, we'll do a Solaris and then collect a tissue sample. Uh-oh, do we lose our footing?
Yeah, we did.
Uh-oh.
It's all right. Hold on, just settle now.
Oh, that was the best one.
I think so. Okay, do what you need to do. Not too close now.
That was a nice plunger pull too.
It went nice and slow. Yep. Yeah.
This time I'm gonna go for
Okay, just remember when you do, you want to come up inside the porch, right?
Yeah, but I think that.
So hopefully you all saw that
Get it up.
squeezer, the plunger pulled up nice and slow. as we mentioned, the piston there is really tight to make sure that that happens, because if it pulls really fast, then it can entrain a bunch of background seawater. So we really want to have a slow release so that you're mostly getting just the water that's inside that sponge as it's pumping. And that one looked like it worked perfectly. Yes.
I'm gonna start to measure the background. I'm just noticing that the dye is going. I can't tell how far. Probably fine with the Solaris, right?
We could wait a few minutes and see maybe if we can push it in a little bit more, or move the wand farther away from that green stuff.
I mean, your microbiome
Then I'll wait.
will be as it is, right?
Yeah, no, it's fine. But I guess I'm just being curious to know now if this works. I also was thinking if we want to see the background water versus the
If
squid.
you need to
That's the
avoid,
idea,
just twist the
yeah,
rest.
right.
Yeah, roll it up. Watch the camera. Watch the camera. See where you're at on the science camera now. Yeah. You know, close your arm. I'll try it. Get back a little bit more. Okay. Now I got you some more room. Thank you.
Oh, so the sweet determines the water resistance strategy.
It was basically almost exactly the size of the squeezer. So I just estimated that. With the lasers, yeah. We can have them do the laser. So we're gonna do a
Might try
Solaris
rolling it up a little bit more. Should straighten that out. Maybe keep going.
And then in case you
Should have more control too. There you go. Now you can, yep. There you go. Just bring it in. Yep. Might have to come past it a little bit, shoot down. Scan. There you go.
Yes, so I was doing it. It's tough to know.
Yeah, and then it will, like, the CC will, feels that way.
Oh, okay. You might have to get it where you want it. You hold it tight. Then let your loose. Let some loose.
Do you wanna take your sash, your birthday sash?
Can't have my birthday sash.
I said, do you wanna take it?
Almost there.
Ooh, it's the hot seat.
You can see in the sit too where you hit
Really
the
is.
other one. So if you come down here,
Have the squeezes been behaving better in general?
Generally, yeah.
Okay,
come
awesome.
down.
Yeah.
That's a good.
Even if you're not wanting to,
Generally,
just keep going.
Except that we just had one for three.
You can push it against the other one straight.
But
You're
previously,
in? Okay, perfect.
Not many of them
Get it?
are working.
Nice and
So,
Three have been working really well so far,
okay.
but it didn't this time. They didn't
Alright,
get
fire,
fire
try
on
and
the
rechange this
last.
hose.
So I don't think they did service or anything.
Yeah, we can try, cause I've already retracted as much as I can retract.
Yeah, stuff.
I mean, I can even try and look
We
at the
can
back.
talk, is our plans to figure out something?
No, I can't, because I don't have it worked out.
Connie and Heber.
I saw you thinking.
We're at 400,
Still
it came up.
so.
I
Just
wasn't
trying to
here.
recover you.
Maybe it just popped
We
out.
came up, this is where we started seeing
Maybe it just
things.
bent over
Do
for
you know
some
how
reason.
long the dye was gonna go for? Oh, we just went in to get it right.
Yeah.
It was, but it was squeezers that I don't know the numbers.
How many squeezers would y'all have to try? Was the first one you did work?
They tried, oh, I tried one, two, the third one we had to do. Guess the third was a little less. It was scooped over.
Let's try to do it, I'll try to keep it the other direction.
Okay.
From this side.
Alright.
I gotta get liquid.
Might have to start.
Just time out. Bowling or what?
It's like a shoelace.
Shoelace now?
So that'd be the idea.
It's came alright.
Yeah, that
Yep.
direction, I think it's like better than.
Yeah, that shut it off.
Have you
part of it too, though, is it's probably gonna look like it didn't take at first, because that guy's so close.
Right.
There's still so much in that little.
In the two, yeah, exactly. All right.
Alright, what's next?
Next will be.
Sorry.
Solaris.
Yeah, but I'm just wondering, is there any way we can put the dye somewhere else there?
We just stashed it, so it shut off now.
Is it just me or is there something?
I think
It's
you'll
wafting
We'll
away.
wait for, you can wait for that waft to end.
Yeah.
It's also going that way.
5.7.
Just wanna-
On Seabolt. All the way to sea, so.
We will be able to tell pretty soon. It looks like, it's gonna be hard to tell, because a couple inches is gonna do that for, so.
This display.
I think that works.
I think when it goes over the end of the TA, though, it has a chance to make a little bit. But if you bring it straight back, it'll zip that. It's more like.
Super scientific.
Measurement on that. Hope I do. That can be too close, I think, get you a measurement.
If it's
For the background?
Yes, or
No, for when we actually do that.
You think you're too-
I don't
you-
have any more stretch on that. For like this.
You're talking about the manip?
Yeah. You should be able to get that right. On the side. Yeah.
Oh, we lost our science cam here, didn't we?
Oh, why did we switch back to one?
Hmm?
Yeah, put that back on.
Aha. So you think you're too close?
I can probably get it across the
should be
front
able-
and come back to the left a
Should be
little
able
bit.
to go back this way, then up like this.
Yeah.
Okay.
Or you'll want to sample it inside? or-
Yeah.
Yeah.
Oh, yeah. We'll give it a shot.
Yeah, I think
Okay.
it might, I think it might actually be all right. If you're able to just elbow.
You ready?
Yeah, I'm gonna start
Elbow over and in.
Okay.
I thought it might be the same.
So we're gonna start measuring background now.
Okay.
What do you, I can go.
The problem with these walls is that if you get actually, it's really easy to get too close.
Yeah.
And so I think if we put it between the bumpers like normal, it almost, it's almost worse.
The SH number for
Yeah,
this
so just like
one
you
is
come
the
up
one
with it, you adjust it.
675.
They try having this right.
Really
elbow up
steep
and
cut.
see how close.
Mhm.
Yeah, they're doing the background now.
Okay. So,
That camera.
Are we
shows-
just supported by one?
Yeah.
Yep.
And the thrusters are on, so he's
Oh,
holding himself
heating
down.
up.
Yeah. Well, they're holding us down. Right.
What's the name of those balance pieces?
Rock'em and sock'em.
Hmm?
Rock'em and sock'em.
Really?
Yeah.
Rock 'em, sock 'em. Okay.
Rock'em isn't on the right for on?
No.
Right. I don't think we've actually officially designated which one is wrong.
Oh, okay.
But are they just called like rails in your body?
Rails start on rail.
Yeah.
Oh, you get a hang.
The S is for starboard, so.
What? Thanks, I need that now. Comforting.
Rock'em is and sock'em is.
All right. We're collecting data right now and getting a background measurement, so we'll be. So we've done a lot of servicing on Solaris over the night. By we, I mean William Pardis, the lead electrical engineer here on the Solaris team.
Oh, you usually run it.
Jack of all trades.
It's a long
It generally is like when it's stabilizing like this, it seems to be
Yeah, I feel like if we're not producing ROS, it would be interesting to do another quick dye to see if it's still pumping or not.
Just use a stop.
Oh, just to see if like the ROS, like if it's
All
not
right.
producing ROS.
Go ahead and
if
let's
it's pumping.
position the wand. Just kind of down the throat
Because it could have stopped.
as best you can without ramming
Like
the.
4 foot tweezers over that.
Outside, so you just be wary of it.
I mean, not a lot, but like.
What's that stage number again? 186. Sam, what's that stage number again? 186.
675.
Yeah.
All right. Sophia?
Yeah, you're looking good, man. You can see your shadow back there, so.
Give me
The smaller.
We're at a depth of about 99.5 meters, right around 100 meters here. Just over the break of the La Paguera reef here on the southwestern side of Puerto Rico. Great. Shelley Partis says hi, William. I believe that is Mom. William is currently asleep. Well earned sleep, rest.
Could we do a measurement on the outside and then on the inside?
Oh, okay, sorry. Yeah, we're gonna do a measurement just to the outside of it first. Yeah, right there looks good.
Alright.
Hold that. I'm just gonna get a momentarily tight zoom on the where the tip is.
Scott, can you take a picture of that?
Sure, you wanna get a screen grab there?
Frame grab.
Got it. Frame grab.
Look, you have 2C logs open for looking and taking pictures and doing stuff.
Solaris is
There's
just a
evidence.
start.
Oh,
That little algae around the rim, it looks like there might be some photosynthetic symbionts.
Yeah, for sure.
Super cool. Although sponges are usually heterotrophs, there's evidence that sometimes their symbionts provide them energy
Yeah,
as
a
well.
little bit of
Not
coral
just
there,
from
too.
filtering.
Oh, down here? Yeah.
if possible, how would you distinguish them from just eating algae growing on?
That's a good question.
Well, I guess that would still make them heterotrophic, but it puts them closer to being photosynthetic. You're acquiring energy from photosynthesis, not just feeding them from water.
It's also different to have like symbionts versus just epibionts, right?
Right,
So
that's true.
these guys might just be growing on top of them because they get a little light.
Or they like the nitrogen. Not seeing much of a difference.
All right, you ready to go inside?
Drew on the sponge.
Okay, we're ready to position the wand inside now.
Take the algae only grew on the sponge. Not around. This one that's significant.
Yeah, this is a fascinating sponge. Earlier, if you didn't, for the viewers online, if you weren't here about a half an hour ago, you could see we deployed the fluorescein dye and got very clear visual confirmation of this sponge pumping. It's pretty neat. If you want to, you can rewind some of the video and see that. So we've used the squeezers to collect the fluid sample now, and now we're taking a look at superoxide production by the sponge and or everything that's living inside the sponge.
Awesome.
Just touching it down here.
It's easier to choose from here.
Uh-huh.
It's about as close as I'm probably gonna
You
get.
might be able to just pitch, oh, you're already pitched all the way, so you might have to elbow a little bit.
Yeah, but if I elbow and then I get flatter on.
Watch the tip there.
Oh, sorry. There you go.
We're gonna try to get the tip position a little better.
Oh.
And you know,
I
you come
think
to
it's
the-
There you go.
that's
Weird.
right.
It's just a bump from this side.
Okay.
It's about perfect when it lags in the back. So I don't think that's.
No, no, or-
But it's interesting that I mean, you see that consistent.
or show, let me see.
You don't think you think
Back here. Yeah, that's what you're worried about. Yeah.
That's.
superoxide?
Already in.
Yeah, okay.
Yeah. Doesn't look like much of a signal.
You got no more elbow up.
I can, yeah, I can have a, but it's gonna take me further away.
Right. Okay. Now, try to, tell them. To the right. See if it'll go way up in there.
Alright, should we call it? Oh, are we hitting the
Yeah.
tip?
This is also kind of.
Scott?
Yeah.
Can we move on?
You wanna move on?
Yep. Yeah.
Alright.
Can we squirt the dye to see if it's still pumping, so we can see if ROS is
Oh, wait.
related to pumping?
No, it's again, it's just
Bump?
a bump, I think.
Okay.
Yeah, I'd rather kind of move on. Yeah.
Okay.
Got a birthday mission.
Can we? Okay. Do you want to sample some tissue?
Yep, sample tissue.
You firing this again?
Yeah, do we have time to do a little dye squirt, or?
Three, have some shoulder
It's
down.
unlikely
Let's
they would
do that.
Stop, it was unlikely they would stop.
Yeah.
They don't have like a nervous system. So if you pump it, if you bump it on one side, it's unlikely to have some kind of systemic reaction. Stop.
From what I
What's the
read,
where did the
it
bubbles come from?
can.
That I don't know.
Yeah, there are air bubbles in that.
Yeah.
They shouldn't be making it out. I probably could have speak to those.
They might actually. I see the sample pump is working. Oh.
Easy, what was that?
That was.
Okay.
All right. So
43 of that.
just in the interest of time, I think it's good if we
Okay,
grab the
you
Niskin,
guys the boss.
grab the Niskin, and then grab your tissue sample.
Something weird going on there,
It
Lena?
stopped. Do nothing.
I just stopped the pumps. Oh, okay.
Alright.
You feel good about that?
Yeah, you're gonna get a Niskin, yeah?
Yep.
Okay. Which
Which
one?
1, 2, 3, or 4. Might as well just stick with number
Dealer's
1.
choice, number one.
Dealer's choice. Good. 1 through 4.
This camera is.
Okay, did we stop on one kind of
No.
issue?
That's
Not
fine.
one sort of dial.
What?
It stopped on what kind of issue?
Well, it can switch inside the tissue to antioxidant. And so it would be important to know what
No, I
the microbial activity is based on the toxic part. But also,
You're
it's
gonna
gonna be
also-
many hours until
Yeah, I
we
was just
get
gonna
the
say, you're
tissue,
gonna
so
crush
the
the thing and then-
metatranscriptome
Yeah.
probably won't tell us that much information. And we do have the background and
Seems okay.
who's involved with that, which can tell us.
One,
If
yeah.
we see a difference, then we know who's coming.
You here, you gotta be on set.
We live in a half an hour.
Oh, yeah.
It's not perfect, but we just-
Oh, you know, I know what, 4 hours, ripped it, and do preserves and then.
But what else we can do is use it. I feel like things are so restricted.
Which one are you going for?
Four was an option.
Yeah, go for 4. That's fine.
I mangle the cool as much as we mangle sponges.
If we can do like a pie slice. of
Did you
this-
bring your pie slicer?
No, but they did that before with the,
Yeah, you kind of.
with the
Oh,
Exoduda.
what Niskin are you about to play with?
4.
Okay, cool, thank you, sorry.
Looked like 5%.
Scott, can you write that down, please?
It's written- Yep, got it.
Okay.
Triggered.
Alright, just freeze it or hold it. See.
Which one is it? The one we can't see.
It is the one we can see.
Oh, I think you see it, right?
No, that wasn't that. No.
Still texting Courtney.
But
Oh, that's bigger than
flag
you.
went up, no.
Flag went up.
What are your sample IDs for your wristbands
This will be FKT 23.
I don't know how that is, but is there a way to do the lasers to
That's
see the size of the sponge?
Yes.
Dash. Yep, that's it.
We're gonna take a sample from this one, it's an area. Situation where you'll be able to grab it.
Alright, so now tissue sampling.
Yep.
It's gonna go in a squeezer. I mean a quiver.
Yep.
So you probably want to open a quiver. Okay.
Yeah. It's on the left, and you're gonna like switch it forward.
I think nothing's gone in a quiver yet.
So if you want me to.
Yeah, on the,
Yeah.
on the porch out and all the fireflies go open.
Annoying ones first this time.
You want me to run that? So I gotta watch where you have the port arm. It's that with the porch.
21st.
So.
Yeah, we need to hear that first.
Maybe go back.
Haven't heard any news about my animals.
Yeah, whenever is best for you guys to do the size.
Yep.
Snaz, yeah. Until we're clear.
Yeah.
Are you?
I don't know what's over there.
Jinzy? Is Jinzy?
No, it's Sophia. I used to watch the
Oh.
Myself.
Yeah. Should be clear there. Okay. Just when you get this one unfroze, just lift it up so I can bring the porch out. I just want to watch the lawn tip while we bring the porch out. But it looks like we're clear. Alright, ready?
Yeah, but I think it's
Now
just good.
porch.
I feel like it's good practice, bouncing it off.
There just might be something cooking in the water here pretty soon. We've got a sponge sample.
Which one are you going for?
We'll go for number 6.
Maybe we can
Back
see
one.
the sample from
All
there.
one.
I think it's number 6. Alright, camera's still in order.
And then we'll put these in the bio box so that it's.
Oh, yeah.
We're afraid of losing a scout.
Oh, okay.
For 6, I'll find a swap. I think this button is pretty long.
Sam, are you logging this in? Alright.
Yeah, we can just come over in between the little square there. You don't want to get hooked in the handle though. I had trouble with it yesterday. when I was looking at it. Yeah. That's okay the way it is like that. Exactly. Very nice.
Oh, so with fresh water, yeah.
I'm gonna go ahead and set the board back in. This way you got plenty of reach without having to worry about that bio box too much.
Yeah.
Weird.
Alright, and then I gotta try and get the lasers on it.
Yeah, let's get the size.
Right.
Can we?
Yeah, maybe
Nicole, can we
screen grab the laser size?
Yes.
Kinda hard to see.
There you go. Yeah. So
Yeah.
can I zoom?
The hot water on the shit.
Oh, that's perfect.
Oh,
There you
I
go.
called it
You got
10.
it?
And turn it
Called
on
it
hot.
10 centimeters.
You were spot
Pretty
on.
good.
So that's 10 centimeters between the
Lasers.
lasers?
Yep. Huh?
Bigger than I thought it was.
Yeah. Well, with that squeezer,
Yeah.
you know how big the squeezer are. So it's like, yeah, it's like about that big.
Awesome, thank you.
What are we doing to this guy?
So if you can take a chunk of tissue, I don't know if you can take a pie.
Yeah, just 2 cuts, like a wedge at the
Like
top.
a piece of pie, yeah.
Came in at this angle, and then first the jaws came in at this angle, and then I grabbed the piece in the middle and pulled it.
So just literally just from the top 2 cuts and then pull it out.
Yeah, whatever you think is the easiest way of doing it. We don't have enough space
Remember the
now.
size of the piece that you take this.
I don't know which one of those.
Otherwise, you have to use a piece of the file box to get it.
A coral quiver sized
The
piece.
coral folks won't share the bio box with the sponge.
No.
We'd have to
They could
chop
get
it.
confused between which one's which.
They're not colored. One has a little black inside, and it has like a little like black strip on it that doesn't do anything, but like when you twist it, like
The first and second cut's gonna be a problem.
it gets
You might have
hotter
to,
and older. And then I
you know, smart first cut, yeah, you might,
It's a pretty thick wall.
yeah, it's gonna be interesting
And I
too.
thought maybe the middle thing.
Yeah.
No, it's a little like.
Watch that camera.
I don't know. It did not come in the middle.
But I knew I fixed that.
Down, pull it down.
Yes, I mean, the first cut is close to where it's about to be okay. The second cut.
Yeah. I get bored.
Just don't.
I got my crochet or something.
And don't forget, you can use your keyboard camera. Yeah, you look like you lined up pretty good there. Right.
Does your shoulder kind of?
Remember, you don't need a huge chunk.
Yours doesn't?
Well, just like having my brown and blue, I think might be the same, but just.
My daughter's in number 38 on the right.
And you can let all that clear.
At least I was thinking I'd try, because I thought that would be what it was.
And once you can see good,
Just eating
just
these
cut
with
it.
Text muter,
That's a smoother?
apparently. Yeah. There's a lot of different growth forms of it.
Really?
Nina broke the chair.
No, honestly, this is how it came when Eric left it.
Oh, you're blaming it on Eric.
Well, I mean, look at this. He was
Yep.
Nina.
Oh, I gotta get a picture of this.
So you don't see it.
Or is it just one?
There you go.
Awesome.
That's all right.
That's okay.
Okay, I was like, they're saying that they have two, and I'm like, I don't think the other side moves.
It's just red and blue.
Don't come in
That
anymore
makes
with
sense.
the line. You see how close you are to the camera? Hit yourself.
And have you noticed that the sink gets like
I'll bring the sports back
really
up.
odd?
Way across the end. So sure you're leaning through that way and you're gonna send it back. Stable that? So now we're forward.
All right.
Fabulous, guys.
So we think this was a Zestaspongia muta, the giant barrel sponge. These guys can get much bigger. We are leaning on some of our shoreside expertise.
He's got him set up there. Got you there, Alex? Switch out.
Have they done any squeezers on the other lots?
The only thing.
Oh, sorry.
Squeezers, the other lots?
I don't think I ever did on.
See, that must be why they find so many corals and can go so fast. Yeah,
So they
we're not
give
just all
us,
these
give
species.
us fake board numbers.
Yeah, yeah. I swear, Santiago tries to list back there and tells gives us the back part.
It's like making fake treasure maps. Treasure maps out of fake locations.
Oh, you guys haven't found any coral in there?
It's not Santiago's hiding it.
Hiding the coral in.
Just a bunch of gold corals.
Thanks again, you're here.
The secret of how you stop.
I've had people send me this video.
I thought that was your, I think that was like an earring or something at first. You were taking one up.
Years ago, yes.
Pretty big stud.
How exciting!
All right.
Alright, let's find some coral.
All right.
Back to Coral Quest.
Soup to nuts. That was an hour.
Back to birthday mission.
Yep.
So we got pumping, squeezers, Solarising, Niskin, and tissue samples. All done in an hour.
Several people send it
Pretty
to
good?
me. Nicole.
Yes. That's the best version of that name I've seen so far.
So I think now after this we're gonna continue up slope.
Okay. Directly up or?
Fantastic.
What do
Thank
you
you
want?
guys. Yeah, back
Okay.
to you.
I'm gonna give it back to the birthday girl here.
I was thinking Colleen for the sake of science.
Happy birthday, Colleen.
Wait, why is that?
Get a photo of the pumping at an angle like this would be epic. We don't even need to sample a sponge. We just weight it with a nice angle.
Right now?
No, not right now.
No.
Eventually. Because the angle we had, it was very vertical, but we could get like an epic science communication shot.
That would, yeah, require the right angle.
It would require the right angle. I'm just saying.
They
if you
have
see
a down,
it,
there will be a down looking angle in the data after this guy's over.
Oh, well, I was thinking something like this, where it's like,
Down looking angle of what?
oh, an epic pumping photo of another sponge.
I mean, this is pretty good.
Alright.
It's pretty good. It's really good. It's pretty awesome.
Yeah, pretty easy to see.
Are we going up or?
Yes, let's keep on going up.
Sebastian was here.
Great job. Thank you guys.
Get like a knife and a card into the sponge like Sebastian.
All right.
There's some barracuda hanging out. Tether.
You see a barracuda?
The tether cam, upward looking cam.
Oh, yeah, yeah. Above us?
Yeah.
Behind us.
Oh, that's cool.
Above and behind.
Walks in on the action.
You guys got
Come down and say hi.
St. Croix. What?
You been in Windsor Bay?
I have not been in Wintershire Bay.
Oh, Frank. Frank is a 6-foot barracuda that hangs out there and he'll follow you.
Sweetheart, total sweetheart.
Total sweetheart, sure. He hits me every time. He'll just be like cruising around, and, you know, doing the refloating. And then the one moment you'll go like this.
Open up the
There's Frank.
ice. Where's Frank? Where's ice?
Greater Windsor Bay and St. John.
Oh, it's alright.
But that's, they've all named him Frank.
Did you
He
Max?
hangs out there. Max saw him, yes, I introduced for Frank. And then maybe Frank's neighbor in a different bay.
Oh man, the Florida Keys, there's this nice diving spot on Blue Key where this huge grouper, it's like 6 or 8 feet long. Is there? It's like this big around. And it just hangs around the reef, just like keeps distance from you. She's always there. Here. No.
What's up?
I was just looking at how deep we are. 93 meters. Wonder if that red is if we're starting to see Siderastria.
Let's look at it.
Yeah, could we just take a little peek really quick? Because I know that it can be a little deeper.
Metabolisms and corals. Different metabolisms.
There's a fish over on the left there.
Are the metabolic rates just gonna be different for species? Probably. Yeah.
Like based on eating
You
strategy.
have so much different biomass.
You can take the
I guess
camera
it's
if you like.
better at the biomass.
Romans.
Oh, is that a little puffer fish?
Oh, wow, look at it, we're getting the fish. Where did
Oh, up on the left corner there.
Yeah.
Is that a squirrel fish?
Wow.
What is that?
Are there different colored squirrel fish?
Oh, I don't know.
And take a closer
Oh,
look at-
I think is that a sponge? It might be a sponge, actually.
Like one.
Maybe zoom a little bit.
You
Oh,
wanna-
do you want me to do it?
I don't know, you wanna look at it?
Wrong way.
Does that sponges hang out? One of the ones with zoanthus on it?
Which one?
Upper left, where the scroll first was.
Yeah.
Oh, yeah, that looks like it. Yeah. I was trying to figure out if this orange, I think it's probably a sponge, huh?
Yeah, it looks like it.
I was wondering when we might start seeing.
Oh, I still got you.
Yeah, that's why it scares me every time.
Is that a little Halimeda there?
This here, it's just, it's got two Oscar.
Which one are you looking at?
I'm looking at something that looks like a very light city with probably just that little guy. What is he? I think there's another one.
There's a little fish.
Oh yeah, it's the sponge with zoanthus.
Yeah. Look at that.
Yeah.
This is pretty cool.
What?
Yeah.
Wrong way.
Oh, wait, there's some
Oh
weird,
yeah, he's just a
there's a weird
fancy
brown thing that
fish.
looks like it's got lower the camera a little bit, almost looks like a coral.
Almost look like a coral.
Yeah, oh, it's just a potato sponge or something. You see that potato?
Oh.
Oh, on the bottom here? Like down here?
Potato
It just
sponge.
has like little white dots, but it's probably just a sponge.
Yeah. Yeah, more sponges down here.
Frank's pretty harmless.
All right, well, I think we can keep going. I think we might start seeing some stony corals here.
Do some night snorkeling.
I can bring you into the yeah, keep it. Just hold it, maybe.
Look, watching. So is this site like it's got like a peak at like 40 meters? Or how shallow does it get?
So, well, it goes right into a shallow reef, but right at the reef crest, you know.
Oh, here, it
Look at there.
flattens out quite a bit, coming up here over this little ridge.
Yeah.
Of course, we've seen just sponges with no watch.
There'll be corals. They're coming.
That's good. That's good. Bring the vessel towards me.
We're about 84 meters now. That's 40. Hold on a second.
Was from where we were.
Yeah, so from where he is now,
Yeah.
I need to bring him about here.
Got it. So not the waypoint current.
No, where he physically is, yeah. I see. He's 155 meters from me. I'm trying to get him a little closer so I can move forward.
Got it. Just 20.
Yeah. All of a sudden, Bridge ROV, we're going to leave about 20 meters to the north. Thank you.
So we should start seeing Madrasis at about 80 meters. Also Agaricea. We should see.
Kind of stuck for a minute, so. Yeah, a little bit of something. Just don't go too far with the tail, but then I'll go a bit down. 30 meters long.
We don't know. We're at 85 meters.
is that a coral right there in front of us? See that sheen on it? When we look down at this brown puppy thing? There's
The
a sheen
brown.
On that. You see that sheen on it?
Oh, yeah.
That looks suspicious in a good way.
That looks like something I've got.
Oh, I thought like green one.
I think that's gotta be a coral. But it's clear to him. Are we passing over it?
No, I'm just kidding. I'm looking up right now, so.
There might be another one over here. Something bigger.
That's correct here.
Can't
Is
tell.
that right?
It's all pretty big right here in front of us.
Yeah, true.
Oh, I see what you're saying. That one's got, looks like it might have a sheen too.
Yeah.
I'm gonna go look my.
Oh, that looks.
I'm gonna go to the same one.
Oh, okay.
Just real quick.
I can't do it right here. Oh, it's got that crown edge. It's an iris.
Somebody might have.
Oh, that's an Agaricia. Yeah.
Nice. Okay, on right here.
No. Yeah, go ahead. There you go.
Why does it look so wet?
It's like platy because we're so deep. So it'll get less platy as you move up.
Oh, my first platy coral.
75 meters.
75 meters, yeah.
Holly, can we do a Solaris measurement?
Of course.
Okay, I'm starting back.
No.
Oh, me.
Hold on. I have to get this off of us.
You okay?
Yeah, just that.
That rock.
That rock fell on us.
So we're at, let's see, 2009. Our first sighting of an Agaricia. It's a scleractinian coral. This is the one I talked about earlier that has a really large depth range, yeah, so it can be all the way up 40, 50 meters, down past 80. And it changes its morphology based on the light conditions. And there's a sponge with some zoanthids above it. So you can see those the ridges there.
It's probably fighting over there with that sponge with zoanthids.
Yeah, it might be.
Squirrelfish.
Oh, and a little squirrel fish right underneath, saying hello. Come back out, little fish.
676. 18676 is the SH number.
18667.
6. I think she's getting ready. Do you want to get ready for a background measurement? Oh. Oh, okay. So we can go ahead and start moving towards the coral.
Oh, this isn't this order. This is what's the name?
So we've never measured Agaricia before. We've done a lot of work with scleractinian corals in the past, shallow water corals, and I think I've mentioned before that they vary. Some species produce a lot of reactive oxygen, some don't. And the interesting thing is it turns out the kind of more resilient species seem to be able to produce a lot more, and they kind of constitutively express it, so all the time they're making it. So we'll see what we can find here with Agaricia. Looks like it might be fighting with that sponge there. Yep, I think we're ready to move in. We've got a good background signal here. I don't know if it might be easy to kind of get in a way, so there's like a little bit of a ridge there. And a great zoom there.
Can we just hold it for a while?
Yep.
Somewhere around there. That looks like a great position.
Yeah, so it has really small polyps. You can see them along the ridges. You want to try and move to a different position?
Sure.
Is this dog 5'10"?
Yeah, what are you thinking for the next one?
Yeah, maybe. I don't know. I was thinking maybe more along the line, the edge, where it's actively growing. Can maybe even come in somewhere around here. What's that? Getting hot? Okay. Do you want to?
If I come up and come back down, because turn my gains down quite a bit, but since I'm frozen right now, it's not really been a lot until I let go and come back down.
Okay.
It just take a second.
Okay.
But it's okay to pull that away just for safety.
Yep. Yep.
Can you bring that up just for a second? Safety, come off these things, down.
The RV's hot.
We've done a little bit of that, but it's probably, once we see a signal, go, okay, now, is that any of the lights? Like, do the lights, you have to leave it? Yeah, it's not.
So when we're in shallow water and it's warm, so the ROV gets hot. So every once in a while we're gonna have to jump off and hover a little bit till Sebastian cools down a little bit.
Typing down, using brilliant for us to see if that makes it easier for us. Alright, as far as the little thrust, I can do it through the ceiling. So I'm gonna show that needs to be ceiling.
What's the happy temperature?
48.
Oh.
They're nocturnal.
Like, the speed.
Is that up?
Yeah.
That's what it says. I mean, what way are they nocturnal? What are they doing?
I don't know, it says they have very fine tentacles, but they're seldom
I just
excited.
lost my jaws. You're good. Keep going.
Is 417 day or?
Are we moving that?
I tried it out.
Are we gonna measure?
Yeah, we can try to one.
Okay. Yes.
Amazing. I mean, it's good design. Hold on. What was that? How's that? I can go a little bit more than so. Alright. Actually, I think it might be a good idea because we are pretty stretched out. I don't want to shade that. Alright. Yeah, cool. You can take it just so we have it.
Okay, you want me to watch it?
That's all I can do for you.
Yeah, 524. That would be how it's done.
And that worked. Yeah, we dropped 5 degrees since I did that.
So we're just hovering, is that what we're doing, or?
No, when I sat down, gained were up. So I just shut the gains down really, really low. And sat back down just as lightly as I could, just so that I'm not pushing nearly as hard.
Okay.
Just sitting like this.
Yeah. So since it's low current, can you do that? Yeah.
Nicole.
Yes.
The Scleractinians we found on the beach. Rose coral. Rose coral?
Yeah, this is
You good?
Yeah.
All right. I think we're good with the Solaris.
Yeah, see you. Yep.
Sam, do you
Just
want a
noticed
piece
that.
of this?
Yeah, I think we should
Yep.
get a piece, yeah.
I wonder if there's a way we could it's going to be fragile, so maybe pull off a little wedge on the side there.
So it's coming out to the right there.
Yeah,
Just kinda-
maybe you
alright.
can kind of
So
snap it.
this is gonna be pretty delicate. Crafting piece of time, so the best way usually to do it is to come in, not really grab it, sort of like, just with the rip and not even blades. Yeah, just like kind of like if this is it, come in like this, and sort of just like, don't squeeze it, just go and then kind of hold it. He can help you out a lot more with that.
Ooh, thank you.
Happy birthday. Gummy
I got birthday gummy bears.
Whoa, Who wants a break?
Back off. Those are mine.
Oh my god.
Show you how to sample these guys. I don't think it's me you gotta worry about.
It's fine.
No, actually
We
Luke.
are hot.
Luke has a
and
really big,
he's hot. I'm sitting down just barely right now to drop the 64 to 58. So
Yeah, we're just gonna have to let it go off here.
yeah, we're real low on our. What do we need to push out, or you have to?
Where are we stowing it at?
My guess would be,
Okay, what do you think?
I don't know.
Where do y'all want to stow this at?
Whichever's easiest, if you
Depends
can
on
get
the
it
size.
over, yeah.
I don't know how it breaks off. Because you don't want a big piece, you want a small piece.
Well, probably will want a big piece, because everyone's gonna want a piece of this.
Right.
Yeah, so
what do we
price.
price?
I'm just gonna wanna get a piece of this.
Like, do you want to push out with rock and then we get it stowed, or do you want to leave it work?
If he can bring the porch out, just elbow up for now.
Yeah, let's see, also let's do the porch. Just get it out, put it out.
Yep. Elbow out and pitch in. Maybe do a little bit of rotate to kick it up a little bit higher. You know. It shouldn't.
I don't think so.
No, it has a bungee right there. Now that they got it all zip tied, it's much better.
That should be fine now, I think.
Is it too much fun? Too much fun? What?
Piece of push this off. But this is
Yeah.
all we've got here.
Handling it is unwise, causing a smarting sensation in the mouth.
Even it, file box one completely empty. Is your birthday box empty?
I think the birthday box is empty.
Alright, concurrent's not too bad, right?
No.
Sam, a bio box is fine, right?
Yeah.
Yep.
Alright,
Have y'all seen the polyps, go yet?
We didn't look too close at it.
Zoom in, Chris?
get you set up on the sit.
Y'all gonna watch this, it's been really cool. See this actually? These polyps?
No, where are they?
So you see that, like, it's just gonna do this crazy hyper color change. As soon as you go and you touch it, they're out right there.
Memorial.
Okay, well, you see the fuzziness?
Oh yeah.
Yeah,
Uh huh.
there it is. I'm gonna go for, I think this one
You
just
see the
already
little
broken.
dots in the rows?
Yeah.
Each dot is
In front of the side.
Well, you'll see it like, kind of does like this weird carpet thing.
I am touch just
I'm trying to move out slowly where I'm not messing with it. I'm just gonna come underneath it and pop it, and then pinch it.
Do you want me to come down just touch with the words to make me just for you?
No, I'm gonna put that.
And a little bit of it.
Look at the colors. See it change colors?
Oh yeah. Oh, very cool. Oh, is this gonna be hard?
On that piece, I gotta come to this one. You have to get it further back.
Move if I need to.
You can?
I mean, yeah.
No, I think I'll be alright. This is gonna be good, because I need to come up underneath it like this.
Alright. Pretty extended. We got some elbow.
This is all I think. About pushing you just a touch.
Fine.
These are gonna be hard to see.
Right.
They're not too bad.
Look how thin it is. The skeleton is so thin. Whew.
Wow.
Oh.
Nice grab.
Nice. Wow.
I'll go back and get that other big piece. We're going into the plow box?
Five bucks. Well, I don't think
Unless
anything
you
will
want
still
me,
fit.
unless you want me to go into quiver, I can set this down and put it in quivers. What do you want? Do you want to say plow box or set it with something else? It's up to you guys. Either is fine.
How hard is it gonna be to pick this back up when you set it down?
Not hard.
Okay, then I think it's good to put it in a quiver.
Follow me real quick. I'm gonna set it right here on this.
My trying to find some big bulky corals that we'll have to go on the bottom left.
What we got open?
I don't know.
Anybody know what we got open over here?
Everything except for Quiver 6. Everything's open except for 6. 6 has a sponge in it.
Do we work?
Those little rubber fingers really help on it.
Yeah, we used to use Tygon, like a hose, but because they're slippery, you would lose stuff. But because we 3D print these, it actually has like kind of like your fingerprint. You don't have to work as hard as what we used to.
Those red tubes are 3D printed?
Yeah.
Oh, wow.
Yeah, they're 3D printed out of a flexible material.
It's very, very cool. It's so thin. It's amazing you can be that delicate with it.
Wow. It's like a Dorito.
Tortilla chip.
Coral Ranch Dorito.
Yeah, tortilla,
Coral Ranch.
tortilla chip.
Agaricia chip.
An agaricia.
You mentioned Dorito dust made from these tortillas.
All right, one piece.
Jen, I think if this next piece stays clean like it is, we can use this one, Asher should last us after, Trace Meadows.
Yeah.
And the other one we can do for Jones.
Is that? Did you really?
So, looks like it'd be like, maybe Japanese, like, this big.
Oh, it's way bigger than that.
That's like camera out, a few centimeters, surface area.
I don't use like maybe a quarter of that.
So we're at 83 or 84 meters right here.
We don't want to keep it clean as possible.
Yeah, I know it's not for all. So I can put the particles in here.
It ran away.
I guess we'll save the skeletons after water sickness. And ribs and back. That's what they used to do. That one time, Andrew had a collaborator was telling us that every time there's our tank that bleeds and we kill all these save the skeletons. So they're like berries and sand. Yeah. It's just wrong. Just on the surface area.
How come it feels like the ship is like tilting? Is that just me?
How does the ship feel? What?
It doesn't feel like we're going.
Listening?
That could just be me. That could just.
They also might be doing something like this.
Oh, it looks like just hanging on just a little bit.
I don't know, speaking
A little bit more, I'll be over. You're over.
Look at that.
A little bit more, just a touch. There you go.
You know another little piece after this?
I think we're good. Is that good, you guys? Or would you like more?
It should be enough. I mean, if there's an easy piece again to grab, it would definitely know that.
That,
Might be
If
this
you
one.
can-
If I come in from this way, I might be able to pop that one.
or if you come from the back, you get a little bit of space, you can see the shadow there.
Back or back. Oh, coming from.
Let me see this one. From like this side, you can get underneath there, but I don't know how deep it is.
Angle that I can't get.
Yeah.
You might be able to. You need a clean edge or.
You can, what do you mean, like intact or like?
Yeah, or where I've already broke it.
It can be roughed up on the edge, like it can be snapped.
Yeah,
on the
that's
edge.
fine.
That right there, the crack piece.
Yeah.
The stringy bit.
I think I can do my nose on my
Yeah. I might be able to bring this in on Rockins. There you go. There you go. That's cracking this all this.
You think if just the rubber thread tip wrench that edge, it just snap it?
Could push it down.
You're just gonna get little pieces. I'm trying to fracture it down deeper.
Oh, I see.
I don't like to fracture by pushing it down. I just kind of like fill them up. This one seems like I should be able to get it.
Decent, the small piece fell off.
Yeah. I pick up all these little small pieces, that's for sure.
Yeah, that I think that should be fine, right?
Yeah.
Yeah. It's a little like Scotty.
A piece of Steve. When you going from? That one right there?
Wow. Broken chips. Really game changer. Broken what's better to me? There are ROTs that just have hard metal fingers. Just like it's so hard to hold it.
Anything else up there?
There was a little piece, but I don't know if it was
Needed something to struggle there to it.
like just two small pieces.
You just get that larger, cleaner one.
This one right here?
Should be, yeah, that should be fine. Get that one too.
I can't really see it opposite, but.
Need your water.
It's coffee still.
Oh.
Big grab.
All right. How's our temperature look?
It's been dropping. We're at 54.8.
Great.
It's supposed to 65.
That's good.
Wonderful. Thank you.
Thanks.
Changing that.
If you find some of the smaller ones that like are more like bowl-like, those are a
Mhm.
little bit easier to get, because you can get our jaws underneath them and just.
Okay.
and then
From your hero.
Okay.
You guys do a lot of dives here, like the shallow.
We did a lot in the Coral Sea Great Barrier Reef area.
Lots of little sharks over there.
Oh, did you say you saw another one to the left? Earlier?
I don't remember. Did I say that?
I think so.
You can look.
I don't think I don't see it here. We're probably at 5.
That's okay. We can keep looking.
Alright.
Well, you, up to you. You get it all? I'll say for a while.
I want to move that up the way a little bit.
What's that?
Move the chip up a little bit. Just move it.
Good.
Tip.
Come on.
Appreciate you. Alright.
Can you bring rock and ice?
Yeah. Oh, it sucks.
We noticed this coral because the lights gave it that little sheen in the
Mhm.
tentacles.
You can,
We noticed it because you said like a little sheen.
Oh,
on
yeah,
the
yeah,
brown. The sponges don't have that.
Oh, you look more of that?
Yeah, so we have the, I think the lights having a little.
I think in the
brighter
chat,
focus.
there's some people talking about there was something black.
it did.
If I know what you're talking about, I think it was just below what the Agaricea that we were sampling, and I believe that was a sponge, kind of a circular black with some spikes on the top.
Bigger one maybe?
Yeah, that looks like something, maybe.
Oh, it looks like another Agaricea there, right?
Yeah,
Wanna
maybe
go
different
do it?
kind. It's kind of going elevated. That's what's doing that.
Cross them up. Come off to the floats now?
What's that? Oh, you come off the foot.
Yeah, just, we'll put you back in on it.
You want to go under this one?
What do you think? Oh, what is that over there? Is that a sponge over there? Yeah, looks like a sponge.
You wanna have a look at this one, or?
Sure. Sure. Do you want to look at it, Sam? Or do you want to keep going?
You want to measure it?
Let's keep going.
Alright.
We could always come back.
Yeah. I think we're going to find a lot of them
Yeah.
as we keep going.
Find some big, massive shells.
Is that an Ex Muda?
Where? Where did you sit? Oh, coming up?
The red one, right? Kind of off to the
Oh, wow.
side.
Look at another big one.
We're at.
Oh,
Holy cow.
yeah.
That's gigantic.
That's trying to live up to its name.
Look at the fish right there, too.
Is that?
Fish love it.
Can't tell. Is that a little barrel?
One looks like it might have-
That's cool.
I don't know.
The big
That
one?
that big one.
Oh.
That was the one I thought you were
I
talking
didn't, no, I
about.
didn't see that one. Didn't see that one until I got here. Oh my
That's
gosh.
huge.
Left or straight ahead?
That looks like an Ex Muda.
Yeah.
Alright.
Let's keep going for a little
Okay.
while.
Is that thing ahead of us a coral reef or a sponge?
Which, where?
The brown
On the
That-
right?
Pirate or hat?
I think it's
This,
sponge.
I think that's a oh, right here.
Like a trifoil hat?
I don't know.
That might be a
Oh my
coral.
gosh,
Egg
yeah,
Arisia?
I figured that.
Tri-corner hat, Paul Revere.
Yeah, that looks like Agaricea there. Is that what you were talking about, J-Rod? Where it's like a little cup? This one?
Small.
The smaller ones, but yes, this would be easier to sample than
It's
glass.
pretty big.
Interesting information.
That's an interesting way that it's
Yeah, it's not
growing.
flat, it's upright.
Huh.
Closer look. Sit down.
What do you think, Lena? Do you want to sample that one? Yes. Okay.
The one on the left, right?
The one on the left, yeah.
This one you can really
This
get
guy.
the wand right inside there.
Yeah.
Yeah, I think that would be good. Hopeful.
Me too.
Like a sponge.
Optimists.
Like what if you want to put your squeezer in the
Patience, persistence.
Is it orange?
Bring forcing down the back away.
Yeah. I'll find
Oh,
it.
there's another.
Probably
Oh, never mind.
What are you seeing?
No, it's a weird sponge with like a sheen on that thing.
Yeah,
Go look at this, it looks weird.
If you put those documents on the nose, and then just step down, take your automatic off.
Yeah.
It's all.
Very red.
It's all.
This sponge?
Happy birthday,
We're
Colin.
doing another,
And so this is something you don't have to use very much now,
Yeah.
because we don't have a lot
like
of.
agar, yeah. Yeah, it's the same thing as we collected earlier.
Y'all doing background right now?
Yeah, I'm just like background.
Wow. SH18677. A different Agaricea than
Oh, okay, yeah.
Yeah, that's, it doesn't have to go all the way. I just wanted to get that.
Alright, we're good on the background.
Okay, and then you want to go inside it, correct?
So you lost the sponge. That's been great.
Sorry.
Choked myself on a gummy bear.
Just don't be sure it's there. I missed it.
This is 03, right?
Sam, did you already say the SH number for this?
Yes, 677. SH18677.
It's been alright.
That's really crazy. Those black spots, the polyps.
Yeah, good. HD4 on our left. This is it, by the way.
This is a sponge.
See, this might be a sponge.
It, I think it might be, huh? Cause
Oh yeah, it's flexing.
Wow, we got fooled.
we were totally fooled.
That, but we're following.
It's kind of crazy, right?
At this depth. It just really doesn't.
How's
You
that?
tricky sponge.
Yeah.
That looks fantastic.
I
Thanks.
just didn't want to get down into that sediment too much.
Yeah.
We're gonna salvage that SH number for something. with
Yeah.
wine.
Oh, okay.
So we have to leave the
Yeah.
SH number.
So staying here though, right?
That'd be great, yeah. Thanks.
Oh, yeah.
Sorry, I had to pull up a piece of sediment going towards the house.
All right.
Okay, I think we can, pull it out, thanks.
We were fooled.
You want a piece of it?
I don't think so. Do we want a
No,
piece?
we haven't done the squeezer chemistry. That's what Kate wanted for sponges.
Okay.
Did it give a signal, did it?
Nope.
So squeeze me?
No, I think we're good.
Okay.
I think they're more interested in kind of the barrel sponges
Yeah.
and the tubes.
Yeah, that one was fun. It was good.
So we're about 78 meters or so.
I'm gonna back all the doors so I can sniff some stuff out a little bit.
We're at 109 or so.
Here, looking good, no
4 meters. There's a coral coming out here. Big size coral right in front of us.
You see something? Oh, yeah.
Fish.
Little fish there.
What was that for? No, sponge. Sponge.
Just a whole bunch of ducklings. Sure. Oh, pretty nice.
How shallow did you guys go in the coral sea?
Can we look over here for a second?
You don't
There
want us to
was,
go that shallow?
What are you, gatekeeping your
looks like another sponge.
No, we got about,
I wonder,
for
can
a
we
very
look at
short time,
a little
we
bit
got
like what
about-
this is here?
What was that? Is that another artificial?
It is. Right there, center screen.
Yep. The brown one?
Yeah, there's your
I
one.
think once we get
Oh,
to,
yeah, two, maybe.
yeah, this is gonna be like a little transition zone.
Barracuda vines?
It was
It's
earlier.
a triggerfish.
The triggerfish.
I think.
It was a very
Or, no, maybe not triggerfish.
What's the one?
Look at the shape. It doesn't look.
It'll kill your, those little ones.
Oh, there's a couple fish here.
Was she blind or clear?
Yeah, Chris was on it.
Is this Agaricea down here too, I guess?
Yeah, I think so.
Yeah, it's much darker, right? Than that one up there.
Yeah.
Or is it just,
We're gonna. Yeah, we rotate it.
Yeah, to the left.
Yeah, it won't be fun, that's just fine. Are we gonna be sampling this?
that would be a really hard one to sample, huh?
I
No?
like a challenge.
Oh. Lena, you wanna do a measurement?
I do, but I'm mineral calibrations.
Oh. Okay.
You wanna pop up and have a look at that black hole while she's doing this?
Sure,
To the right.
yeah.
Who's doing your sit?
Just a little slug over here. Yeah.
Oh, there's a lot of stuff on it.
This might be the one that you're.
It's not something.
Wow, do you know what kind that is? Okay.
It's not this one. This is a little bit more wide. Oh, yeah, not quite this one.
That was really pretty.
Should have back it up a little bit.
No, because that font is mine. Just to show that. So, that's
That's why I said came up.
I try not to lose control. That kind of has 5 years.
You just get pulled off a lot.
No, I just clicked down and it destroyed all of the where I sat down. It wasn't very firm. Days bad, I'm not. Let's see.
So I think we're very on that is. Colleen, we're ready for that measurement.
I opened up that.
Okay. So we're just taking a look at this black coral here.
Harder than making it. You have to zoom where you're.
Without the intention of
Is that-
stars or?
I'm not very stable.
Yeah, we could go ahead and do that, Lena, I think. Might as well, we're over here.
Yeah, just.
What is it, Colleen?
Do you think we should sample it or no?
This is something
Yeah?
Patty's.
It's something pathies. There's a bunch of stuff all over it, right? Oh, yeah. No, is it just particles all over it?
Yeah, it only has like. It screams. It screams spread. Some black corals have like over.
Oh, this is like a plumopathes.
A plumopathes?
Yeah, let's call this plumopathes.
We're just
Yeah,
gonna
it's
call it
black.
that.
It's like Calogorgia black coral.
Are you saying Puma?
Pluma, P-L-U-M-A, like a feather.
Oh.
It might be cool,
You know what it
but
is?
lights off and take a picture of snow.
That's the
Well, it says that because he described it.
Yeah.
I think you're ready.
Yeah, let's call this
I
plumopathes.
should Puma.
Right there.
Are we ready for a, whenever you guys are ready, we've done a background signal. Event 4, all right.
I can bring it in too. I'm pretty far out.
Let's see, 2055 UTC, we're gonna sample this plumopathes. Oh, looks like there's others over there, yeah.
Oh, yeah, that, actually, that white one's a black pearl I saw.
It's a what?
The black one's the one I saw going in.
Oh, okay.
So this one will be 677, which we salvaged from the fake coral.
From the fake.
The
That's
background's already done?
Yes, uh-huh, thank you.
And you want to be down?
Yes. Is there much current, or?
No, but I can just see the particles.
There's particles, yeah. So we're at 67 meters.
So it's,
Your camera?
This was disgusting.
Wherever you can get like a bunch, you know, the branches together.
Looks like we have a signal.
Really?
Signal. The filthy coral.
With a filthy coral.
Telling you happy birthday.
Coming back down. Maybe if we can get closer again.
What about like inside this? Inside of it?
Is that a sponge in there?
It looks like a sponge. It's other encrusted stuff. There's filigree and polychaetes.
Yeah. Would it be possible to try to move like in this more densely?
Yeah, I was just trying to get in the middle of it.
Yeah.
Why got it again?
Oh, I was just thinking over here. I mean, at least from this angle, it looks dense. I can't really tell. Yeah, over here. Yeah. Yeah, it's going back up again.
Oh, go.
On this side of it? It doesn't matter.
Yeah, I don't know if it matters. I was just trying to get to where there's more kind of biomass, where there's more branches.
So like just right here, I'm like,
Sure.
It's slightly elevated.
Yep. Definitely got something here. Well, that's strange. It's a black coral, right? Just keeping us on our toes, I guess.
a little bit more zoom.
Maybe it's freaking out because of all that stuff going on.
Yeah, maybe it's trying to get it off. All right.
Sorry.
You feel good? You want to go back to background?
Yeah.
All right. I think we can move back to the background. Thank you. All right. Well, we got a signal on this guy. Like Sam said, maybe it's trying to ward off that sponge that's growing all over it.
Do they want to sample or?
So we'll definitely sample this one. This is going to be SH18677.
Yes.
Did you get a screen grab, Nicole?
Yes.
Yeah. All right.
Coral quiver?
Yes, we do. Coral cover, yes. All are free except 3 and 6.
I'm just gonna go to 17 first already. Out there.
Yeah.
Yep.
like that.
Just for like
What is that?
keeping track of projects, how long is this dive planning for? How long are you planning this dive?
I think Eric loaded it up for a long one for a 12-hour, so I don't know how much
Wait, it's going to go to where?
actually in there.
Going
Yeah, oh, I just wondered how long, do you think we're gonna be on until
Where?
tomorrow?
17?
Oh, okay, I got you.
I don't know.
I don't think so.
I'm not sure.
I got a question, do we have the repeaters?
Why is it like we're hold on.
Well, we do, but 2 didn't fire. So, and then one has the pink thing on it. So then I'm not sure. So how many,
Yeah, it's
how many are on there? 6?
Yeah, so we have 2
on
left.
the outside.
Yes. Yeah.
It was
Yeah.
disturbing, but I wanted to change it.
We're on 17.
Yeah.
All right.
Changing the lids around.
It makes no sense the way it's doing.
So it matches the.
Put it back on the outside one, and then when you put it on, you just take it off and then put it on.
Right.
Okay, cool. And then we'll see $10.
Sir.
I'm thinking it'd probably be a little better to fill the have it if you want to see water hose.
Pretty noisy, but
Okay.
definitely elevated, right?
It seems like it takes.
I don't think it was very
long
noisy,
time
and
for
I've
it.
not seen
Like,
it as such a
yeah, 5
consistent
minutes.
elevated
Sometimes you guys are
signal
quick
whenever
with
that has
samples.
happened present before.
Okay.
This is just me to
And then
pumps off.
that-
Okay.
even like a
Okay.
few percent salinity difference can really stress. the
Weird. It's a
coral
black
out.
coral, right?
Yeah. Although, yeah, I mean, we haven't seen many measurements of our corals in general this, and we only measured one in specific, so.
Water is really steady.
Yeah. Yeah, that's true. The current. Yeah.
And it's during the day.
It's during the day.
The sun is
Nicole.
still out.
Yes.
You know that picture that Book uses on his presentations where he's got,
California?
where he's got, it on the screen, like you're looking at it like this.
Oh,
and he also
yeah.
has it, that's a plume pack. Like this.
Oh, it's cool. Oh, it's not California?
Is that enough right there?
Oh.
Yes.
Doesn't want to let go. No. There we go. You think that's enough, Sam?
Yeah, I think
That
so.
piece? Okay.
Yeah.
Because it's pretty light and feathery.
Yeah. Because this won't be for trace minerals.
It's gonna be another one of those black holes that's sticking us further.
Yeah, that's it.
There it goes.
I saw you guys fight the other night. I don't know if it was you or J-Rock with it, flying around with the corals getting stuck on everything.
Yeah, 2 nights in a row.
Did Jeremy request that? Black coral?
All right. Well, that was interesting. So that's a black coral, and it gave us a pretty sizable and steady superoxide signal. This cruise has definitely been very different than what we've seen in the past. We haven't had many signals from black corals so far, but also where the site we were at before was really high current. So I think all of our signals were really low, much lower than we would expect because of that current.
Want to look at that white one to the left or back to where we were originally?
Which one are we looking at?
So we have over here.
Got another
We
bushy
have
white
that
black
one right
coral.
there.
Oh yeah.
A few things over there, and then we had further to the left where we were originally setting up.
Oh
and then
right, for the
she was-
Agris,
Yeah, while she was doing the
The calibration.
Calibration,
Yeah. Yeah.
which would you prefer?
We could. Do you think we should do another one of these black corals since we're here?
Yeah, I think it's gotta be a different species though.
Oh, it is.
I think so. I want to get a close look at it.
Yeah, so maybe we could go over and look at those bushy white things that were over there,
Roger that.
and then see what it is. Mm-hmm.
Might be a really cool map.
Kind of looks the same, doesn't it?
It's gonna be pulling you right across
It'd be good. It looked like there were maybe a few more of these black pearl colonies in Illinois. So we could knock out a few Artesia little backwards right here.
Yeah, I think you just come at it just with your safe heading. Yeah, I think that's gonna be best. You're not gonna be sitting down on anything. Just like
Oh, there's some fish down there.
What's that?
turning the flip off.
What's that?
Are we going to the black hole or are we going to
I thought we were gonna take a look at this other black pearl first to see if it was something different in the other buildings. That wasn't like.
black hole first, right?
Yes. Yes, please. These like these algae that look like platy, the seaweed. That's really cool looking.
There's another one of those black holes right there. Just following.
Oh, I see. Yeah, it's small. Looks like it's got a bite finger.
This is a cool looking sponge.
I'm gonna start some
It's like sunset colors.
Oh, yeah, that's a cool spot.
Isn't it neat?
Okay.
So you got a smaller one of those.
Yeah,
Does that look like the same species? Oh,
I think so, but I think we need to zoom in just to make sure.
on the one, on the black hole,
On the black hole, yeah, just to make sure it's.
loud noises.
Oh, it's not the same.
oh, it's not. Wow, pretty.
This is a Tennessee patties.
Wait, a Tennessee?
Tennessee
Oh, you said Tennessee.
Tennessee to pass.
Tennessee patties.
Y'all.
Kentucky
I
patties.
can tell you how to spell it. It's
E-N-N-E-S-S-E.
Tennessee.
Tennessee to Pathies. All right, I got it. Whew.
It's in the Myrio Pathies family.
Tennessee patties.
Already got it in my.
Back, so keep flying for a second.
Move the vessel over toward this.
What are those little yellow things on there? Is it just particles captured? There's all kinds of stuff flying by.
Okay, so what's the plan?
Yeah, it was playing it.
Should we sample this one? Yeah? No?
Yes, yeah.
Yeah? Let's go ahead. All right. You didn't seem so excited about that. All right, Lena, do you want to?
I'm gonna start the background.
All right, we can start the background.
Are we close enough to reach you a solar?
Hold on, before you go in too far.
Coming up. Anything happens, we're supposed to be
Yeah, I'm just gonna bring us a little closer.
up high. I'm gonna need the arms. I need the arms to get to the other side.
Hold on.
I'm just standing right here while they do the background.
Okay, so this one will get SH1 18678.
18678.
678. Okay, and it's a Tanacetapathes.
Calling it Tanacetapathes hirta, H-I-R-T-A.
Oh, we have
Ooh.
species.
H. What was that? H-E-R-T-A?
H-I-R-T-A, hirta.
Wow.
Like Harry.
We got it down to species here.
Yeah.
It's a pretty little bushy black coral.
We're good on the background.
He's moving towards you, so it should be all right.
All right, we're good on the background. Whenever you're ready.
I'm moving in.
Alright,
I'll move with you first. I'm gonna say hi.
well, not to say I can't.
I see a little.
The camera real well right now.
No, you're fine.
Fish moving along in the back there. Kind of tuck in that big bushy part on the top would be great.
Just close to it or underneath it?
Oh, if you could get underneath it, that would be great. I don't know if
This is one of my favorite fish.
Yeah, what is it?
From this.
wow, there's some great fish in the background.
They're just so simple and sweet.
Yeah.
This is my chicken.
That's your chicken.
Yeah. From a day.
I love it.
I like the soft pearls, Nev, yeah. Yeah, that's a little too, but I don't know if that's good.
What are we looking back there? Nothing yet?
Nothing?
Let me get going.
Oh, no.
You might be my favorite.
Erica?
Yep, I was just seeing if I could let off the kind of birds and I wasn't
They're
just.
so beautiful. So
I
bright.
touched it, I'm
No,
sorry.
they remind me of like cherry blossoms. Yes. Oh, I love it.
Anything?
Doesn't look like it.
So this is this one's filthy also.
Well, I think we're good. I don't think we see anything. Are you happy with the? Well, not happy, but are you gonna? Oh, you're trying to take a minute. Okay.
Really cool if we find another bloom of that, so we can get another steady background signal.
All right. I think we're good. Thank you.
No sample or sample?
I think we'll sample. Yep.
Yeah, before you sample it, do you think that Agaricea there is, you can sample that?
Oh, that's right.
I need to have a good look at it, but probably.
Because might as well just Solaris that right now.
Oh,
and
right,
sample
before.
both of them.
Yeah.
If we can collect tissue or sample from it.
Alright, stand by where you are, Jared.
I'm standing by.
Are we okay temperature wise?
You told me what's the plan?
We're gonna Solaris the Agaricea before we move to sample.
Okay, are we in the, should I start with?
Should we start background?
Here, let me have a look quick while Chris is
Hold on, hold on.
using this camera. Yeah, I can sample that.
Okay. So yeah, let's go ahead and start the background, Lena.
There you go, Chris.
So we're gonna look
Alright.
at this.
Can't see where the rock is though.
There's another Agaricea here.
What's that?
Let me move this anyway. What's that?
Yeah, I'm floating down on that side. Hold on. I can't go down anymore.
That's okay. Just keep it easy. I'll float.
That's a really pretty view here with all the cool
Yeah, it is.
sponges and
So beautiful. That like orange sponge next to it, that white orange.
yeah.
I'll get Solaris out of the photo for you.
Able to see anything?
There you go.
I thought it was more beautiful with the Solaris in the photo.
There you go.
You're doing that more for me, actually.
Did you see this sponge back here, Scott? Look at this.
Am I good to go?
How pretty. Yes, we're ready.
No, just hold on. All right.
More Solaris hang?
Yeah, we measured this black coral. Okay, event 6.
Meters.
The SH number of this is 18679.
18679. Wow. That is really
It's like
pretty.
got little eyes looking at us.
So the polyps are up on those ridges, huh? Or is it
They're between.
in the between the ridges? I see.
Little white, white blue spots.
Yeah, that's really pretty.
So kind of like a rain coral, down the roots.
I think each little white
Oh, wait, no,
dot
it's
is
sunshine.
is a mouth. Lips out mouth.
Yeah, very cool.
And there are cactus corals, plate corals, and lettuce corals is my
That's Agaricia.
lettuce. I guess this is like plate shaped. So a plate coral.
Yeah, that's beautiful. So this is an Agaricia. It's a scleractinian stony coral. You can see the ridges, the skeleton, and then those white dots down where the polyps are. I think we're good, thank you. I love all the algal and the macroalgae. It's so pretty.
And this is going into a quiver,
Yes, please.
both of them?
Yes, please. So we're gonna try to get a sample of both of those.
You were the smoke, the black coral, you was the signal. The first black coral.
Yeah.
Slow and steady, just signal. Alright.
Which one you go for? Oh, no, we're following
So you got a signal off of black coral?
Yeah, crazy, right? None of this is making sense.
And you moved it away in the back and it came back and
Well, yeah, it was pretty stable. We're up and came down.
you took it away and went away.
I mean, now that I'm looking at the Agaricia, it kind of makes sense. We've never gotten anything off of a brain.
Yeah, the tiny
I think the
polyps.
ridges and, oh, right. I love this.
Everts is like the stiff gloves that I've had. Not actually up there yet, so you can HDMI or AirPlay or whatever.
We're watching my laptop now.
What's up?
Yeah.
Two is open,
Yeah.
Yes, 2 is open.
I got 18 and 2 open.
Yes, both of those are open.
It's gonna be open and closed for both. And then if I get this whole rock on this one, would you wanna put it in a bio box?
If it would fit in the quiver, that's good, but if we can't, then.
I mean, I might be able to take it up as a rock, you know what I mean?
Yeah.
Would you want that, if that's possible?
Yeah, yeah.
Do you think that was the Neofibrillaria, Nicole?
I don't know.
But I think he asked Alan or something.
Okay.
Oh, wow.
That's kind of perfect.
Oh no!
Not anymore.
Alright,
It actually came off that you were better.
hold on.
Is that what Luke wanted us to sample? Did you see that?
The neovicular.
Yeah.
You see?
Luke Garlow?
He said
Oh,
Yeah.
the sample. Oh, he just wanted to. He just likes it.
Oh, okay.
When he discovered that sponge in the video, it was like a freaking corals for a sponge lab now. Look at all these
That's
fish.
the Irish
And
Mountain
this
fish.
sponge supports the diversity of fish.
Yeah.
See, he just really loves this coral. I mean, sponge.
Oh, okay.
Sponge.
Probably gonna have to go in the bow box.
Happy birthday, Colleen.
Where did that come from?
It's on screen.
Oh.
Thought we were just standing there a little bit.
Every time the bio
5 or
box
10
comes
minutes.
out.
I mean, every like half an hour or so
Yeah.
to remind the viewer.
Is this your birthday all day long?
Don't do that.
Switch you up just a little bit. What did you order for your birthday dinner?
Oh, you need to order it.
I didn't know about that.
I saw like
Steak
French
tips?
fries and steak and
There's
tomatoes
mushrooms.
and mushrooms.
Yeah, there's gonna be
Ready to go to the bow box?
Okay.
Most of the makeup.
Mm-hmm.
Yeah. They're good either way.
So we're, it's 3 days we've been in this whole area right now. They're a little bit more now.
I like that.
All right, we're grabbing this Agaricea that ran away.
Ran away.
And the reason why we're collecting samples is we're trying to pair some of the geochemical measurements we're making with
Alright, bring the camera back.
the microbiome, so what microbes are living in the tissues of these corals,
Box one's coming open.
as well as we're looking at their stable isotopes of carbon and nitrogen. That tells us how they're feeding and what they're eating, so corals can both
Okay.
get food from symbionts living within them. For instance, these organisms have algal symbionts who are fixing CO2, providing carbon to the animal. But they can also eat particles in the water and
Right here, bud. Right there.
feed heterotrophically. So they can just eat carbon. So understanding how much they're getting from their symbionts versus how much they're getting from feeding on particles in the water can tell us a lot about what type of biogeochemical cycling is happening within the coral itself, and then how they're living in certain environments as a function of light and nutrients.
You can see it's mucus.
And then we're also taking a look at,
Like through the sediment, you can see it soften off.
yeah, all the mucus.
Yeah.
Yeah. You can also look at the trace metals within the coral.
Good job.
So all life needs trace metals. So we all of us have enzymes that do all the processes that we need to do, from metabolism to move, to signal our enzymes to do certain things within our bodies. And many of those enzymes are called metalloenzymes, so they have a metal in the enzyme that is the active site, which does all the important chemistry of the enzyme. So these are things like iron and manganese, zinc, cobalt, all these trace metals, which are what's in your vitamins. So if you look at the back, you'll see, a whole list of all the different metals, and those are to be able to help serve in these enzymes that you need to be healthy and have a strong immunity. Well, corals have the same thing. They're animals just like us, and they have a lot of enzymes that have trace metals. Some of the most important ones are an enzyme called carbonic anhydrase. It needs zinc to be able to be made. Here, I was rambling on about trace metals.
Keep rambling about trace metals.
Okay, I'll keep rambling. So I was saying that one of our students is studying the trace metal requirements of corals. So corals are kind of crazy because they live, for the most part, in what we call oligotrophic regions of the ocean, so areas where there aren't a lot of nutrients and not a lot of trace metals. So we consider a lot of these regions kind of the deserts of the sea, and yet you get these beautiful coral reefs and coral communities. So that they're pretty fascinating. They can live at really low trace metal requirements, nutrient requirements. But this also means that they're really living at the threshold of life. And so when there's a stress event, say like the ocean heating up or disease coming through, they're really already living kind of at the limit. So one of the things we're interested in is, can we help corals become more resilient by providing them vitamins, just like we do, just like we take when we're starting to feel sick. When we feel a cold coming on, you take zinc or you take a multivitamin to kind of boost your immunity. We want to understand, can we also boost the immunity of corals? But to do that, we first need to know what their nutritional needs are. So we need a baseline. And so one of the students on this cruise is collecting pieces of these corals, and she's going to look at how many of these different trace metals do they have, and would they grow better if we gave them just a little bit more. So my colleagues at Hui like to tease me about how I'm trying to make vitamins for corals. And I think that would be pretty cool if we could actually do that. We could provide them with kind of supplements to help them grow better, faster, and have a better immune function. Yes, Flintstone gummies for corals. That would be pretty amazing, right?
And it's dinner time.
Half hour.
So understanding their nutritional needs could help in a lot of ways. So a lot of the coral restoration is going on right now, in particularly in shallow reef environments where we're losing a lot of our corals because of coral bleaching or disease events. There's been a lot of loss of coral ecosystems in the Caribbean lately because of a disease called stony coral tissue loss disease. And unlike a lot of coral diseases, that one has been kind of indiscriminate. It's not specific to a certain coral. It's kind of hitting most of them. So it's kind of wiped out a lot of reefs.
Does it hit deep sea corals too?
There has been some observations of stony coral tissue loss in mesophotic areas, and actually In Vieques, which is just around the corner of where we are right now, there was some observations of this skittledy in that area. Actually, Yanel is, she's not in the room right now, but she's studying that for her graduate work here at the University of Puerto Rico. And so a lot of the coral restoration efforts that are going on right now involve growing corals in nurseries and then outplanting them, putting them back out and growing them. So if we could build stronger, more corals with a better immune system, they could hopefully then have a better chance of surviving once they're put back out in the environment.
We're gonna have to just fly around and look for one.
Fly around.
Really hot.
Oh, yep.
It's getting hot here.
It's getting hot.
67.
So Sebastian's getting hot, so we're gonna fly around for a little bit.
Just running a sample, not a good one.
Oh.
But it's okay. I'm just trying to make
G-Rod, do you like to play video games?
Of course I do.
Thought that was a good question to ask.
So we're now at about 63 meters. Pretty shallow.
Are we wanting to go back to that other set of those coral things?
I think we're eventually gonna wanna sample those two big Agaristia right
Yeah.
now.
Yeah, if
Round out the replication for that species.
yeah, we might see more if we just.
Sure we will.
I think at this depth we're probably gonna see them. Oh, here's a little yellow fish here. That's the extent of my identification of fishes.
You're like, Trace man, super rocks, ooh, and a yellowfish.
Yes, you can tell I'm not a biologist.
That one, yeah. That's I think the first one we sampled?
The other one's the regular.
Yeah, that one was here. Here, and then just lower is the.
Colleen, do you think we'll stay at this depth for a little bit longer, like 45
They're the
minutes?
ones that are right there.
I'm not sure why. But
I'm just trying to give you the decisions.
oh, eDNA decisions. Possibly. I mean, I don't think that we're gonna. We can kind of move along this depth, maybe.
But yeah.
Similar corals.
Is it possible to move kind of west just along this depth, maybe?
Marker right here for these two guys.
Yeah. I'm just thinking where the point that I had originally, the waypoint, is where we know that there's like kind of a nice reef. I'm just wondering if around the corner it might be a little denser.
Marker's like, ooh, that's true. Waypoint's like that setting up this sky. End up with 33 for no reason right next to the 29. We're gonna go west.
West?
Yeah, we go west.
Come back up on those floods.
Alright.
See, up above you doing. You get your setup together.
That's it. It's fine. Back off a little bit, get a little broader view. oyster coral.
There's another one of those sunset sponges.
Sunset?
Doesn't it look like a sunset?
The neophyte there. It does.
Italian garden.
Talking about Olive Garden.
Olive Garden.
That's all Olive Garden over here. And Sam was like, how'd I got that from? We got those burritos.
You should start seeing madrasas.
Another one of those hard cores at the top of the screen.
Okay.
Lionfish.
Oh, there's another big sponge.
Oh, that looks like also maybe
That really looks like a volcano. It's got a volcano shape.
Short.
Yeah.
Oh, and the lionfish right next to it.
Oh yeah, the lionfish there. Yeah. Wow. Yeah.
There's a whole bunch of that hard coral right up there.
Agaricea on top.
Oh yeah.
Ooh, really nice one off the left.
Wow, look at that on the
big
left.
sponge.
Oh, that's the sponge, right?
Yeah, there's another. Look at this big sponge coming up.
Oh yeah.
Cool. Bunch of that stuff.
Sam, if you wanna go eat, go eat.
Sponge on the left, corals on the right.
That's an interesting.
Yeah, this sponge is like tricky, right? Is it?
Yeah.
Is that a sponge?
Yeah,
That's
we
one
we
we
got
saw
tricked
earlier.
earlier and measured one because we thought it was Agaricea.
This sponge looks cool.
What is this red?
Looks like a sponge.
I'm looking for Siderastria. Like we should be seeing
Big
this.
polyps.
Yeah. And also, like we should start seeing madrasis, and
63 meters.
at this depth we could even see Porites, which would make me very happy.
I know.
There's a big black coral on the line.
That's a cool sponge.
What's that?
We
Like,
could
catch
drop
on
it.
those-
We could
those
drop a
corals.
target if we want.
You want to drop a target here? Scott likes this sponge.
Dropping the target.
Yeah, just I don't know, yellow sponge, large yellow sponge,
4 meters left of west of the last.
double.
Okay, I'm gonna go
4 meters west of the last marker.
Might be a. Might be.
Yeah, and then we didn't go very far, did we? We're gonna have markers like all the way. across.
It happens.
We name it Scott's Cool Sponge.
I'll text you guys.
Yellow. I like that. Yellow. Yellow.
So did you see? I
There's-
put
there's
this
deep
in the
sea-
WhatsApp.
Deep sea coral.
Yeah, there's a
Yeah, I saw
black
that.
coral, so that we could all see, like this. You know, that's like a madrasis, I think.
You put it targets to come back for it.
It's funny, right? Like without the benefit of the lights and just the sunlight, it all looks kind of gray and
Yeah,
ugh. And
yucky.
then you light it up and you're like, oh my.
"Ooh, wow." Yeah.
No, it's just-
16 hard coral down there.
Yeah, down is this one here?
To the right.
Oh, to the right. That one?
No,
Your other right.
Just to the right of the first thing you pointed.
Oh, to the right.
Yeah, there's, it's like just above center screen right now. Okay, really cool, big columns.
Oh. Oh, this one here?
Yep.
Oh yeah. Is that your Siderastria?
Just look really weird.
Is that your Siderastria?
Is that,
Oh
see
no.
that bottom left one on the red?
Hmm.
That's what we've been sampling, right?
Yeah, that's
Yeah,
Agaricea.
so that one.
So that's different.
That is definitely something different.
I don't. That might be Montastria. Is that Montastria?
Yes, that looks like it.
It's a bubble. What do we call it? The gumdrop coral because
Polyps so big.
they look like little gumdrops.
Sit down if you'd like.
Is that Montastria cavernosa?
57 degrees.
No, Montastria annularis.
You feel comfortable sitting down?
Yeah, 57's in normal.
Thanks,
Not you.
Oh. Thought you were asking me. Please sit down. Sir, please sit down.
Every time I think I'm gonna do one ram, I do the other ram because that way it's to the left.
Rockin' and sockin'.
I think.
I think we'll find more of it, so it's not a problem.
Where'd it go? There, that's it.
What happened to our other?
There's the yellow sponge.
Interesting, Montastria is another one that I never get a reading on. So there's like Passalopera and Porites and
what would you do with it?
those I always get signals on.
The telescoping.
Pointing stick.
I don't know, it does this, you do that.
You don't have the other one over here, do you?
Yeah, this one?
No, there's two of them.
No, I don't think so.
There were.
Oh, here's one.
Aha. Nice.
Thank you.
So how come?
You know where you're going?
That's our big. Yeah, that's our big yellow sponge.
Yes.
Oh, I see.
Yeah, I still want to land out on the other ones we want to look at. It's gonna be back then.
There's something up here too, yeah?
Starboard, that's-
Yeah, I guess.
You might be able to do it with your rock and stuff, just like in there.
That might be sponge.
You think that's a sponge? Yeah.
On a border would be enough.
Everybody needs to keep their eyes out for Porites. The mustard coral.
I knew what to look for, I would.
I think it's yellow.
It's like a little mounding yellow coral and has little nubs on it, and it always produces a ton of reactive oxygen species, no matter what species. Actually, any Porites species. Look at that. This is amazing. How much color there is here. Wow.
So what type of coral is it? The
Porites. If here we should find Porites asteroides.
I heard gumdrop.
This
Gumdrop.
G-U-M. Separation D-R.
this one is the gumdrop. This is Montastria cavernosa, I believe.
Is that what we're going for?
Yep. I was saying we should keep our eyes out for Porites because that one will be our positive control.
I'm just slowly moving up because of umbilical jet.
Erica, do you want to do a background signal?
Yeah, we get a spot.
Is this a good spot? Yeah. And then we're going to measure this Montastria. The time is 21:46 UTC, and it is 63 meters deep. And we're going to look at this Montastria.
Bring your phone back to me. That's perfect. It's still up above it, but we don't yell at it.
And about half a meter out.
Stay up here for a few minutes.
If you want, this is kind of like a nice spot.
Ulva almost.
Doesn't it? It's beautiful.
Or
Yeah.
Padina.
Is that not like set out,
Algae.
and holding it in position? If you want to try it, I mean,
We'll get done with this.
yeah.
Okay, we can measure.
All right.
Right.
Look at those polyps. They're so pretty.
Can I do a science, a little bit of zoom? Not
yeah.
yet.
That's how I was gonna.
Okay, you go.
Yeah.
Stop it.
I know.
Step away from the zoom.
I believe this is 680.
What's that little thing on it? Is that a fish?
I
Yeah.
think that's a little fish.
It's a little, yeah.
Don't suck it up.
A neon wrasse, I think.
Sorry. Yes.
so should I write down?
I got it.
So these, do you know if these polyps are open or closed, or like?
Well, it's hard. So it's actually not the whole gumdrop. There isn't the whole polyp. The polyps inside
Yeah.
that I can't remember what that's called. But they look like.
Calyx.
How do you spell this one again?
G-U-M.
Drop.
No, this.
D-R-O-P.
Okay, I looked up.
La Paguera. La
M-O-N
Paguera. La Paguera.
M-O-N.
Montastria.
T-A-S-T-R-A-E-A.
Oh, this is good. This is a good highlight.
Be right above it.
Yeah.
Come on.
Well, this one never gives a signal in shallow reefs. This is the only time we've ever gotten a superoxide signal off of this one was when it was diseased. And then we had a huge signal where the lesion was.
Okay, well, this one looks healthy.
This one definitely looks healthy, but I think you can see the polyps like out here. But yeah, they're tucked into these little gumdrops, whatever these little gumdrops are called. I can't remember.
Someone on the chat says Elactinus cluna goby,
Oh, the little.
little goby.
which is maybe the fish that's swam off.
Yeah, that little,
Thank
yeah.
you. I'm not seeing a signal.
No signal?
Nope.
Maybe touch it?
Sure.
Give a little bumpy bump.
Someone else says it's Elactinus evelini, blue stripe with a yellow nose, so it's Elactinus something from here.
That's a little touch.
All right.
Nothing.
Nothing.
Look at that.
You see that out of the corner of your eye.
Got a little bump there, Erica, but it's really small.
Yeah, we'll see.
So we're gonna want to sample?
Well,
Let's see if that goes back down.
You got what a sample or no?
Do you think you can without like grabbing the whole thing, or it's pretty small, huh?
Yeah, it's pretty small.
It's
Size.
Yeah, I can try to pop off. This bit.
maybe try to pop off something on the bottom there, if possible.
Yeah.
Maybe it was a
Yeah.
tiny pulse.
Yeah.
Try to fly it a little harder.
yeah, but if you rescaled that.
If we rescaled that, I definitely think there was a little bump up,
If I
but
spin in, we'll be able to access it.
This is good.
Wow, the family and genus are monotypic.
I think we'll be fine.
Hold it?
Yeah, you're good, okay.
Yeah, fine. Alright, I'll just hold it.
Really?
yeah.
Something, I guess.
That's a tiny little guy, huh?
See what happens when it into the work first.
What's that?
So 63 meters, we have a Montastria.
I'm not touching you.
No. Oh, a little bit there now, yeah.
So that.
Yeah. I can always stick the pointy end of the wand into the sieve. Get this nice and firm in there.
Yeah, I'm just
Left to.
trying to balance. See if I can do this stuff all.
I might be able to come out and give you a better angle if that'll help the camera. Yep. We'll bring it back around. Alright. And I can bring in Rocco.
Yeah, it's a weird angle, huh?
Swing your arm more. Mm-hmm.
Oh. Look at all that mucus.
Hold on. Yeah. Spin you back a little bit.
What's that?
Maybe spin you back a little bit. Back and around. Oh, to the right?
Yeah.
Oh, coming off. Hold on, I can't do anything right now. Yep. Alright, I'm pushing in now. Yep, stable. Alright, no, I'm not. Alright, we can fly back in there.
I think I got enough of a ledge now.
Alright, hold on. Watch your arm. Watch your arm.
I swear the next one we pick will be easier.
Somehow I don't know if I believe that.
Just giving you a good challenge.
It's your birthday, we'll see.
Looks like there might even be one up there. Does that look like another Montastria right there? Maybe. That would have been an easier one, I think. Possibly.
Yep.
Made a little hole there, a little cavern.
Yeah, I'm trying to pry it.
No, you push it now. Solid. You should try to get to that at an angle. Good view on the sit.
This is somebody. This is a little guy, right? That's got polyps.
Yes, it is.
Not that we want to measure that, but that's definitely.
Yeah, it's somebody. That's the one that maybe with it. Is it the one with an S?
Is that Siderastria? Maybe. And maybe even up there. Might be a Sid too. There's so much going on right here.
I think it's on there. I know what I did.
That thing will not break. I don't think I'm gonna be able to get that off of there.
Okay.
Sorry.
That's
Run it.
no, that's all right.
I can just run into it.
No.
Yeah.
No, it's okay.
I'm not gonna be able to get that run straight.
Thank you for trying. That was yeah, that's a tricky spot. We'll I'm sure we're not gonna be short on corals here, so we should just wait until we find a better one. All right, so we're gonna,
Filter on the surface, but they're decent.
remove that SH number, or just keep it?
Yeah, I'll write.
But I would say
Okay.
it's just closer.
Yeah.
They're 143 meters from the vessel. And there you go.
All that.
Good.
Scott, did you go eat? No? You're waiting for me? Then we'll have Sam at the helm? Nicole at the helm?
You said there was another one of these right above us?
I'm not sure, but I think we could move for a little while and check things out. I like how we're just kind of moving along this this depth gradient, this depth. There's a lot to see. I feel like it's getting denser as we move to the west.
Bringing these ramps in.
Just going to the west.
Oh, just going to the west, okay. There's lateral along there?
Yes. Look at that fish. What is that? I love these macroalgae.
You could just point, you don't have to actually. Yeah, those new ones.
I'll turn on the door.
Is this a sponge, you think? That looks like a sponge.
Looks like a sponge.
Yeah. Wow, so cool.
And out here, keep going.
Let's just keep.
Mm.
I love all that macroalgae here.
Mm-hmm.
I don't know what kind it is. I don't have any pictures of. Maybe someone online
Could
knows
be
what that algae is.
Eudodia.
Halimia?
Is
It's
this
not-
is?
I don't think it's too big for a Halimia. I think it's a Eudodia. There's a little Exmuda
Oh yeah, look at that one.
little sponge.
Yeah.
I think it's a Eudotia.
Disturbed.
Or a Padina.
I'm ready whenever you are.
I have,
And this red, I guess, is a sponge.
Laying right here. And this lid is laying right here. There you go. So if I put the bungee right there. so-
Maybe not. This is a Calcareous. That's a Eudodia.
Oh right, the fans.
Yeah. But I'm not sure.
Yeah.
I'm not sure that's it. I don't know how leafy that is. Is it calcareous or is it
I don't
floppy
know.
and leafy? Look at that
Might
little
be something
little
in
purple
here.
sponge right there. Bottom.
Yep.
Cool. Little parasturgeon fish up there. 60 meters on the nose.
Stop, okay?
Hmm?
Just call out if you want me to stop.
Okay.
I'm just trying to go nice and slow
Yeah.
so you can see what's here.
We've definitely seen some of the barrels. There's a deep water sponge or whatever. Or I mean a coral.
Oh, I see that coral there.
Here again, nice little section. Oh.
Oh, and look at those
You wanna get a
big
zoom on that one?
Agaricea there. You want to zoom in on that to look at that?
Sorry.
Wow. Wow. What are these? Is this sponge too, I guess?
Yeah. Oh, there's a sponge. It's got
Yeah.
might be another X muta.
Wow. An Agaricea here, bunch of plates.
It's funny joking on chat.
Record X sponge.
A calcareous algae, yeah, so I wonder what species that is. It's really cool.
Okay, see, I said it's some kind of Antipathes.
Black
this,
coral.
this purple.
Yeah.
Yeah, I think that's a Eudodia. Some kind of Eudodia. Eudotia.
Mm.
It could be a Padina. The Padina looks like that too. Padina has that kind of,
Mm-hmm.
but it's really leafy.
Mm-hmm.
Eudodia is like hard, stiff. I can't tell.
Neat.
You're fine, I'm just
Yep.
looking up.
Okay.
Yeah.
Just kind of checking things out here. There's so much.
Yeah.
Yeah, I was at that Antonio.
What is that? Is that a sponge?
I'm sure it's a sponge.
So
Sponge,
crazy.
yeah.
Is that sargassum? Can't.
No.
It's a
no, I
brown
don't-
algae.
Antipathes,
There's a
but why does
Dictyota.
it look like an
That might be Dictyota.
Look at this sponge, this bright orange one down here. Oh, and there's some little corals down here.
That Dictyota. Oop.
Yeah, that's sure.
Possibly.
That's it.
Yeah, maybe.
It's weird. This looks like it's about to get really good.
Yeah.
10-meter shower, so that adds up.
Yeah,
Once
yeah.
we get to 50.
Pink sponge. Looks kind of like the volcano sponge, but younger, smaller.
Mm-hmm.
Which I think, yeah, was an X muta.
Oh, and look at this yellow one back here.
Is that like an Aplicina? Whoa. Or not.
All right. I think we have exhausted our staring at a spot. Thank you.
We're still 60 meters, yeah.
Yeah, I thought it would just be nice to kind of go at 60 meters, just kind of move
Laterally.
along west, just to
Reverse
kind of
laterally.
see how much variability there was just, along the wall. And this is definitely much denser over here than it was.
Bridge, make sure we're not gonna cross over into that fishing
Fishing
area.
more, try to avoid that more. Yeah.
For the vessel, or for all of us? Bridge, are we?
You think those Antipathes? Do they have like these really small polyps or something?
Yeah, go ahead, everybody. Yeah, I think it was some kind of where we are with respect to the fishing here.
Yeah, sure.
Just to watch.
Sure. Me here?
Yeah.
Oh.
Calling you on your dinner.
Can somebody watch my eDNA?
Yeah, we've got one forward of the beam and off to starboard, so if we carried on going straight north, we should pass clear of it.
Are you running it right now?
Yeah, we're trying to run this set before we get to most of the times.
So that Montastria you guys are after,
Yeah.
did they abandon it, right?
Destroying it.
Yeah. Well, you know, I think she saw a sickness
Said that you guys
spot on that, so
Riverside one.
they knew it had died in Lanesville.
Or past the first one, the other one's here. But they said that means we're past Creek. Typically between buoys is not good.
I have to find somebody who can press stop.
So should we stop traversing
Watch this.
west? Is that the?
Bridge, are we?
Is Bianchi eating?
Yeah, go ahead, Ruby.
Hello.
Yeah, he
Yes, just trying to understand where the boys are at. We're currently working a bit towards the west, but if we need to stop going west and just go north, I just need to know. Obviously, we don't want to cross between buoy sets.
Yeah, I appreciate it. Yeah, I can see your boys on your land floats on the surface. I'll go and have a look now and I'll get back to you.
Yeah, I'm concerned with the fishing buoys, yeah, as well as, of course, our catenary with our buoys.
Yeah, so I just had a look out the window and the fish club that's ahead of us to the west. Further west, the land floats it looks clear at the moment, the land floats are passing to the void.
Okay, so we don't want to go any more west then, yes?
Correct, yeah. No,
Roger.
there's more room to the east to get my drift. To stay exactly on the beam of the ship, 90 degrees off our heading, you'll be well clear of both of them.
Okay,
So we're gonna start going north then, yeah.
that sounds fine.
Okay, we're gonna start working our way toward the north.
Yeah, very good. That sounds good to me. And if any of you guys can, you probably be able to see it from the deck as well. You can see the two fishing floats, so you'll get a good visualization of where things are.
Roger.
Sam, do you want to take the helm here for a minute and go grab dinner?
Well, I can say when the mic's not to.
If you see
Say when the mic's not
see anything interesting, like I've been trying to find kind of some madrasas. We should be seeing it at this depth.
What are the madrasas like this? Are they like thick fingers?
So there's if you
Oh,
go,
the pictures you sent.
yeah. So there's
Is that what, oh, there it is. Like, is it?
yeah, there's a bunch here, and then there's Psittastria too, and then we should start seeing Porites. But any other like black corals or octocorals or anything that you see.
Okay.
All right, I'll be right back.
Whoa. Can we get a look at this thing here? Are we after something right now? Or what are we doing right now?
Whatever you need me to do, man.
Let's just take a close look at this. I think that's an octocoral. Oh,
Oh, I get to get my eye.
oh, there's three of them. Ooh.
This is the
Ooh.
problem with you. The dynasty just gets crazy with the pointing stick.
No,
It's ready.
that's a lie.
It's just his arm. It's a
Lena, you
natural
wanna
extension.
see this octocoral here? You wanna Solaris it?
Yeah.
Okay, let's set down. There's 3 colonies here, so if this gives us a nice positive signal, we'll probably want to hit all 3 of these.
Okay, so you wanna start with this lower one then?
Sure, yeah. A lot of fun stuff right here.
That's just fine. Nope. Yeah, it's not.
There was.
We're just saying Oscar Coral for now. You might get an ID in about 52 seconds. He's getting real close to it. Oh, you're lying. Weird.
Doctor Georgia.
Yeah.
You got your background?
Yeah.
See through images. Sorry?
You got your background?
Lena,
I
you-
got one. We got the back.
Yeah, she's got the background. Uh-oh. I'm gonna steal a gummy. Steal a gummy bear. Yeah, this is 18680. Not sure what to call this species just yet.
Okay, yeah, you should have room to come across.
Ooh, this is a good one.
Cross first.
This coral might have algal symbionts, given its color, that kind of brownish, greenish color. Anything? Nothing? How?
You happy with that location of the
Yeah,
wall?
Lena, what do you think? It should be a little closer? Or is that location?
That location should be fine for measurement. Thank you. I'm just sensing.
Alright.
Yeah. Can we take a sample now?
Alright, still.
bad timing.
Yeah. What's our time on UTC?
2219.
2219. What's our depth?
54. 55. Depending on where you look.
I don't know which one is open.
It's
The one on the outside.
18's open and 5 are.
Okay, 5 is outside.
Yeah, so which one do you want to go for?
Which
Time's up on
event
Saturday.
was that?
How much is this you want, Sam?
Like 8 inches.
Just like one of the branches sticking out.
Yeah, maybe where it forks, so you can get like 2 branches of that length.
Oh, no, it's okay. This actually works out perfect. Okay. So right now, this pump just reached 75,000. So I stopped it. And then I'm going to.
So
once you reach a certain power, you stop.
And then I'm gonna log in this one. So
I think I see what.
this is a 4 size comparison.
down there, you see, right there.
Yeah.
See where it branched out?
We do exploratory ones that
Yeah.
we're moving, but we're not
Yeah, like
prioritizing that right now.
that part.
So it's pump one, or one, lubricants on pump, so it's not always
Yeah.
the same pump.
Yeah.
Oh,
So I just finished
yeah,
pump
right
one,
below where
which is
you're
the
at.
second pump of the series.
Right.
The filter size for that was
From there,
1.2.
just past that, it looked like there was 2 of them together. See it underneath there?
Oh,
Is
yeah.
there a legend?
Yeah, there's a legend. I'm gonna also write it down.
And then back up.
I just turned it off. Press it. So,
Well,
that's that. But right now,
now
we're sampling
you
it
can-
below, so that.
Oh, it's kind of, yeah, just above it, you just see it, yeah. Like, yeah, where your jaw is, and then just kind of rotate, the other way, and try and finish the top of it. Right where you're touching now, you see it? Oh, it's branched right there. Okay, or you can go
Wait,
for the one
I'm
below it.
missing the.
So, I go to start next.
Which way?
That one that
Pointed down to
you're,
the right.
no, the one that you're holding right now, you see how it's got 2? On the jaw right there at the top, see how you got one piece going off to the right, one piece coming straight up? Yeah, that one.
Yeah, 75.
It always starts from C1 for one.
Yes. Yeah, it might be like within 100 or 200, just because when it's moving, like accidentally. So I'll press start after they finish this sample. It's clean enough. And
I don't know which pump only.
we're gonna use
So
one
like
too.
right there.
No, you were actually on it. Can I borrow this for a second?
Yeah, tell me where you want it.
Yeah.
You see right there?
Yeah, one that goes this way, and one that goes this way. Grab it right there. Should be alright, yeah.
This is like the last one of the series.
Yeah, you're talking about right.
Yeah, right where you, right here, like above here, right there.
Yeah, that should
You got
be.
one branching down that way and one branching out that way. Or you,
I think-
or that one, or that one.
What about?
There's like a
can
couple
you-
around there.
It would be easy to just cut this part right here.
See
So
that one?
I have like a climate
Where's
estimate.
it?
So,
Sorry.
except for these data, yeah, this could take like a half an hour. It's kind of hard to time it. Yeah, so I'll speak to really about an hour. So after this coral is put in a quiver or biomass is sealed, and there's no dust going on, do you think you could just press start?
I think just like the lookup.
Yeah, and just watch. Yeah.
So when it's around 75,000, just be stop.
Yeah.
And once that gets stopped, it's like.
Yeah,
Stop
just,
everything.
yeah. Like you click start just right on the time. Start and stop.
So what do you want to know? Like, okay, so for this one, I'm gonna click, let's get started once the total is, right, we're putting the light box, it's
Yeah.
on your song 7. So, there, under
They give a little cutter, just scratching it.
onto,
Like,
zoom it out.
Yeah, I might really trust you. You'll press
Yeah, it's
start
like-
here,
I think
and
you
then
got it
you'll
there.
click EDNA sample.
I really need to like know this myself.
Yeah.
It's-
for sure.
Or
They
text
could have
messages.
got much, but that rocket.
Sample ID.
Huh?
Those cutters got messed up by the last.
Yep.
560.
More samples.
The start and stop is, it's, we give a different number for.
Ooh, there you go.
Storage location
So,
is.
I don't know how that's all goes in quicker, it's quite wide.
Yeah, these cases speeds good.
Because we started at zero.
Well, we can try it. At least see what it's like.
Filter
Which one
water size.
you wanna go for?
Is 5.0.
One in front of the 5, okay.
Pump status.
Coming out with the porch.
Submit.
That's like a noodle.
What's for dinner?
Camera.
So there's steaks, sirloin steaks, there's salmon steaks, there's fries, there's roasted broccoli. Steak also has gravy, there's like grilled tomatoes, like tomatoes. So,
Pretty good view
yeah, it's
there.
actually pretty good. And I think there's also mushrooms.
The tuna steak.
So
Tuna.
exactly how it is inside the ass.
Hopefully it just folds in there.
Yeah, right. Exactly so what
A bit further.
Oh my gosh.
So it's just sitting there with the water behind it, so it's just like the water in there. So once you press start, this filter will start, this pump will start pumping water in past the filter. So it will first remove the dry water and it will start pumping salt water through past the filter.
Okay, so it's like kind of like just taking out the water, pumping it with salt water.
Yeah. And so this volume is how much water it's pumped through.
So let's say after this one, like right now it's hitting at that blue box.
Yeah.
Those are not the bio boxes. The bio boxes.
Yeah, bio boxes are here and these are clippers.
Yeah, exactly. Bio boxes come with this happy birthday on.
Yeah.
So yeah, just get started. Like once it's closed, everything
You might have to get it started at the bottom, because
start.
the stop and hope it folds over.
474,000. Follow that guy.
Yes.
The really only thing that's showing there is like the storage location, right? And the filter size.
Yeah. So like this is all the right information.
Well, let's say for example it's 1.5. That's not gonna be the same filter or size.
Oh, if you use pump 5?
It's gonna
No,
be
it's gonna be a different one. Today we chose pump 0 and 5 as 13 as control. So you want to pump that one.
Okay.
Where's it
So
at?
I've done
Alright.
0 and 1.
Yeah.
So it's just following this order.
Yeah.
So for 2 it's 5.1. That's the 4 size.
Yeah.
So then for 3
You know, you'll get
right,
the soft
so
tones there.
after we the 2 and log it, excellent. So I just have to wait until I do another like 5 box.
Come up just a little bit.
Yeah, but see, these are nice sites. So here, we're not seeing a lot of traditional square heating girls, and really wants another suite of pumps to
You got some
filter
length if you wanna try
where
it.
there's a
Go
lot
this way and
many
bring it back this way, you know what I mean?
diverse
Try and get it one side,
hard corals.
like that.
So that's when I would start these. So we'll see how the rest of that goes. I might not
Bring it
start
down a
these.
little bit, twist it in,
So
and then
this
bring it
can
down.
now,
Yeah.
this is like how it goes.
Yep.
Yeah.
And then like just every like after like I log in everything, go to screen.
Yeah, you're good. Yeah, you're good. After it stops, then done.
I don't have to like do another one.
No, this is
So
Looks like a stick figure.
Right?
So it's going to be EDA sample, right?
Yeah.
EDA sample with the follow this guideline.
Yeah.
So for pump 2, it's basically this information. This is that. So I just gotta like put all that.
Wow.
There you go, put that part in.
Additional taxes are really like.
No, you don't have to fill it out.
Watch
Sometimes
the
that's
other side
it's usually
come out
like
once you go away a little bit.
saving time that happens, right?
See where it's right on that?
So you did tell me to write down.
It's
the
alright,
time.
it'll go down in
Don't worry,
So-
if you do this, you don't have to worry about writing down.
Perfect. So as long as I stop and click that immediately, it should be good.
Yeah.
Oh,
And then once you
it's alright.
press this, what's nice about it is pushing all metadata associated, so then like
Yeah.
X, Y position of the ROV, lat long,
No, I didn't break
temperature,
it. That was the
drive,
end of it.
altitude,
Okay.
all those things. My computer
Now you
should
might
not
be able to
lock,
just push it,
but if it
stuff it
does,
the rest of the way, you know what I
my
mean?
password is this.
Let's see, hold on, I haven't seen. That's what I don't get it though.
It's just a good number and that just used to be in Pennsylvania. So.
Oh, wait.
Yeah.
Oh, I got you read something
Now
else.
I just pick like different sites, like different species names. So that's why I passed it to my
Okay, cool. So,
Yeah, Main Street down.
alright, so I just gotta be like on the lookout for that next time they close some biobox.
Yeah, whenever they close this river. So I'm gonna hang it up.
Too far out.
You're gonna sit here.
Okay. So they're gonna close this door, right?
Yeah.
And after that, they're gonna open a biobox.
I don't, they might not because they don't have the numbers.
So I should just start as soon as they close that door?
Yeah.
And it's pumped
There you
to
go.
Yes.
Just
Yes.
far away.
just close.
That's where, yeah, the mouse is right there in front of you.
So they close it. Sorry, I'm asking a lot of questions.
No,
I just
it's
wanna-
okay.
Was that a crack in the quiver?
So that's gonna close, right?
Yeah.
Okay, close, whatever. Let's start. Just
Wow.
read it out. Be on like.
There it goes.
Yeah, just be watching this number is going to update.
That's
every
good. I think the stopper will push that down.
Think so?
Yeah.
So it's either like.
Where is it? Good question.
So
Laying over the
And then
front,
it
see on
updates
the
like
far
every 5
side where we go from the left, that's where it is.
Alright.
Okay, you got this. You got this.
Thank you. That was a tough one. I accidentally asked for too much on that one branch.
That one, I'm not even doing anything else. Okay.
Yeah. Like technically.
It's a lot longer than I thought it was gonna be.
Fine, but I'm gonna wait until they put that lid in that close.
It's on the background, not the forefront.
They should be here.
Okay.
Thank you.
¿Tiene la casa, verdad? Sí. Entonces, no, no sé, una cosa así, así que hace un plano, para que se con la casa y yo. Wow. Entonces, cuando tú necesitas qué utilidad a mí. Ayer David se me compró un café. ¿Qué? Que David se me compró un cafecito tan chulo que le hago la web. Me encanta, yo lo tomo para mí. Yo. Me encanta ver tan sensibilizado. Cuando ves casa y te pones, me pone a pensar. Tengo que esperar y disfrutar de ese momento. Y lo entiendo porque como que todo está explicando lo que voy a ver y
Feels warmer in the front. I don't know. Maybe use this chair, this blanket, and crap on it. It's too warm. I feel like I'm sweating. Yeah. Okay, let's pass on those other two colonies of this for now. Try to look for some better species. Colleen wanted to find some particular species of hard coral that she thinks should be around here at this depth.
Happy with the snow on that thing?
I guess those other two colonies. Lena, do you think we should Solaris those other two colonies of this?
maybe if we could do one more, I think, is it the same species?
Yeah.
Yeah, maybe if we don't get another signal on the second one, then we can move on.
Okay, you wanna try the one just up here on this next
Yeah.
block? Okay.
Yeah.
Am I okay to start the background signal? We close enough.
Yeah, it's just it's like a meter in front of us.
Okay, I'll start collecting background now
Okay.
for like a minute.
Then I'm gonna, since these two colonies are right next to each other, I'm gonna suggest we just do them both. So we have three, so good replications of this one species. Which way is the current going? I guess at us.
I think it's coming at us, yeah.
See if I can figure out which species this is.
so that might just might be.
Yeah.
We'll let you go.
Oh, try not to sneeze. There we go.
Sorry, was that? Alina's ready.
It's crumbly.
Oh wow. Yeah, it is really crumbly. Knock the whole front of that off.
So.
Okay, this is going to be SH18 moves, 60 arms, 18, 60 right?
Event 9 on our side.
What?
Event 9.
Event 9. We're still at 55 meters, Juliana? Are we still around 55 meters?
53.6.
No signal? Okay, can we measure the colony just to the right of it now? Do you need to reset it if we move to the colony to the right, or can we just do that real quick? Okay. Lena, the second colony is SH18682.
Thanks. I'll make it event 10.
event tent. Okay. Do you think it's worthwhile to bump them so they follow the track to see if it's missing signal? You hear that? Just a little boop. Not with the tip, but maybe like the side. Just, yeah. To where we can see the polyps retract and then. Yeah, there we go, they're closing. Are we kind of upstream of those ones that closed?
I'm gonna push them and go downstream.
Okay, that's. We can move on. Yeah. Let's get some samples of these two.
Twice the size and 6 branches.
What?
Twice the size as the last one.
No. No, smaller.
Which quivers are open?
All except for 2, 3, 5, and oh, yeah, 18's open. 4, 7, 8, 7, yeah. 1.
You wanna go over there? Is that what you're talking
No, I thought when he was asking what was open.
about? Oh, right, okay. Yeah, that lid is open.
Are we doing 2 samples right now?
Yeah, one from each of these two colonies.
It's all open, 17. I let it air out there.
Okay.
17 has something in it.
Okay, next one. What's the next one?
18,
Yep.
18's open, 19, 20, those are all
So
open.
I'll open up 19.
Yeah.
Sorry, we kind of filled these randomly.
Stick something.
So, it's right there. Here, right?
I'm stealing one. Oh, these are good gummy bears.
That's all right. I was looking at it, just log it. That's okay.
Yeah, it's 20 open.
20 was open, right?
Yeah, 20 is
Okay.
20 is
Yeah,
clear.
the,
Nothing's in 20.
The cover on that one says 20, so we'll have the far one, which is 18, and 20. Sorry.
One of those quivers came up empty with no water a few dives ago.
Yeah, we replaced it last time.
Okay.
Okay, so
I guess, yeah. If we can get a long, straight piece that makes so it makes it easier for you to put it in the quiver. Yeah, like there or there. Actually, this one looks like it's something's going on. So, do you think can you reach this right here?
Yeah.
The scroll's so floppy.
How's it going, Chief?
Good.
It's still going slow.
These guys didn't make a signal, but
What?
these guys didn't make a signal, but we got 30 right here.
Okay.
Gives us our replication for a species.
What is this guy?
I'm trying to figure it out. I don't know.
Yeah, go ahead, do that. There's no rush, you've already clicked it. So
yeah.
So,
I'm just trying to, like, how do we get hot? Like, I was like, you know, so conflict, it's fine varieties, right?
Oh,
Which would be semi-popular, maybe not really, but can we put a sauce?
this is strange sauces right now. Yes, that is.
Yeah, we put it in the dye bag. Use that same apparatus.
It's great.
Oh, that's like a large one, but 4 hours of magic, 15 ounce fountains.
So now it's paused for
At some
the
point,
same.
I'll have to review.
Punch through.
Oh, it's just
I figured it's
that's great.
Oh, that's where you're going. I didn't realize you were going now. Okay.
which one is that? Is that 20?
This is 20 on the left, yes.
So now what I
What
want
do
to
we
do
just,
is wait
I'm
for
just
the
getting to
sample.
the point where it's like, is there something we're missing that we're not?
Start again until it goes past 10,000.
Just a lack of signal.
Click stop.
Relative
How to
to
set
what
up.
It's really all about the cutting action. Once the little, the minute it's the coral lot.
There was that
and
whole
starts snipping,
pandemic.
that's kind of when the coral comes out and releases a lot of mucus. Plus
Is that
it's
what-
the coral around.
So whenever like, there's like, I don't know, like, she clicks stop.
Yeah.
No, it is.
Coral the lab constantly shedding DNA or DNA. Puts it to the water.
So I should,
Oh, sorry.
I should start it as soon as
Yeah, so
Lena.
yeah, this is a chlorus back.
and once I click
I don't know,
start,
I'm just
stop
like-
before start.
I want to prove to myself. that-
Yeah, yeah,
okay,
you say you
okay.
turn it, yeah.
It's working. And if there's super runs down there, we're seeing it, and if all these negative signals are real. There really is no surprise. Because that might, if that's true, like that's also an interesting finding,
So
right?
this
It's contrast
two
from
is this a pump
what
itself?
we've seen shallow water, what we saw in
That's
California.
a slurp. So
Lots of
that's,
slurp, yeah.
yeah, it's like
I
a
don't know,
long
just like
tube that's
thinking about it.
connected, this circle carousel. It's like, acrylic, and it's one big circle, but then it has like little cylinders inside it. It's actually it's in this lower left screen. That shows us what's inside the slurp. Yeah, so earlier we slurped some.
That's some
Is there
circle.
also something like,
I should
was there
start.
fitting once not, like, is there, are we actually
Yeah, we were
drawing water
and then I'll feel
into the
really good that it's not
the water?
adding extra DNA into the water column
You did a wand
that
kind of.
isn't usually there to clean off there.
So I'm assuming they were like on the inside, they were like open.
Yeah, I was just finishing, yeah, I was finishing dinners, and I was like,
So you
wow.
see, so on the Solaris end, you see things change from pump. But if, for example, it was leaking up into, up from the one to like right by the pump head. So you're just drawing in a flow right there. Yeah.
Yeah.
But just, that's what I wanted. Right, exactly. So I'm like, it's just scrubby stuff.
Yeah, and,
So what kind
yeah,
of?
it's called scrubby
Which
stuff.
one you want?
Yeah, so you're gonna fill it out the same, but down the same.
Branch wise,
About some sort of positive.
anything that's long and straight. You think, is that grabbable, you think, in here? Because then that's just these two pieces. Right.
You could, yeah, you could mount it somewhere on the edge of a milk crate. Whatever, and then it's paraffin.
I guess that, or there. Whichever's easiest.
So why not before? Why is not before the sequence?
So weird.
Why click you being accepted?
We'll wait until it stops, we'll just, like, we just pick it every time you put it down there.
Oh, I didn't realize. Is that the camera on the hand?
Yeah.
That's pretty cool.
How can they not be doing that?
That's pretty solid.
You got here, I think you're just overthinking.
Yeah, I tend to do that a lot.
There we go.
That's what I was with this too, because it's like, there's so much risk involved that you get extra scared about it.
It's like,
You're
this is
like,
so
no, there's
important.
gotta be more, you're not telling me.
Okay, so should I, wait, that's all. Should I stop? Yeah. No, I don't mind.
Is that quiver 18?
Yeah. The same, but stop.
Yeah, do that with stop, and then I think that's gonna be it for the same, for second floor.
Storage location. Set is off.
Yeah, and then, oh, you got it.
Oh, the four sides.
I forget that one.
So really what's only changing is in this case is the pump stack.
So now, I'm just gonna keep this pump at 72.
Did you start seeing that too?
No, it's just so close. I'm just gonna wait until we get a new, sort of an interesting site that's different.
And then we get the pump through.
And it'll be a 0.2 microfilter.
That's switching.
Yeah.
Oh,
And then USH.
We swap seats.
Yeah, when I started with it.
Sorry, you stopped.
Yeah, I mean, the whole thing.
That'll never.
It's in, right?
It's in there now. Sorry.
Okay.
Okay. I just wasn't
Okay,
sure.
cool, yeah. Let's do it. It's nice.
I should have been more calm. I just thinking, like, they gave me my t-shirt, I think because I'm leaving early. So,
Did we get a preview?
it was cute. It was so like, it was like dry fit.
Yeah.
It's like a brush. Little 60.
Yeah, I got
Mine
brush.
was.
Yeah, it looked big. But yeah, this is every
It's like a foul board.
on the back. Yeah.
What's going on here? This is- That's really nice. It's good.
But you're gonna hear haiku.
Oh, you guys got a little signal on fast food?
Yeah, a little.
Very little.
Very, like, this is the tiniest.
I'm convinced we need positive control down there somehow.
How do you envision making a positive?
I mean, so we have our SOTs on the analyzer, and we run that, and it looks good. So we keep a solution on inside the analyzer. We run it. We get a signal. Can we put SOTs
outside?
Yeah, like either squirt it out or put it in a little Falcon tube and like go dip in there.
We
Samples.
have large syringes. You also, oh, gotta think of the movie.
Yeah, that's the thing. Is they're hard to kind of.
Is there like a those bags you use, can you have a bag like on the side of that quiver and just while you're measuring the Solaris like that?
Right.
Just slap it.
Something like that, yeah.
Yeah, I guess the dive thing, that nice freely flows out, right, so you don't know if the socks would
Yeah.
flow out of that nice, you wouldn't know how well it's flowing out.
We were thinking of making a dilute solution and then just sticking the tip of the thing, like through parafilm, into it. Suck on that for a little while and just see. All right. You know, we put a few of them. We could like mount a few of them.
Oh, okay. So it's so often.
Yeah. I mean, just for a sanity check. Because like, if there really is no signal here, and I'm just talking.
Right.
If there really
Thank
is,
you guys.
then that's an interesting finding, and
So
it
now we wanna
totally
fly.
in contrast with
I want to
other
fly
places that we've
and try to look
seen.
for
So
some
maybe
specific
that's
species
cool and
that
interesting, but we want to prove it to ourselves. But that's actually
that Colleen wants to sample. It's kind of like this. These like finger-shaped hard corals. Can you see that? That's the kind of stuff we're after. Maybe also one of those black corals that gave us a positive signal earlier. It'd be cool to try one of those again.
Right. Yeah.
So it might have something to do with the water quality.
Totally. Yeah. Like the nutrient content in the water and the carbon content. Absolutely. Good. But we want to make sure that that's really, yeah. And that's
Are
totally.
you guys going to some part of the east?
Yeah.
Of the island?
I think so.
That would be interesting.
Are you on the whole cruise?
No.
You're getting off.
I'll be leaving at 24.
Yeah.
Leila and Juanita.
So me and Sophia.
Okay.
Yeah, so the four of us. And Juanita and that's it.
Yeah, I'm staying and two other students from the science department.
Okay, we're looking for a few particular species of coral.
Depth are we at? 50 up to 52 now. Are we kind of transiting? Are you, what's been your kind of course, Doug?
We're not going west anymore.
Right. Are you kind of come up the slope? Okay. That's getting you to shower.
Yeah.
Okay. So maybe once we hit 50, we can start laterally back to the east.
To the east, yeah, we just can't go west anymore. We're limited by those fishing gear.
Yeah. One of your gyms fell off.
I have the one. It's the year of the cruise for me, so I know Crocs are really even wearing boats. So I went all out and made them ridiculous.
You think those are ridiculous?
Oh, no. People go nuts.
Yeah.
With their Crocs.
Yeah, the only one I'm very connected to is my senior.
You gotta see these. Look at
Can you zoom in a little bit on this one? Is that another one of those Montastria colonies?
They don't have Crocs.
Or a
But they're
sponge?
Croc sandals. They're cute Crocs. They're like fashion Crocs.
These are Crocs.
But
Oh, that
these
looks
are
like
also
probably a
Crocs.
sponge.
They're fashion Crocs.
But it's got that white edge like a coral might have.
Your daughters might ask for them.
Yeah, right.
Just a quick look.
You never know.
We can get them fashion Crocs.
Yeah, you gotta love your
Oh,
Crocs.
here's a coral.
I also have fuzzy
That's right.
Crocs.
Yeah.
There's some down in our Mauricio. That one? Una Mauricio, la marca.
Oh, this is something new, right?
You say Agaversia Lamarque?
The one at the bottom.
The one down here.
Yeah.
What do you think about this one?
Oh, I see.
I have to say it closer, but it doesn't have like.
That looks kind of like.
I am not sure about that one. Yeah.
We don't know if this one.
It looks like
does.
Madrasis farensis. Come here, look at this. Do you see this?
What's
Looks
the
like-
name again?
It says it's Madrasis farensis.
I am used to madrasas having like a lot of
Branches.
ramifications. Yeah.
Like this.
Mm-hmm.
Apparently there's some that are kind of flat formed.
Yeah. To me, it looks a lot like Agaricia.
This one?
Which of the species, I am not sure. There's one that is actually smooth.
Oh, without ridges?
Yeah.
Okay, well let's continue. There's a lot of junk on this one
Yeah.
anyway. So let's continue to try to find the specific species that Colleen wanted to try. Oh, there's a nice big one.
Varieties?
Yeah, she's looking for parietes and maybe madraces that are bigger, branching. Yeah, these flat ones probably, even if they are making some ROS, the current just flowing like that probably just, I don't know, maybe it just takes it all off.
I mean, they've used disco and shallow water coral reefs by scuba diver in currents too, and they measure things.
Oh, what's that? Right ahead of us.
Is that what you're wanting to see?
Yeah, see this
Oh,
guy.
that, okay.
Oh, the sun's setting on us now, right? It's getting darker.
Yep. That's one to look at.
Yeah. Okay, I think we can continue.
Do you want me to?
Yeah, continue. That's enough seeing it. I think I know. Can identify it from that. Yeah, I think that was. I think that was this. That's Pseudoptera gorgia. That guy right there.
43 meters.
Yeah, we're pretty shallow at this point. I don't know if we're gonna go any shallower than this.
Okay. But kind of yeah. So let's try laddering west, east.
Yep.
Well, there's a different kind of coral. Looks like. See that? Well, let's continue.
Okay.
Let's try to find these others for Colleen. It's her birthday.
Yeah. Varieties kind of look like that little orange guy. Not quite. They're a little more mustardy color. But
Yeah, a little yellower, but same same shape,
Yeah.
same form.
I'm just gonna get back down to that 50 meters and go back west. Or east.
at some point between now and midnight.
Oh, can we look at that black coral right here on the left? I think that might be the one that gave us a positive signal earlier. And if it is, then I should probably do it. Check it again. Yeah, I think it's going to be a good one to do. there's also scleractinians here. Okay, I'm pretty sure this is the one we want to measure. Yeah.
Lena, this is the same black coral that we got a signal from earlier, so let's do this one.
Species or colony?
Yeah, let's put species, new colony.
So we're gonna want to Solaris this, guys, and then take a sample.
Item settled once. Just let us know when you got your background.
This is SH18683. So this is a Plumopathes. This is the same species that gave us a little signal, a clear signal earlier in the dive.
Okay, we got the background.
Alrighty.
You know what that number will be for
It's
the...
gonna be 11.
Yeah, sorry about the setup.
I guess you can go up on elbow quickly, but that's it.
Yeah, you could.
Just only watch for smashing and
There's a much healthier colony than the other one. The other one had like a sponge and junk on it.
This is much healthier, Colin? Yeah.
Than the previous one. The other one was filthy. Maybe that one.
They also have stills.
This one's not going to be just. No, this is like a sponge just growing up through it, and looked like sediment and junk had settled on the branches.
We need to change it for the corals, so we can go back and have good image. Yeah, and we'll have it on the format that I mentioned to here's one.
Yeah.
see it.
Oh, it looks like, is that a slight rise? It's kind of like what it did before.
Come up with an ID for that optical thing you're doing.
All right, thanks guys. Now we want a physical sample. SH number is 8.
I would have no idea.
You see that? Now we need more pulls at least to test Luke's idea about some of everything in fantasy that instead of just being switched up. This one's orange and a lot shallower than the previous one.
Which one would want to open here? Yeah.
19's open,
Yeah.
21 and higher.
One on the porch too, so. What do you want to do? One on the porch?
Yeah, because this is probably
Yeah. Okay. Well.
Gave us a signal like the previous colony.
It did?
A low but steady signal. Yeah.
Signal?
Yeah, so both times on this species, we've seen kind of this slow increase to
So it looks like
maybe 800, 900.
1, 4, 7, and 8 are open on that side. 1, 4, 7, and 8. Or you can do 9 or 10, which is closest to you here. I'm gonna rack the camera back. So which one do you go for?
Happy birthday, Colleen.
So that one right there if you want.
1,
That's 8.
4.
Do 8. Right there. You can do the next one over. One that is all the
What
way
is
to the
that
far side.
recovery right there?
Raise it.
Oh, is that pumps off?
I know I did
Oh.
6, right, I thought I did 6.
6 has something in it, right?
Yeah.
With a sponge.
Maybe yours not so bad.
Okay.
Wha-
Put that cover somewhere on this side so we can grab it. Like through that little gap where you're going.
This particular black coral here, twice now I've given a.
Yeah, we found the same. The black coral earlier on the dive gave us a signal. There's another one of those species and gave us a signal too. It's like the other one's like 100 meters. This is like 50 meters.
Really?
It's bright
Slow
orange
but
instead of the
consistent.
other color, but it still gave us the same kind of signal.
Huh. Interesting. So same species but different color. Weird, right?
But still signal.
Huh.
It's pretty. I like that.
That's cool.
You coming back to the helm?
Unless you want to stay there, I'm totally happy with you staying there. You,
Well, you need to catch some of the in situ images from these that I've been
I can do
saving.
in situ images.
Okay.
I got it. Yeah, nice try.
See the birthday seed.
Nice
oh,
try.
sorry, I just smacked you in the head. What's up?
This is actually all the way to my head.
I've been getting the images anyway, so. But,
You gotta entertain the audience.
they're a little bit like.
I took some like quick highlights, good pictures, so they're in there somewhere. You just gotta find them.
Okay.
I'm gonna try to do simple highlights also.
What you looking for on this one, Sam?
Yeah, I do all that. Yeah. I like
Sorry?
to look.
What you looking for on this one?
Do you think you can get here?
That one that's coming down this way.
Yeah. I'd like to find a variety.
How much do you have?
Yeah. I said we need a positive control.
Yeah.
Yes. like around,
There's supposed to be a few around here.
Okay.
Yeah.
At what depth?
yeah, something like here, even there. That's yeah, that's good.
It's recorded. Yeah. Yeah. You can even find it as low as like 70. Like, not many, but.
Okay.
This I can do for a haiku.
Yeah.
Ooh. Did the fingers get offset from the
Rock.
the
No,
rock?
from grabbing the rock. Dulls the cutters.
Oh yeah. You gotta sharpen those up.
Ooh, jeez, this one's gonna be hard to sample without that sharp. Ooh, it's slipping.
Yeah, see the slide through there.
Ah. Do you have a sense for how much that increases? What's that?
That's good. Is it the same branch?
What?
He said it looks like only the sunlight in here, but it's just.
Is it monitor light?
Yeah.
It's the skeleton's kind of like fishing line.
Okay.
I think if you rotate a little bit more towards us like this and pull, it might make it snap. So it's like tugging against the blade, even though it's dull.
There
There.
you go. Nice one.
I break these with forceps, so I'm trying to just mimic what I do in the lab.
What's your lab at forceps?
Good to know. All right, ready for the porch to come out?
That's really what that is, just giant forceps.
Yeah, when they're not sharp, they act like forceps, I guess. Where's Jen? Oh man, she knows when I try to use her plastic ones.
Oh yeah.
Snap her a piece
Yeah,
off.
that's pretty.
Hopefully this one won't be too much of a pain getting in the quiver.
Look
Look how bright orange that is. Look at that.
You keep messing up?
He's like, take 50. He's like, you said corals don't have a lot.
Oh jeez. Really
I feel like I was like, he deals with like, okay, go and come back.
We're really interested in how the chemicals here make a living. Corals. Red corals. There's just so many chemicals here. I mean sponges.
Did it go in?
Yep.
Cool.
Yeah, you can see it in that camera. Oh well, it fell all over the bottom actually. Could see it for a
Didn't he
second.
interview him first day?
Yeah. He's getting
He's so happy and just you can see. First day, first day.
Last day. Last day. Yeah. They got all hairs, all these.
Loretta keeps sending me pictures of little chickens in the parking lot.
She's so worried.
She,
I think she left today.
Oh, she left
I
today.
have more.
Paguera. La Paguera.
That's kind of what we are, right?
Yeah.
Finally.
This is Ghostbuster.
It's your chicken?
Okay, let's go and try to find another one of the, or one of the ones that Colleen wants. The kind of lumpy, the thumb coral. It's kind of like shaped like thumbs. It's gonna be a hard coral too.
Yep.
It's my favorite stage, little awkward teenager.
I think we're gonna get 3 more.
What?
Thank you.
I didn't want to start raising them without you.
Alright. I'll just spend there all day.
You missed
Oh.
like all the time.
Yeah, that is all the fun. You need like a live show.
I bought her, I got her once for her birthday cameras that she could launch from anywhere and see her chickens, and she was like, return those. I will spend all day long just looking at
Wait, you said that?
Yeah. I'm like, this is a great gift. She's like, that's dangerous. I can't. One of them was a pan and tilt. Even. It
Oh
was
my
like-
gosh. It was like spy on the chickens.
So she could find the chickens around the yard.
You seen that movie Chicken Run with like
Yeah.
where the chickens like are smart and they
Yeah.
It was too good of a gift.
It's like Wallace and Gromit.
yeah, yeah, yeah.
They were pandemic. Our daughters were
Hey, with
like,
egg prices these days, it's a good idea.
when our daughters were like, "Look at the chickens." We're like, yeah.
Oh, there's line there.
Yeah, there's a fishing line right there.
I'm trying to get with my parents. I want to get chickens. I want to, because we have a trampoline that we don't use anymore.
Turn it into a coop.
You want Parietes, Asteroides, and some
Parietes
foamy
would be wonderful.
Madraces.
Yes, and yeah, the braces would be great too.
So do we expect to see Parietes, Asteroides around here?
Yeah.
This depth?
Mhm.
This depth too?
Yeah.
About 50 meters?
Yeah.
Okay.
It's going to be kind of yellowish, right?
Yeah.
Is it still that color at deep?
What?
When it's deep, is it still that color?
Yeah, they still have the mustard color back. Yeah.
Okay.
These are my favorite chickens.
Whoa.
Is
Is that a cable or what? Yeah, it's fishing line. Or cable or something. Yeah, control line. Yeah.
Yeah, that's the. What kind is that
They
one?
could stick the buoys in it, I don't know.
That's old. That's been
You
down
have
there a
to
while.
get
There's
ones
stuff
that are
growing
really
on
hard,
there.
since you're up in
Cold.
Boston, you know, it's cold, really cold. So there's like a bunch of species we can't grow because they don't do very well.
Huh?
There's the ones that are
Great.
Oh my god.
Can we get a
That's
look at this
It is
a
big.
little closer?
Look at that.
Yeah, just to check if it's a sponge or coral.
Yeah.
Holly, do you guys live on the Cape
A
or?
little squid.
Oh, yeah, the squid. Yeah, down there near Woods Hole.
In Falmouth?
Yep.
Yep.
Yes.
Oh, it's probably a sponge.
I love Falmouth. That's a nice town.
You know, found it?
Yeah, my grandparents lived in Nashville. They just
Oh.
moved like.
Yeah, it's got to be a
I
sponge
wanna
with
say
that
last
much
year,
junk
they
on
moved
it.
over to be a little closer to us.
Okay. Nashville, that's
And the
nice.
house was really, it was
Actually,
too
very
big.
close.
Yeah.
Wait, is that Montastria? Like a ball? Or just, nah, I don't know what it is. It's hard to see. Let's continue on.
Oh, there's one with done. Shingles on the side.
They even have a wave.
Yeah, that's the.
Oh, so beautiful.
So we designed a little wave, but then we realized because it's the window, the chickens go over there, and, and it looks like a chicken head. It's exactly a chicken head. It's supposed to be a wave, but actually it's a chicken head.
Slow keep going.
It could have a double meaning.
Yeah. Oh, here's Rossi.
This is the kind of coral we're looking for. It's kind of yellowy, mustardy, lumpy.
Oh, okay.
Oh, it's a water.
Yes.
That and that thumb-shaped one I was showing you earlier. Yeah, we're looking for Parietes, that mustardy one.
Oh, no, it's a sponge.
What are you pointing at? That? Oh, that's a sponge. That's a sponge.
Too smooth.
So, most Parietes species you've looked at just make a lot of ROS?
Every single species.
Mm.
And it doesn't matter where, like
Oh,
Cuba,
hi.
St. John,
There's a
Hawaii.
squid.
Yeah.
Yeah, cool sponge.
Yeah. What happened? Oh, a squid.
Yeah.
Oh, there's a big barrel sponge.
Oh yeah. We could always stop and do some sponge work. At some
What
point we
is,
should.
what is that more?
Is that a sponge you'd want to do?
Mm.
Scott, is that a sponge you'd want to do some sponge work on?
Nah, it's.
I've seen better.
I've seen better. Oh, now he's getting picking up.
I do have a
Are you a sponge snob? Are you a sponge sewer?
I'm a sponge snob.
Just sewer.
Oh, I like sponge sewer. A sponge sewer.
This looks a little more sedimented over in this area, right?
Or 50 meters.
I think it's, yeah, kind of.
Because we're heading more west, I think.
No, we're going east.
We're going east.
This is going east again?
Yeah, west is where the fishing buoys
You
are.
can't go-
Oh, right, right,
Can't
right,
go
that's
west
why.
anymore.
West is,
We went as far west as we could go.
okay.
Well, unless we back all the way up and go around it
Right.
with the hopes we can cross it.
Did you get the C2 images of Greek popping towards
Where's this guy?
I think that's one of the Agaricias down here. Do you want to look at it, Colleen?
Yeah, can we look at that real quick?
Yeah, can we look at this guy? Oh, it's also a big one right here. I think we were just looking at this one. Want to look at that?
Yeah.
There's that big patch.
The what?
This big patch right here.
Window rib. Yeah, there's a huge one
Oh,
here.
that's an Agaricea. But I was wondering what that smaller, if
Is
that's.
this Gracilaria?
Oh, that is. That's a Gracilaria, Lena. Look at that. This one? No, no, no.
The
that-
algae.
oh, the
Yeah, she wants to see
algae,
this.
sorry.
This guy.
Let's find the name.
Well, there's a huge sea cucumber right
Plant
here.
that for Loretta. That's what she was growing on the farm.
I know.
Oh,
It's collected for her lunch.
we would bring it home carefully and bring it to her.
She said, oh, nice. Yeah, it's some kind of coral.
And yeah, I don't know what that is.
Hmm.
Well, it's cool.
Wow.
You guys know what that is?
Is that a Montastria?
No,
No, it's not Montastria.
No.
Annularis?
Could be Orbicella?
Oh. No.
Hmm.
No.
Sidastria?
It might be.
Might
That's
be
my best
Sidastria.
guess.
Yeah.
What do you want to do? You want to put the Solaris on it?
Yeah, we might try Solarising it. If that's okay. There's like a little tiny fishy right there.
Close enough, yeah, hopefully. Hopefully I do.
I know it's little. Actually, was this like a mound and it's being encrusted?
Oh, maybe like dying back from that. That might-
Yeah.
Trawling back the rest of it so you can scale it and stuff. But I know it's nice.
Do you see UTC somewhere?
No, it's easy. Thank you.
Okay, Rich, we want you to
Yeah.
continue following the Great White East.
You can, yeah, just follow it a little bit more to the east, please.
Okay, Lord.
Thank you.
There's a bunch of little stuff swimming around there.
Which Solaris event number will this be?
It's gonna be event 12.
And then when you're ready.
Doesn't quite look like Citrostrea. Citrostrea's got some pretty deep corallites. This looks like really shallow.
Yeah, it does look really shallow, huh?
Orbicella's got some chunky polyps sticking up pretty far, prominent.
Orbicella has the star, right? Isn't it the star?
Yeah.
I think maybe Cerastes.
But the polyps are so small.
They're really small, yeah.
Oh, look at those white patches.
Yeah.
You know? Think that's disease?
That must be predation, maybe?
Oh.
Like bite marks?
Yeah, maybe.
Yeah, maybe. Oh, sorry. It's like,
Nothing. Squirrels are fake. Just graffiti. Some graffiti to coral on the side.
Yeah.
Oh, there we go.
No, I think it's.
To the bump. The bump. Should we consider bump signals just
well, I mean, on the one hand, it tells us that they can make it, so that's good. But we were just trying to understand like what they're normally making.
Seems to stand.
Yeah, it is standing up there, so that's interesting. It has been shown that, like, I think it's, what's the one in the Red Sea that's really common?
What,
Stellophora. Like they've done some. Stellophora. That it responds to physical stimuli. So if you like put even like
Get the
one
wand
polyp.
away so it goes down.
Yeah.
I'm gonna move the wand away. See if the signal goes down.
That's like suspiciously level.
Yeah.
Right, this is steady. Oh. Well. Slow down.
There's a lag, but
Minute. It's tailing off.
have you tried doing an SOD injection anytime recently?
No, just call it again, but do you want me to?
Yeah, it'd just be good to see like how far down it goes.
That's an Agaricia, for sure.
Yeah.
Oh, fish.
Squirrelfish.
It'd be nice to know, how much superoxide we're just having in the waters themselves. Like kind of running some just sort of background water through and then doing SOD. A couple times just to try to constrain that. Maybe there's the waters have a lot too.
The bright orange splotch of sponges, right?
Yes, we think so. Sometimes you also get some tunicates that are brightly colored and kind of amorphous, unless you look really close at them. Look close enough and it should look like something, right?
Yeah, no, this.
What do you think, Nicole? Sponge or
Oh, this one's
tunicate?
really bright. Well, it would have to be a colonial tunicate.
Okay.
I'm sorry?
It looks like our baseline signal is just
Okay.
Do you think it wasn't a real signal?
trying to stand with the lines.
But we were up at 10,000, so it looked like there was a little bit of a.
It's possible that we have hard customers that just pop.
Yeah.
Now I'm just pushing it out all the time. I don't know, it's like.
Yeah.
Oh, that's a sponge with maybe some zoanthids on it. That's like that purple sponge, Nicole, I think we got from the Gulf of Mexico. Yeah, it's got zoanthids on it. But different kind. Remember the one we saw that's like white with the red zoanthids? It's like purple sponge now with dirt colored zoanthids.
What, they look green.
Yeah, they look green. Maybe they might even have symbionts in them, those zoanthids. That'd be like some tripartite symbiosis. That'd be cool.
Someone in the chat there is saying Orbicella fabulata.
Orbicella. Let's take a look.
It's actually a type of coral that looks like a heart.
It looks like what? A
That
heart?
looks like a heart.
Orbicella fabulata. Oops.
That looks pretty good, doesn't it?
When
It
it
counts.
Yeah. It took a while to figure it out. Go back and see if it's.
See what the background is again.
Okay, you can do this.
I think Orbicella's polyps are bigger than this. You think so, you know?
Yeah, and I actually was wondering if this could be like Abuelita's plate like, but
Yeah, I'm
it's
trying to-
too small the polyps.
Even for parietes, you think?
Yeah.
Yeah, that purple thing also looks pretty. Oh, that orange one looks pretty.
Should we try to collect a piece of it if it's possible? I don't know, it looks pretty.
Yeah, I guess we could try. We kind of
We'll
roughed
have
up
to
the
see it
blades of
closer.
the claw earlier, but I guess they're already dull now.
I think it's pretty slick. Try to break it more off of it, sort of.
Yeah, I guess whatever we can get.
Let's see where.
It's down there now.
Oh, yeah.
Down with the Solaris.
Yeah, I think we're done with the Solaris.
You can put Solaris away, thank you. That looks good. So it seems to come back. Are you taking notes, Sam?
Yes, I'm taking
Thank you.
notes.
Are we trying to collect this one?
I think so, yeah, then maybe we can try to identify. Oh, great, yeah. Oh, that would be wonderful. So someone was asking earlier whether or not there's been observations of the disease in the mesophotic area. And the skittle.
There has been in the north side of the island.
Okay,
Yeah.
and I thought I saw a paper that
If you want, you can go to number
Yeah.
4.
spot too, right?
I think it's called a circle.
Yes.
That's the one.
Yeah.
Yeah, one state.
So do you know that it's getting worse, or has it been looked at at all?
I should have found it.
Yeah.
Yeah.
Oh, just saying. There to
You're asking around.
from my diagnosis to make travel up in the
Oh, okay, okay.
And then 17, 18.
Yep.
20, I think it's in 19.
Oh, okay. Not what they want, but these are. Oh, yeah, okay.
Good over all the way up and close that one bioanalysis.
Yeah.
polyps in this are so tiny. The collected one like this in the Gulf of Mexico, somewhat similar like that, a little deeper.
What did you collect?
Something like this in the Gulf of Mexico. We have no idea what it is, but the polyps are like so.
So tiny. Yeah.
It looks almost like a sponge.
Oh. Oh. Thank you.
Be careful about getting
Hey, just do it. What?
No money? Don't give cards, right?
It's not fun.
Still a grad student.
Okay, report back in.
So I just started calling. So Maria was saying that after each dive, they have like 10%.
Oh, you're going out.
Oh, really?
Which I didn't expect. I thought that they were.
What are you looking for?
Yeah,
Oh,
they're
sorry.
filling them up. Well, all of them in the, but even in the classes.
Oh, okay. Okay.
So you can not so great. So if you're coming for us, that's fine.
Yeah, yeah. So do you want to use, if we don't get like Solaris data and Squeezer data on it?
You might have to spray this.
Okay.
I don't know.
Yeah, okay, okay. Yeah,
What
we-
makes the project do more?
We are on this.
Yeah, yeah. So, because we saw some other oranges earlier, and we were like, nah, we can't actually like Solaris them because they didn't really, they were really tiny,
Yeah.
but.
Okay, so if we just see something cool.
Yeah.
Okay, I was thinking,
Because I thought that we were using all the space for corals.
Lower spot and straight up.
Oh, really? I didn't realize that. Okay.
I knew it.
Okay, all right.
Great.
Yeah, yeah, yeah.
Good to know.
And I guess if you collect them and then you don't.
I was thinking like, you said
Well, I mean, I'll play them for sure.
Okay.
And I also think that, so I still haven't seen any which is a good thing because it means that the bacteria isn't taking over. I had an antibiotic.
Yeah, I just assumed once I started trying to grab this one, it would appear.
Making the big growing sooner.
Actually, you wanted to coach me though. Was it that one right there?
Yeah.
That's a tough one.
As
Yeah,
colonies.
it's gonna have to be sprayed right.
Yeah, but,
Yeah.
but
Okay.
you have to get your jaws in as
How's
much as
Solaris
possible.
doing?
Ooh, what's that?
And then you can
Here.
try to lift it up.
Oh,
the
like a
edges, just
worm
right the edge,
wave
yeah.
on the top.
You have to
Yeah,
get
yeah.
Yeah,
Is that
that
one of
little
those?
worm. Yeah.
So
And
Oh,
it's like
you
a
just
nice-
opened
up.
Kind of the
Yeah.
front, come right to left or something, because there's that orange sponge.
So we're trying to figure out what this coral is here.
Yeah, just Google it.
I can't figure it
How
out.
do you tell that it's a coral and not a zoanthid?
Zoanthids don't like calcium carbonate skeleton. So
Yeah.
they're just
But how do you
tissue.
see it as different?
Oh, there's a white edge, this the calcium carbonate edge. It's like the leading growing edge, where the algal symbionts aren't as high density in the older tissue.
First thing I can see that you can
It has these little tiny polyps that just
It's right there.
So cool.
it's too.
It looked like it had.
I mean, in the low light.
Flamed it off like
See all
that.
this
Yeah.
stuff.
As you say.
Yeah.
Yeah, but it's also really shallow.
And then there's the zoanthids. There's zoanthids single polyps growing on large part. Well, and there's the like small polyps growing in the orange above.
Oh, yeah, yeah.
Yeah, really tiny ones. There's a purple star here that has white, larger than
It looks like it was a boulder that
This one isn't like.
half
Died.
dead, yeah.
Come back in.
You look up with a.
Yeah.
Well, it is a crystal one.
How far up do you want it?
Right there in that zoom in.
Or
Oh. You wanna give a explanation of?
So like maple.
Walked in right as it was being celebrated.
Yeah, thank you for all your help.
We're there.
are they asking? People asking?
Okay.
Yellow and purple fish.
Cheerio.
Oh, the difference between zoanthids and corals. Zoanthids are kind of their own, I think, order, Zoantheria. They're People call them colonial anemones because they're kind of like anemones where they don't make a large skeleton and they kind of encrust things, but they don't usually make. They don't ever make a calcium carbonate skeleton like a stony coral would. So we can see on the edge of these coral colonies that they have a little white edge where you can tell that they have a calcium carbonate skeleton.
Is that an Orbicella? A really small one? On the bottom right, over here? Over here.
Oh.
Yeah, a tiny one. Orbicella fabulata, I think.
Yeah.
Exactly, open up.
We lost it, yeah.
Oh, maybe. It looks like it has a little stars.
Oh, yeah.
Get that sick orange sponge.
Yeah.
Okay, I have some questions.
Squishy.
You okay if I squish it?
Yeah.
The sponge?
Sponges are
It'll be
doing
fine.
fine.
Be nice though.
There's another one over here that.
The whole thing comes up like a rock. It's probably too big for the bio box, right?
Ramp.
But
No,
you want
the ramp
the
itself.
It would be funny.
I was just thinking if the whole rock moves, but we could take it. But I think it's too big for the bio box.
Yeah, if we can't get it, it's all right.
Yeah.
I know this is kind of a
See
weird,
if I can lift this green portion off of it, see what can be.
if there's like a little lip.
Stay away from the gummy bears.
You be snacking?
No, just one, the green one. I had to try it. See,
They're really
test
good,
them
aren't
out.
they?
They're pretty good, yeah.
That's free.
Oh, yeah.
Yeah, I find usually it's 2 hours, depending on the situation.
Maybe you get to know from one.
Yeah.
Okay, so in other words, we've got to get this happening tomorrow.
If it's possible.
Maybe, I did that dinner meeting.
Was the mighty convention.
Yep.
I love that. Yeah.
Summer blockbuster.
I always miss the end of room tours on ships.
I know, I always forget to do that.
Oh, no.
All right, I think it's okay to let that
That's right.
let that coral be.
Yeah, unless you're gonna grab the whole rock, then.
Yeah, let's move the whole rock.
No, I don't think it was gonna fit in the bio box. I think we're okay with passing on that coral.
Yeah, it's okay. Thank you, though.
Yeah, thanks.
We'll try to find it again. And we got a picture of it, right? So we'll
Yeah.
be able to
Yeah, we got a
send that
picture.
and try to get an ID.
Habits.
So we are on the hunt for varieties.
Sure. This is what it looks like, J-Rod. Can you see from there? This yellow,
The
lumpy.
mustard coral.
Okay.
So we get your pen?
Which one do you want?
I'll do this one down.
That gracilaria is so clean.
Yeah, Gracilaria.
Just a little bit.
I love that seaweed.
50?
sargassum.
I stayed about,
Could that affect the baseline levels of superoxide in the water?
What it was for
The
us.
sargassum?
Yeah.
How about-
I don't think so, but
So what do you get about the?
waters are way more productive than I was expecting. Yeah.
What, this big coral? Are you trying to say something about it?
I think we were interested.
Yeah, just wait. Just message him. Alright, where we going?
We are keeping the same depth, heading east, looking for
You
that.
wanna go any shallow?
Well, yeah, I think they were being cautious.
What are we at right now? 50?
You're right.
No, I wanna go see what that is. So
If you wanna go shallow.
Yeah, don't wanna go shallower?
Oh,
Just have
we-
If you feel comfortable with shallower.
shallower. Come back up onto the deck.
Yeah.
Look for corals up on the deck.
Oh, what is that little swimmy
Oh,
thing
squid,
there?
cuttlefish or something.
Oh, cute.
That's the squid that Mac has a tattoo.
Oh, really?
Right there.
Oh, nice.
Oh, there's two of them.
Kirby is obsessed with them.
Oh, so cute.
Weird. Screen dad. I've never seen them do this though.
Me neither.
Oh, there's some of these. There's some sticopathes,
So cute.
some black corals.
Oh,
What I said the other day is not always true. That the curlicues, whip ones, are black corals, and the irregular ones are octocorals. That's not always true. Sometimes in shallow water, these irregular ones are still black corals.
I think they're not friends.
You don't have to be.
Oh, maybe they are friends. Maybe we can leave it that way. I don't know what this guy's got going on though. Like, is it a mustache? Like, never seen him do that. Have you guys seen him do that?
In shallower areas, there's like Ben doing that. Yeah.
Really? I have to
Together.
Oh.
I think he's trying to scare us.
Oh.
It might be. Why? Because it's actually like.
He's like trying to aggressive coloration.
Zoom out, Chris? Chris? Zoom out? Yeah. That's 4K? Right there in front of you. It's so neutral. Oh, yeah.
Oh, there's a third one.
Oh, those corals we're coming up on are sponges.
See, they're just gonna slowly grow in numbers. We're gonna zoom
They
out.
better-
There's gonna be like 30 of them.
They better take us right to some good corals. It often happens. We'll like zoom in on some animal and like follow for a while, and then like take us to something cool. Well, there's some Agaricea.
Hello, little fuzzy. There's 3 of them, I think.
Look at them. 3 of them. It's a family.
Oh, they're really showing off.
They're totally
See all those
show
hard
us.
coral? Is that what you were just talking about?
Yeah, but we're not interested in them right now. We're going after those, that yellow one. Because Colleen's pretty sure it's going to give us a pretty strong signal, and we're trying to like get a positive control for some of these because we're suspicious why so many corals we're testing aren't giving us a signal.
That is so cool.
Maybe they're just really
He's just
vain.
hanging out there. Take my picture.
That's what I think they say. They're either saying, hello, welcome, get out, or take my picture.
They're curious, like, why are you
wow,
here? What
this is
are
so
you?
loud.
What is light?
These people have been this close to the sun before. What's their
Yeah.
name? There's not corals, yeah.
Yeah.
And look at their little eye, I wouldn't say little, they're kind of big eyes.
Yeah.
Their body size, those are pretty big. Oh, it's so cute. I don't think I'd ever like be saying that, but like it's really cute looking. It's a little mouth and it's big eyes.
Yeah, you could all do them.
Sophia, look up banded sea piglet. That's a cuter squid. Banded sea piglet.
Just being called a piglet.
Yeah, that's
Piglet or
how you
pig?
know.
One of the two. Banded sea pig or piglet? I don't remember.
Oh my god,
Is
it's
it
so
luminescent?
cute. It
squeezy toys. It looks like those.
Yeah.
Oh. Do you see that, like,
They're just, I think they're just looking out for us. Sitting an hour late.
They're so much better at keeping still than I am.
Yeah, they're really good at it. Oh yeah, we were looking up the piglet.
Banded sea piglet. Sophia, was it piglet or pig?
You want any more helps? You want any more?
Oh yeah, this guy classic.
can someone get a frame?
Wow, there's a big barrel sponge on the left there.
Oh yeah.
Look at how active this area is. Look at all the fish.
So amazing. This is the
Sun's setting. Maybe some things are coming out.
Oh yeah, we're in that transition period.
Evening in the mesophotic.
Yeah.
I love how they're just staying right there. Where? What? Have you seen these guys before? Oh, I thought you said you saw someone scuba diving. I was like, what? That would not be good.
Look at Santiago. It's Santiago.
Yes, it's Santiago. He's got like a bag of corals.
Bag of coral. How shallow are we now? Oh, 60.
Yeah, they took me up and then back down again.
Oh, they're so great. Look at them.
And they are different. Like, look at the different coloring.
They all have different feelings, Kurt.
They're all like lined up in a row.
Rings. Yeah, there'll be like 30 of them, and they'll all just like line
Like
up.
a squadron.
Seriously,
The perfect
curious.
squadron.
I wonder if we should tell Scott about that sponge. That's a pretty good looking sponge.
Yeah, it's pretty nice.
Where is? Oh, there. Yeah. Where is he?
Oh, you have like a really
Oh.
good TV in front of you.
It's his own fault he's not here. Look at these guys. They're so funny.
And we're stopping the dive.
Ooh, that's a cool
What do y'all look at?
Agris, yeah. Well, let's keep looking for
Yeah, parietes.
the
fishing
parietes. Yellow, lumpy parietes. Parietes asteroides.
Alright, coming up.
What's on the left? Oh, no, that's algae. Wow, there's another big sponge.
You see a sponge?
You know, it looked bigger than that, didn't it?
Don't tell Scott there's a sponge.
I already did.
There he is.
I heard that.
Look at the ex-muta there.
Oh, that little guy down there on the left. Yeah, yeah.
Is that little?
It's a little small. It's smaller than some of the ones we've been
Okay, it looked
after.
really big.
There's another one up there.
Oh, there's another one.
Looks a little
Oh.
bigger.
Are we getting to
I was,
Spongelandia
there's two of them
again?
next to each other.
Look how short that one is. It looks like a mug.
I was outside to see. That was the
Mug
one
and a
I was
pint
watching you
glass.
guys. Did you see me commenting?
No? On where?
No.
I
Oh,
was
here?
commenting, I was wondering if you'd recognize me.
On our what? Wait, on the.
On the live chat.
What were you saying?
I just said, cool squid.
Oh, I just see that.
Safe bet.
I thought you were playing tricks on us.
There's a black hole coming
What?
up.
I thought you were gonna be playing tricks on
Oh
us.
yeah, look. Oh.
Ah.
That's like one of those shallower ones, right?
No, I think that's an octocoral actually.
That's good. We can look at this squid, this
Oh, there's some
sponge somewhere.
Gracilaria. Oh, what is that?
There's another sponge.
What was that? Off to the left?
Oh, is that a sponge?
That
yeah.
yeah.
Darn sponge.
Okay, sponges are cool. That's a giant sea cucumber down there on the left. Right in the corner. Right in the corner. That, yeah. Do they make our way?
Attacking us.
I still got
Oh
my
my
buddy.
gosh.
How many
So
can?
6 to 8.
It's a nice sponge there.
You know.
And a nice parrot.
squid.
I didn't know if
Is
it's the
this
squid.
the?
Well, if you want a sample, let us know.
Shallower?
Sure. I mean, anytime. Looks like there's plenty of sponges in here.
Yeah.
Are we looking at this one or keeping going?
Do you care about this? This octocoral?
Another one up here? Top right? Do
Another one?
you want to have a zoom in on it?
Can we do that? Yeah.
Yeah. No, we've run out of zoom.
Ran out of zoom.
Oh, yeah. That's
Oh
another-
wow. Yeah.
Montastria. Yeah.
There's a hermit crab.
It's a little gumdrop.
I don't know, but were we able to sample
It's,
it?
That's so tiny.
it's another really hard position to get it though. Keep looking.
Alright.
Oh, there's so many holes, like, this is
Did you want to get a photo?
A photo of it?
Oh, I thought you were pulling your phone out.
No, no, no. I'm trying to look up those octocorals.
Oh, did you want to sample? Look
Another
at this
barrel
big
sponge
one.
there.
Wow, another one. Yeah.
Yeah, the sponges are like
Yeah.
That's a big one. Well, maybe. It's hard for me to tell. What is this on the left here? Is that a madrasis?
I think it's probably a sponge, but we should take a look at it. That stuff.
We're at 39 meters.
Are we?
Are you scared?
Are
You
you?
scared? Oh, okay, J-Rod.
Oh, is that
I want
sponge?
to see 19 meters.
Is that trickster sponge?
Look at that sponge. That's a nice little sponge. Meat sponge.
Why aren't we seeing any?
Lots of barrel sponges here.
A lot of barrel sponges. Yeah.
Weird.
So I mean, we've only got 3 squeezers left. So at some point, I'd like to use those on the
Oh,
sponges.
do you want to do this one?
Sure.
We're here.
Sure.
Ooh, yeah, this.
Go look at the
Setup
gracilaria.
and
The
there's
sponge.
nice- What about that coral right there next to it?
Like, I feel like I've seen something like that
We
before.
can try it. We've done
This
we've
one?
done 3 of those already.
This one?
Yeah. Oh.
Oh, that sponge?
Is that what
Big
you're
old
talking about?
lionfish up there.
Wait, you want to do this sponge?
I think that's sponge.
That's
yeah.
the sponge, and then there's a giant the coral right there next to it.
This.
Is that anything of interest?
Maybe. We've done 3, so I guess hitting another would hit that 4 to 5
Okay.
colony target we're
So
after.
if we park
But it
here,
didn't,
we
it
could
didn't
just-
give us anything.
Oh.
Any signal.
Do you want me to park?
No, we could give it a try.
Sponge time?
All right.
Sponging?
Sponge time. Do you want to sit in the driver's seat while you do the sponging?
While we're sponging. Sponge bath.
Sponge.
Time for the sponge bath.
Sponge it up. I need to catch up on some of those slides over there.
Ugh.
What all are we doing to the sponge?
Oh, what
Just,
don't we do?
Just the squeeze or are we doing the
Oh,
dye?
that's the whole.
First dye. First the dye. Make sure it's pumping.
Okay.
And or, you know, hopefully it's pumping.
Should we call Kate?
Yeah. She's outside on the. She went out to make a call.
Are we also doing that coral or just the sponge?
Yeah. I think we're gonna also do the coral, but
Solaris or?
Want a
both.
Solaris first?
Yeah, before the dive.
Okay.
Okay,
Let's do
sure.
Solaris.
Do
and
Solaris
both
and the
of them.
sponge on both of them, but start with the sponge, I guess.
Okay, I'll set the back up measurement.
So I'm gonna back up just, I'm gonna come down a little bit so you have room to work. We'll be working up. Are you alright with that?
Yup.
See, looking HD1. See what I'm talking about there?
Yeah.
Look at the gracilaria. Should we do the gracilaria too since we're right here?
Yeah.
How
You
cute
mean
it is.
with the Solaris?
Yeah. This right here.
I love it. Is this it? That'll make Loretta happy. That's pretty cute.
We went to Ponce to collect gracilaria for her.
Oh, really? You did?
Yeah. It was fun.
It's so pretty.
Are we doing sponge or gracilaria first?
Sponge. Well, yeah.
Yeah. Sponge.
You can get Solaris up there.
We're gonna Solaris first, just
Oh,
because we
awesome.
wanna get the dye in there.
I think that's smart.
Do a background first?
Yeah, I got the background, so ready to go.
Okay.
What kind of sponge is this?
It's a Zesto Spongium Muta.
Does anybody have
X.
a phone charger over here?
What kind of phone you got?
iPhone?
Okay, what is this situation of what I have on speaker?
So we do the outside first, like, just outside the sponge, and then.
So excited. Now you get to see it, Jared, your invention.
I saw it before. I saw when you
Lucky
when it was
number
working.
13.
Yeah.
So excited.
It was
Thank
cool.
you so much.
That was pretty cool.
Let's hope that it's not too like they're not shut off because it's nighttime.
I feel like we're positioned in a good way for a glamour shot too.
Yeah.
Where you get more of like a profile of it coming out, you know?
Yeah, it is kind of to the side,
Yeah.
right?
Yeah, I think he's kind of in the perfect position.
This is the one that we measured in the USVI,
Oh.
the same
Sponge.
sponge.
Yeah.
How deep do you guys go there?
Not very deep, everything's pretty shallow where we are, so. I'd say 20 meters or so,
Oh,
15 meters.
is that like an override?
What?
Do you have like an override button where you can stop me from doing stuff?
I didn't know if you were doing anything.
I was just
trying to get
focusing.
in focus.
Oh, so was I. So you just hit auto?
What's that?
Oh.
That white thing?
What white thing?
A worm?
Yeah, it looked like a worm.
That's gross, that's gross. It moved real quick.
Yeah.
It was like this long.
You seeing anything?
No.
Okay.
You could go inside now.
All right. You want to go inside?
You and Kalina measured ROS sponges, right? When it was shallow column?
Yes, we did a little bit. And then also on the Alvin cruise that we were on in 2019, we did sponges. And most of them, not the glass sponges, but the barrel sponges, got a signal on
How
the
deep
inside.
you wanna go?
If you can get it a little deeper, that would be cool. That's awesome, wow. Yeah.
That's great.
That looks amazing, thank you.
Oh, you guys are gonna get a sample of this sponge, aren't you? So this would be SH18684.
Yeah.
SH18684.
Oh, we're really shallow
684,
now.
yeah. Zesta Spongium.
This is definitely the shallowest I've ever worked with an RV. 39.1.
I'm cooling off right now.
Are you?
No. We were at 58 when I got here.
Where are we at now?
56.
Nice.
Small victories.
Celsius? Or?
Alright, well that's
Celsius.
probably enough to see that there's not a signal, right?
This is close one.
Oh, wait, he gave you 8.4.
You guys need to hold it. That and perhaps it's still close, like it's not.
Done a great job, Chris.
Thank you. Anything is just really bad?
You're gonna take these gummy bears while you do
Little
this?
climb?
No, my stomach's something.
Stop it.
Enjoy the candy right now.
Thanks for your help,
Have to
guys.
get in 5 minutes.
Okay.
All done?
We can
Okay, I'm like, or
dose
we can
Solaris
gummy bear
and let's then set up to do some dye.
Or do we need to Solaris this before we do dye?
Well, do you want to Solaris the coral over here first?
Yeah, let's do that.
All
Let's
right.
Solaris the coral, and then maybe even a little gracilaria on our way to the holster. Or to the, just a little stuff. Just a little drive-by Solarising.
Is that more gracilaria up there?
Yeah. It's great.
It's very red.
Okay, this coral is gonna be 685.
Where would you like to stick this one?
I kind of see a signal increase already.
What?
Zoom in.
Whoa. Is this a? Really has its polyps out.
Shine on, you crazy diamond.
Yeah, I hope somebody sees this.
Looks like a nice sustained signal. The one is in a lot of different places.
Yeah, it's kind of moving around, right? Yeah, and it's still, and it's not like overly dense either. We're not very close, so
I
that's
think it's feeding mode.
it's pretty good. Yeah, it might be.
Its polyps are all closing. You can see them.
Yeah.
The tentacles catch?
Yeah.
Oh yeah, because look at all those shrimpy things
Yeah.
around this.
It's eating.
Oh my gosh.
Oh yeah.
There's another colony behind it. If you want to check that one too.
I see the polyps catching stuff.
Yeah. Do you want to pull back, Lena? When do you think that's a good time?
Yeah. Sounds good.
Can we go back to the background seawater?
Zoom out, Chris?
There's
Come
so
down
much
with the
stuff
elbow.
in the water. Look at that.
Come down with your elbow.
That's a really cool picture.
When we're done Solaris, can we get a close up on those polyps again? I just want to see.
Can do. I'll get you a nice shot of it.
Sorry, while I'm uncovering.
Where did you want to go next with Solaris?
You can do whatever if I do this. Honestly, I don't care.
Where did you want to go next with Solaris?
Just into another spot on that same coral.
The same coral?
They're doing background again, see if it drops, and then go back to the coral.
Like how far away do we need to be for background?
I don't know.
Yeah, same configuration.
I think we're okay.
But
So
it's-
let's just,
it's not
is it
really
not
com-
coming
it's
down?
not coming down.
Oh, interesting.
Almost like,
Oh man.
yeah.
But lioness again.
Swiper, no swiping. That's what it sounded like.
Swiper, no swiping. Swiper, no swiping.
Well, you might be sucking up all those little
yeah.
critters.
That's what I was wondering, yeah.
We did that one time with our CT.
Yeah, because it wouldn't make sense why it would be lower in
Like
this
sort
part.
of
Right.
All right, then I'm gonna get a zoom in for.
You got the what?
Holla
I'm gonna
time.
get a zoom in for.
Solaris light
Wait.
dropping?
Wait, there's something. Hang on, hang on. Hold
What's
up.
that?
There's. I can see something like leaking out of Solaris right here.
It's something's dipping.
See that?
What? Where?
Zoom in. Stop.
I got it. Give me a second.
Right here. Oh, that's the exhaust.
Oh,
Okay.
there.
Good.
Yeah, that's
Sheesh, it's working.
So it's pumping and it's pumping reagent.
Heart
Okay.
attack.
No, I just.
Oh, this
Sorry,
is
just
the-
looking for a
like,
s-
I don't know.
You
Wow.
get a quick
Well,
zoom in
that was
for.
good. For April Fools.
There's a lot of left to right current, which also doesn't make sense to why we would see it when we're up current.
Oh, that's beautiful. Look at all that.
This would be a really good photo for our blog post.
Yeah. Mood lighting.
Hello, everybody. If you're just joining us now, we're turning the lights down low.
Turn back on for a little bit.
You would do this with the spikes and the dye.
Alright, yeah, that should be enough right there.
So are we thinking that signal was fake? Alright,
I gotta grab my phone. This is so
Base signal.
maybe that.
Yeah, it's like base on ship.
Okay.
That's lovely.
No, you hit something, Chris.
What's that?
You hit something.
here.
I hit something.
Are the waters just like
Maybe not,
going
maybe I just
crazy?
slipped it.
Yeah, I'm frozen.
Good spot.
Oh, yeah.
Feeding time.
This could be, this could be a 10-hour long YouTube video right here.
Frozen, I can move.
Just relax, see the flow.
Is that okay, so?
That's great.
That's good.
Wow.
Well, there's the polyps moved through that.
Oh, yeah.
They're catching things. Yeah.
All right, so what are we doing something else with Solaris, or are we stowing it?
I think we can stow Solaris, yeah?
Oh, can we measure the gracilaria?
Oh, that's right. We haven't done the red algae down here. If you, this bright red algae. Yep. All right, Lena, we're moving to the gracilaria.
You take notes, Scott, while you're sitting there?
Yep.
Got it?
In 2 different notebooks at the same time.
Are you good with that?
No, it does. Well, it looks like the other ones. Gracilaria have at least 15 planets. I don't know about the exact stuff.
Yeah.
But it looks like.
Yeah, I'll let you know when we hit another site.
Well,
I'll start it.
I guess it's because these are
I
shallow.
haven't gotten there. But J-Rod might take us
One
somewhere.
of the pieces I read too is I thought.
What is this?
Plasticity of gracilaria. I mean, it was an old meeting one, but like.
Sam, SH number please.
Of what? Of this gracilaria?
Yeah. Oh, are we collecting it?
Are we gonna collect it or just measure it?
Yeah, we don't need to collect it.
Okay, I'll give it one.
We were just curious. I can't remember, Alina, did you get peroxide off gracilaria when we were in Puerto Rico before?
There's metal piece on it.
Can't remember who was the big producer. Was it Dictyota? Maybe? We see it in Dictyota, we can try and measure it.
Some weirdness.
I think at the very least, the gene is right. I don't know about the.
Whoa. What's happening? Oh.
We should get a
Article going
music
through.
exception on YouTube's policy.
Oh, it's
Right.
Gracilaria.
Then we're gonna move to the dye.
Alright, you ready? Yeah. We're good, thank you.
Then we're gonna move to the dye on the sponge.
Dye on this one.
Dye on the sponge eye.
Sponge time. Sponge time. Everybody ready for a shot?
Ready for the glamour shot.
Unless they've gone to sleep already.
Oh.
Do we need to get the tube like on?
The upstream side. There is one to the down the smaller one over there.
If you want to use that one, we can do that one. But you didn't measure that with Solaris.
No, this is really the big, much bigger. I see now it's much bigger.
But I feel like we don't fully understand the pumping because some of them are intermittent and go through all the night and turn off during like every 2 hours.
Right.
Yeah, do that.
I got to move.
Yeah, it'll be interesting to see what this one's doing.
We are diving shallow today, 39.1 meters. We started off today at around 300 something meters, 400 meters. Worked our way up the slope. We're at La Paguera.
You need to start getting the T in.
This one?
For the pump.
We're at La Paguera, so we've switched sites. We're down on the southwestern side
This one?
of
Yeah.
Puerto Rico,
Sponges all over. Wait.
in a site well known for its coral reefs. We're using the ROV to look at some of the less accessible areas of these kinds of reefs. Depths that are really kind of too deep for conventional scuba diving operations. Only really open to kind of technical divers.
Did y'all say we're going to be sampling that red algae stuff or no?
No.
I do not think so. Okay.
Oh, yeah, you can tell these are different. These are the special things. Oh, yeah.
That was a little bit too deep.
Maybe we can hold the die on the right side of this one.
Right side?
The current's going from left to right.
This way?
Yeah, this way. I was just thinking of a way to view, so we can view it and not have it blocked by the arm.
Oh.
Oh, we need.
I'll be able to see with this camera.
Okay.
Yeah. We're not. Yeah. We put it on the right side. It's just gonna be something.
Yeah, no, I understand.
Might be able to.
Where do you want to put it?
Maybe right, yeah.
On the upstream side near the base.
Or even, yeah, that could be close
Look at
enough.
that thing swimming around.
Yeah, I'm glad we're not diving there and be like up in your face.
Yeah. Alright, let's do a little bit.
Okay, I started a
Are we
video
prepared?
highlight just in case.
Oh, yeah, good idea.
You good with the set?
I'll get a before shot.
You can give the RAM a little bump if you want.
When we're going, like, when we're zooming and you're like,
Squeeze.
there's quarterback, like, I literally see nothing. Sometimes I feel like I'm looking
It's a flowing.
Is this glowing?
You can see it flowing at the very bottom of that little crack.
Oh, yeah. I see.
It's a great for looking.
Oh, that's good.
Oh, yeah, I see it way at the very bottom there. Come on, Spongy.
These sponges are working. So they're not doing anything.
Oh my gosh.
What worth is a sponge if it can't pump water?
It might be asleep. It's
I don't
nighttime.
think it's fun right now.
Let's give it a little bit more time.
Remember?
Sometimes it can take a while
One
though.
of the keys
You just
for
know I can see downstream.
deep sea science, even though we're at 38 meters.
Does this count as deep sea science?
Patience, positivity, and persistence.
Yeah, I think it's not three.
Jeez.
But this would be consistent though with the ROS, right? We don't see
Yeah.
ROS or pumping. So that's
And with
useful.
what we saw before in the
Yeah.
USDI.
So that's useful consistency.
It might
That one
actually
was
be.
pumping and that would not also not give us a signal.
Yeah,
but Okay.
it was like 40
You want to go around the edge a little bit more? Think you can go around a little bit more? I'll bring it.
multiple squeezers jaw sign.
You know what I'm talking about?
You'll be able to see the top of it.
If you elbow out, bring your picture up, elbow, yeah, there you go. My arm.
It looks like it's like a cylinder instead of. It doesn't look like the sponge is the base. It looks like
Yeah,
it's
right.
stuck onto the rock. What is that in there?
Somebody hanging out there.
More
Ice.
elbow. Up your
Push it.
butt.
Yeah.
Up your butt.
Yeah, we'll have to try it again tomorrow when during the
Yeah,
daytime.
earlier in the morning.
Yeah. Like the same species.
Might be able to rotate
Okay.
clockwise.
You get anything growing on your plates? That's not.
No, because they're slow
That's right.
growers,
Right, let's bring
which
it
is
up.
a good thing that I didn't, that you know, bacteria are speedily taking over.
I grabbed it a bit deep. I can try to grab it with
So I'm
Happy
keeping
birthday,
it
Colleen.
At water
What's that?
temperature.
Happy birthday.
Oh, that was beautiful. Thank you guys.
Okay.
Now, we're gonna sample what?
Well, let me ask you this. Can you read? There's a tinier sponge down lower to your right. I'm just curious. We may not, it wouldn't be big enough probably for us to sample. I'm just curious if it's asleep too.
I'm gonna stick this next to it.
Do you see? Can we see?
Was it another barrel?
Yeah, but it was a smaller one.
Oh, that little guy right there?
I don't.
Yeah, there's that one right there.
Yep. I'm just curious if it's while we're in
Which angle do you want me to get at?
same angle if possible. Oh,
Just trying to figure out how we can get there with.
No, me-
It's a little guy.
view. I'm gonna come
Yeah,
a little
good eye,
lower.
Scott.
what?
Come up on
Good
it.
eye.
Yeah.
I saw it earlier. This floor seems pretty cool.
Awesome. I feel like we've got a good flow on it too.
Just wait for it to hit on this port side.
Yeah, it would work.
Yeah, a little bit more rotate that way.
Yeah, just trying to get caught up.
Now what do we do if this one is pumping?
Nice. That should be the perfect spot. A good size spot. Looks pretty slinky to me.
Me too.
Oh, there it is.
Oh,
Yay!
is it? Yeah.
Yeah?
You can see it really streaming out.
Oh, yeah.
Oh, on the right. Yeah, and you still can't. Yeah, totally. Vertical stream.
Could we make
Oh,
it
there
blacker
it is.
so the yellow, the green stands out more? Go for the glamour shot?
Roger that.
It's not like glamorous with the arm on.
Yeah, that's true. That's true.
I'm not sure.
Oh yeah,
So-
that one's pumping.
Oh, there it
Yeah.
Okay, I'll take a frame for us. Wow, cool. Excellent.
Cool.
So this guy must like to stay up late.
Should you Solaris this guy?
Uh,
And sample this one instead?
I think we should sample this one instead. We could try.
We could try the Solaris.
Should we try squeezing and then or Solaris and
I
then
wonder what the
squeezing?
negative does with that green.
Gone down?
Whoa.
Not good.
Yeah. Pass on that one.
All right.
Yeah.
I think we can stow the die.
Look at all those squid.
Oh,
Those are my three
The
buddies.
same three.
Oh my gosh.
Like,
Those ones look bigger, but
They're like coming to hang
The
out.
same
They're like, what
three.
you doing?
Those are my friends.
They're there for the show.
Is that Larry, Daryl, and Daryl?
Can we reassign that SH number to this one?
Yeah, I think that. Yeah. This is.
18684. I'll be this one.
They're so cool.
Ooh. Wow. All right.
Look at that. Oh my god.
Oh, that's.
Like they're all trying to point at something, like dogs.
They're so cool. They attracted to the light?
Yeah, they're feeding on all the.
Oh,
I mean, I haven't seen them try to catch stuff, but the lights are attracting all the little zooplankton. Because people go squid fishing, right? Like that.
Wrong one. Sorry. Trying to stop the.
Stop.
My bad.
Sorry.
They did all those Embari cruises, or that Embari cruise with Alvin. What, Alvin and like.
You did one?
No, you guys did one.
It wasn't on Bari.
Okay.
It was just an Atlantis cruise.
Oh.
Yeah.
It was in the Monterey Bay National
Oh,
Marine
that's where
Sanctuary.
you're gonna- yeah,
That's why I was
yeah.
thinking that.
Yeah.
Is there, does there's like electric current in the water? No, there
Well, you're good. You're good. I bet I was thinking about it wrong.
There might be some difference in ROE and Alvin, like.
If you, right there, if you rotate
That's
clockwise.
Alvin's a lot quieter.
Alvin is?
They said.
I guess it was always in the day, right? You guys went down?
Yeah, with
Inside
Alvin.
of it definitely isn't.
Always came up by 5.
Colonies were way bigger.
Oh, okay.
I mean, like the Paragorgia, we were in Swiftia. There's the Campo Borgia.
All the way clockwise?
When you were in
Oh.
there. Yeah, when you were in there, I think you were rotating clockwise.
I mean, we did a bunch of sponges, but not all of them made ROS. Some of them did, but.
So sponges or corals?
So the only coral we tested that did not was the black coral.
I think so, yeah.
The little pathies, yeah.
So you're hitting on the bow bars.
We did looks like I see an anemone in that one.
He's almost thinking,
I know. Acropora.
see why you get good soon.
Yeah.
See it.
Yeah. But, there's a bunch that don't. Fungia, no.
There's a coral
No,
called
Montastia.
fungi.
Tibera, we don't.
What was that last one?
Tibera.
Just want to go there.
Yeah.
Might be more current or something. Was there much current in California?
No. Lena, what do you think about the potential of trying out?
Can we bring a torch up?
Yeah, sure.
Yeah, we can do it.
Do what?
We're gonna have a down day when we do the transfer, where we could work on the methodology a little bit.
Project.
I'm just thinking maybe that would be, we'd have maybe better luck.
Right?
The only reason I will be concerned about it is because
Work out for it.
doing crystallization of flow and
You don't know what?
about the crystallization issue in the flow cell, because I've never seen it that effective at and
What's that?
We could do a peroxide rinse though,
Making
right?
sound effects.
I mean, a
Nothing
UCL
helpful.
rinse, right?
perhaps, yeah, but I've seen it do weird things related to the sigma flow. That it's really tricky quite a bit, so. But we can test it.
We could test it. We could maybe just see if that might work.
Sure.
Cause on the,
Calibration, we get like a one-time calibration on it, or?
we would, yeah. It's just if we have so much decay going on. We might,
Superoxide decay?
yeah, I mean, peroxide, we might have better luck
Yeah.
picking it up.
Well, we could also just grab some water from the stand and inject some sauce and just see how quickly that goes away.
Yeah.
Yeah.
It's a good idea. Do a decay.
Man, great call, Scott, to try the next sponge.
Lucky. I had a 50-50 chance.
Hmm?
I had a 50-50 chance of being right. I mean, I could tell.
You could tell.
Your spidey senses.
My spongy senses were tingling.
Need that to go up in school.
Oh, boy.
Sounds like you're becoming a spongiologist.
A spun geologist.
S-P-U-N.
Yeah. A geologist that goes on a merry-go-round. Har 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 har. Whoa. Special birthday joke for Colleen. Happy birthday, Colleen.
So weird. Oy.
Just grabbing it by the way.
It's a bit of a vibration.
You wanna bring the porch back in? it's not ideal, but you might be able to get it. You just can't hit hard. I don't know. You might be able, if you can keep going that, what you're doing right there, it might go right in. Shoulder and down. Just pitch out. Shoulder up. Shoulder up. Now pitch out. Elbow up. Your shoulder's going down. Just let it go. I mean, you're most of the way in there. And just push it in once you get done. You're just hooked on it with your bolts. So you don't have to push it in.
What's that?
You don't have to push it in. It's in.
Yeah, it's gonna get it off the.
Nice.
Good afternoon.
Just keep rotating clockwise.
Could you scrape it along the bio box to get it off?
I think if you just go out that way and pull.
There you go.
Yeah.
Masterful.
Nice.
What are we doing now?
We are going to Solaris, this little sponge.
Oh, yeah.
Solaris activated.
Solaris activated. I love it.
So I need to move again to get you over there, right?
Where is it? There.
Yeah, which is,
I need to be close.
I can just get you there real quick while they're doing, y'all are still doing background, right?
Yeah, we just started.
Right. I'll get you there before they finish background. That backside.
Yep. See it.
I just want to rotate. Clockwise. Opening the guy out. Turning around.
Yes.
Do this thing right here. There you go. Closer.
Oh yeah. Getting close.
Humanize your tree.
Is that Orbicilla? That might be Orbicilla. If it looks like, once we're done, we can zoom in more. If it looks like stars, then it is like the nice. Yeah, once they're done, we can zoom in there.
Yeah.
Cool.
Right.
Near the outside?
Maybe outside the base.
Yep, right there's good. Yeah, is there something that intrinsically could be in the water here?
You mean like all these little things swimming around?
No, I mean like chemically that is scrubbing our superoxide.
I don't know.
You know,
I mean, unless it's
some
just
sort of dissolved organic carbon
so
compound.
much dissolved organic matter. Yeah, so much DOC.
Yeah, you talked about that before. I think we talked about it.
I mean, we'll know once we get back and we measure all these for organic carbon, but.
Okay, can we go into the contact this one?
All right. probe insertion.
This one has these cool like lines on the sponge on the inside, there's like veins.
Yeah, I wasn't sure what those were.
Is that a zooanthid?
The zoanthid?
Sorry, thank you. I get my zoes mixed up.
I don't
The other one is zoanthid's run the outside.
outside, yeah.
Right.
And that is recently on.
I don't know.
It could be a tunicate.
You got, show the left, show the left, show the left. Alright, elbow up, just elbow up, you got plenty of that.
The sponge is like pale on the right side.
Yes.
You can see the sample pump spinning. It's a good sign.
We might as well look and see what that thing is after we saw our that's because it's coral going to school.
Yeah.
That's it also.
Yeah, definitely.
What was that little peak in the graph?
Say again.
There's like a little.
You want to what? Sample the.
There's a yellow thing
This-
just to the right of
This
Oh yeah,
coral?
yeah, yeah. This big guy.
That might be an OFAB.
Okay.
You just might need to zoom in on that after.
All right, Lena, probe is going in.
We're probing.
Oh, cool, it's going up. It's pumping. Is that a signal? I don't think so. It's just noise.
fake elevated background.
Exactly.
What was that little lump before, Lena?
It's ran into the, I think we saw it hit the grass and then it came up there. So I don't think this is the same one.
Okay.
Even a slight signal? No, so that was like 6,600. I got this.
Try to keep. Nothing, you're good.
Oh, yep.
Oh yeah.
Yeah.
It's off scale. Yeah.
No,
No, he hasn't touched anything. He's remained.
It's also a pretty small sponge, you know?
Yeah.
Eleni, we think
Yeah.
we got an idea on that coral.
Ooh.
Stephanocenia intercepted.
Stephanova?
Stephanocenia.
How you feeling, Lena?
I've never seen that one before.
check keep it center.
Okay.
Next to the varieties.
Nice.
Go towards us more.
How do you spell it?
Stephan. O.
Is that-
Coen.
Oh, I got it.
IA.
On here?
Mm-hmm. This is warm blanket that I have back here. It's keeping me warm.
I don't like this. It looks like pimples. I mean, it really does.
We're gonna get pushed closer to the backside.
Yeah,
That looks like pimples.
I gotta come out of
No,
that
the,
for a second.
the one that's
hands
like
come out
white
for just a
or
second,
red.
just shoulder up.
It came out right now.
That's okay. We also got a good measurement.
All right.
Okay.
All right.
Happy?
We're gonna see the yellow coral right here to the right.
You wanna zoom in on it first?
This guy here.
Yeah.
Okay. Oh yeah.
That's Stephanocenia.
Yeah. That's so pretty.
Can you spell that again?
Stephanocenia.
Okay. Thank you.
Zoom out.
Oh, what was the species name?
Septa.
Intra or inter?
Inter.
Thank you.
Are we going to measure that one?
Yes.
please.
Maybe we should take some of the sponge sample that the rail knocked too. It's already been.
Oh. I just realized that the sponge had knocked that sponge. I thought the rail knocked.
Yeah, we just take a little bit of it for the
That's
level
the
of
one
cultivation.
now.
Yes.
That one down here.
What happened?
If we
The
wanted to take
rail
some for the
knocked that
fungal
sponge.
cultivation.
So she said she could collect it if she
Oh,
wants.
yeah,
but
yeah,
just-
yeah, okay.
All right, we're at the surface, Lena.
It was like that when we got here, what you're talking about.
This was the last RV that was here.
There's a lot of supplements.
Alvin, it's always Alvin.
Alvin's fault.
Wow.
Oh, wow, yeah.
This is too much.
The background's set all too high.
There's even eggs floating around.
Oh, there's something in there. Is it a little bit? Oh, wait, something, that's something. No, that's not really anything, is it?
Okay, that's about a minute. Thank you.
All right. Maybe we're gonna try to squeeze this little sponge. Squeeze sample.
You're welcome. What was it at 14? Okay.
That's a lot more room to come up with your shoulder.
We were moving to I don't know, the optic world.
Okay.
It was at 001. College Route.
Okay, just shoulder left more. And then just use your pitch and your yaw at the same time to get that tip down. You got shoulder up, shoulder back down there.
Is your shallow water corals always in the bay?
Yeah, I mean, for now.
Oh, nice.
You know, Colin,
Work on it while you're doing it.
What's up?
Oh, that's not true. We did do a diagonal cycle one time. It was with a break, so. And, it was higher during the day.
Have you used any squeezers yet?
yes, 3 of them. So,
Mark up. I'll show you some profiles at some point. You'll see like signals are normally like.
you'll see, yeah.
What's up?
Okay.
Oh, yeah.
You'll see number 7 is fired. This is number 2 here, and that one did not fire, and number 3 did not fire. I forget. I think that's number 3. This one is number 4 is available, and I think this one of these.
3 is right here.
So 3 did not fire. I think that's 2. No, that's a 6. Oh yes, that's 6. 6 has the elbow, the 3D printed elbow nozzle on the front, which we're not sure that that's going to give us the best. You know, I think we want to kind of go straight in here with this. So 4 or this guy. I think that's 5. And I think we'll save 6 for kind of the last. These X mutas, I think the straight tubing off the front, I think is probably the best approach.
Yeah. Yeah.
But when we're triggering these squeezers now, you see how fast they're like when they trigger, they go. You
Yeah.
know, makes me think we might want to think about putting the Cerebex on front of them. Like when they're firing, when
Yeah.
they're pulling, like they're not like barely pulling,
Okay.
right? I don't know, something
You could try it.
we could think
Well,
about.
we could try it but in the lab again.
Yeah, to preserve for peroxide, just to have the sample be filtered.
Oh, bacteria.
Yes.
Do you want me to bring
Right.
a torch in for you to set this up?
So
The
that's number
primary
4.
pathway for degradation for hydrogen peroxide.
Alright, so now we're going to try to use squeezer number 4.
Unlike superoxide, the primary pathway for superoxide is like organic matter and metals that are in the water.
It's chemical reaction.
But yeah, so with peroxide, if we can filter the water in the squeezers
I'll bring
before
a
breaking
portrait
up, then
just
they
a little bit, so you can
can fine.
Alright, this one has.
But if we don't filter it, then by the time it turns out
And then
more
here.
microbes, it just like turn on more like that.
It might be better, huh?
Yeah, it's crazy.
Look at that.
Jeez.
That's insane.
Cloud.
Once you look the way you like, quadruple, yeah. Oh, there's worms. I like some. What is it?
Try rotating the rest.
Yeah.
I mean, like, so roll the one top.
Yeah. It's weird.
Roll it a little bit more, you'll be able to see it instead.
I think I'm gonna go upstairs and can I have my coffee cup?
Yeah,
Do
can
you
you?
want anything?
Yeah, I think my thing is in the computer, right? Can you just give me some like ice water?
So that's what you know why. See if you know now.
No. I know the dye wand quiver is there right now, but I wonder if it would help to move that up to the right side, the repositioner over there to that right corner for visibility. So if we put a filter, so one of the primary decay mechanisms of hydrogen peroxide, this is
Yeah, we use a little bit of.
is consumption by microbes. So the filter, the 0.2 micron filter on the front of the squeezer. We did earlier on the cruise. We had some problems getting them to fire, and I think we've resolved that. So they seem to be they're either if they're going to fire, they're going to fire. And that resistance in the barrels doesn't seem to be so bad. So if we put a filter on the front of the squeezer, as a 0.2 micron filter, that would remove the bacteria for the sample. So then when we get the sample back to the boat, it's already been preserved at the 0.2 micron filtration level, which would help. Oop.
Oh, that hit the edge. I could try punching out more, maybe get a different angle for the set camera.
I just The pin.
There. Oh, it's wet.
Oh, oh. Almost catch it right here between.
Chris, do me a favor.
Yep.
You see the bottom of this thing from the pipe? I'm cutting this quicker. Bring the torch out a little more.
I'll stand by.
Where's the lid of that one?
See the next lid.
Oh, I see it.
Preparation. Alright, you're gonna come in.
Whoa, what's that?
Billy. Bye.
Hello, goodbye.
Set it in there.
That looked like an incubator. A juvenile stage of human incubator.
That works. Well.
Yeah. They can be as big as 4 millimeters. Do you think that was a good size?
All right, we're using the manipulator here to grab the squeezer sampler.
It looks like how our snake is about to strike at a mouse when we feed it.
Yeah. And cha.
Like, oops, it skips in.
Chris has got to be super delicate not to trigger it.
Thank you.
Describing to all the online listeners.
All you gotta do is create one. The off to the right. Over here, go more on to the right. More on to the right. A little bit more on. There you go. Keep coming all the way in. Keep coming all the way in. Keep shoulder left. And then start using your functions to help flatten it out. So you're gonna need to pitch up. Just pitch.
That's it. Not real much.
There you go. I wouldn't grab it anymore.
Oh, oh.
Grab, 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 grab. What you do?
Are we kind of in the coral cutter?
What's that?
Are we, does it kind of grab at the blades?
Yeah.
You use Solaris to bump it. Oh, fired it.
How did that be fired? With a loose grip. Loose grip and it fired?
Well, yeah, I think maybe you were like, weren't you?
I hit the metal plate.
Oh,
I didn't touch
I don't know.
the saw at all.
I don't know. Well, it fired.
Alright.
Alright, you need me to spin this thing around in your arm?
Hold on, I'll put it back in and.
It's like a ear sponge,
You
same.
need to get out of here.
Yeah, right.
Alright. Watch out.
Yeah, maybe that could be like the background.
Into maybe.
It's right angle actually, just
Yeah,
stomach.
set it in there.
Alright, move the camera's down with it. Oh man, all that would be fired.
Thanks for all the effort. So this one, could be set just back in here. Doesn't need to be back in this pin even, because these two are out of commission now. That's the one that we're after next. The last one, or these.
Did it not fire?
It got misfired. We were repositioning it a little bit. Thank you.
Oh, you got a background seawater.
Big old air bubble in there.
Yeah, I know. I know. I don't quite understand that yet.
Is that an air bubble inside? Fish inside? What is an air bubble?
Oh gosh, that would be that'd be crazy if a fish got.
It's quite a bit of air.
Yeah. That'll be kind of sad.
Colleen, this water tastes like coffee.
No, I gave you water.
I
Oh, did I
know.
not rinse it out? Oh, sorry.
Did you?
I didn't know you wanted to
Did you
rinse
dump
out.
it?
No, it's just water.
I know. Just teasing. Sheesh. Yeah, it's a little coffee. It's okay.
I didn't know.
I was just sort of joking, and then it was like, oh, actually, it does taste like coffee.
Is that number, this squeezer 4?
Can
Mm-hmm.
you check on the power to Solaris?
Uh-oh. Do we lose it?
Yeah, but it happened last time when we did a power cycle, so I'm
Came back.
hoping the same thing.
You may cycle it off?
Maybe.
Yeah, turn it off and then back on.
Turn it off.
All right, number 4.
Tell me when to come on.
Power's back
Coming back on with it?
Yeah.
Alright. So I believe
Middle one on the left.
yes. They come back?
I'm still recording.
Alright. Camera in the out. This one? Must be.
Yep. This, I think, is number 5.
You know, what do you, are the squeezers primed?
Yep.
Where
But-
is that? Is there an outlet for that look good? Oh, I see. Right there.
There's a little air bubble in that. Yeah, so on the front of the squeezer, down there where that tying on is attached, is a check valve. Only lets water go into the syringe, not out.
Oh,
To
okay.
get the water out, there's another little NPT pipe thread plug, plastic plug that we have to take out and expel the water through that.
Okay.
Yeah, so the J-Rod was just trying to work with Chris to, in that last instance, to push the squeezer back further into the jaws so that it was out of the way of the coral clippers, the coral cutters. No. Hmm.
better?
Solaris?
Yeah.
I'm rebooting again.
Rebooting here.
Is that Team Baba?
Nothing. It seems like we can't connect to the top board.
Is that on the actual pump itself or the main unit?
On the
Main unit. You don't have a ground fault, do you? You do have a ground fault on PSV 14. It's always been there.
Oh, 14 is,
That's
you
far.
guys.
Yeah.
Yes.
Okay, that's always been there, but it's not drawing anything right now.
Even with this turned on. Yeah, 650. Okay, so it's not flooded.
Yeah, but last time, like, if there was something from manipulating this time, they were able to get comms to the main, just not to the actual.
Mm. Just not to the PMT, which is out on the analyzer. But you weren't even reaching it very far. So if the PMT doesn't power on, are you still able to talk to the software? Usually? Huh?
I mean, I'm not able to, I don't know.
No, I mean, if
Just on the top.
does the software that controls the pumps and stuff, does that come on? If the PMT doesn't have power,
No,
it won't come on. The software won't run.
no, that's, so usually this would connect and then we'd see the PMT initialize, but it's stuck on initializing. And
Oh, I
then
see.
I can enter your interface and talk to the pumps, but it's stuck. And when I came down, we had, what was it, like, super high signal on the PMT, like
Order of magnitude,
like a million, and it was just stalled on that. And then did that come on?
Huh.
It was like, remember when the live stream like stopped and
No.
that wasn't then, but if that happened, then this happened, and then maybe like
Little leaning
5 minutes.
to the left. It's leaning to the left a little bit.
Yeah.
Or do that. That's a good angle. for doing that.
Pretty.
Yeah.
Yeah, but I feel like for the month started.
Alright.
Now you can just pitch it down a little bit.
Yeah, I don't know if it's related.
There you go. Now you got that.
Whoops. Something wrong with you.
Can you go deeper first?
What's that?
Can you go deeper?
Alright.
Yeah, I would just let the top.
Give it a little close, and yeah, there you go. Oh. No. Hello? Yeah, I think you need to. There you go. Now, let's get it fired.
You want to just go right in the hole with this thing, right?
Yep. Cross our fingers.
This is my problem now. I really need to freeze this thing.
Yeah, we need to. You're almost there.
These little shrimps and stuff in the water
Just get
reminds me
them
of
in
that episode of Magic School Bus.
All right.
Can you freeze that for me?
God.
Shrink down to the size of Zilpah.
There it goes.
That I can imagine.
Good job.
Did it go?
Yeah.
Yay!
No problem.
Was it Weezer?
I'm burning the porch out.
we using this after you leave, Scott?
What?
Are we gonna use these after you leave?
Yeah.
Alright. Maybe we'll redesign.
You want to redesign?
We're gonna redesign how we deploy these.
Okay.
Yeah.
What are your sample IDs look like for squeezers?
What?
Where should I put SH number?
No. Well, this is FKIT 23-S510-SQ5.
I think if you yank on number 4, maybe though.
Just remove our whole.
So volume or the size of squeezers?
It's about 180 mils.
180
Yeah. Yeah.
What are we doing now?
All right. We have our background sample from the Niskin 4. So I don't think I mean sorry, from Squeezer 4. So I don't think we need the Niskin. So.
Uphill? Go uphill?
Yeah. Well.
sample for
Yeah. We want to sample the sponge for Kate.
J-Rod, can we power cycle again?
Why are you on?
Power's off.
Just 'cause that's all I know how to do.
Well, when you're stuck in this been there
Oh, I
for
heard that 2 days ago we power cycled 3 times.
third time is a charm.
Oh, it's kind of like the hit in the TV set.
Kinda like this dude.
For those old school, you don't even know what a TV set is.
You know, I was born in 1995.
Making us all feel very old. You're all watching
Shake
YouTube
yourself.
on your laptop.
Settle them all down.
No such thing as a TV set.
That's what you on our phones now.
Where you have to turn the dial to find the station.
I remember actually having to like tune the TV.
Oh yeah, you didn't even have a remote. You had to get up off
Well,
the
yeah,
couch. And
but
like,
then it was like there was UHF and VHF channels, and it was
Yes.
Pretty sure that's
like. But
Yeah.
that was an
Yeah.
ancient TV when I was a kid.
Yes, this is a very ancient
Mow the lawn,
TV I'm
change channel.
remembering.
luckily, I don't remember that because I'm not that old.
What are you talking about? Clicking, you don't know that.
Yeah, I'm just talking from Scott's memories. He's just told me about it.
What's that?
What business do you do? You ready for the power on?
Yes, that's so easy.
If there wasn't a U in Mom Baguera, how would you pronounce it?
If it wasn't?
If there was a word spelled P-A-R-G-E-R-A,
Baguera.
you still pronounce it
Yeah,
Baguera?
we are actually really
Mustache.
Shaking the whole box.
And that happens a lot. Is his name was what? K?
Just push it.
K?
Yeah.
Q-U-E.
Whatever, it's fine. It's not going.
It's not going anywhere.
I don't think it's going anywhere.
Alright, now what do you want to do?
Sample of sponge.
We need to take a small piece of that sponge tissue and put it in a quiver.
Alright, we got one open. Thankfully.
Yeah.
Yeah. Look at us.
Like cut a piece out.
Yep.
No left still?
Sam, can I get an SH number?
SH18684.
Yeah.
Oh, we're still on 684.
We're repurposing this number.
Okay.
Make use of it.
Sounds
Okay.
good.
Where'd Kate go? This is Mark.
Whoa.
Whoa.
All right.
Whoa.
It's like cake.
More birthday cake.
For somebody's birthday?
I do not like cake.
Happy birthday, Colleen.
I really liked your cake that I had.
Yeah, the cake's not good.
Is it the frosting or is it the cake?
It's, well, actually kind of both. The frosting is too sweet,
Okay.
and then I don't like the cake texture kind of grosses me out.
Okay.
But do you eat
You're not
bread?
the first-
Do you eat bread? Do you
I
freshly
do eat
baked bread?
I
What
do
about
eat
like a
bread.
muffin?
You know, muffins are okay, but I'm not a real big fan.
Okay. I feel like a muffin is a little denser, kind of like a cake. What about like a
What about angel
brownie?
food cake?
I do not like brownies.
Don't like brownies?
I
At
know.
least you're consistent.
Yeah.
She'll let it go this time.
You must really not like it then.
Well, you know, if it's a
No, I had
texture
a-
thing, at least it's not
I had a
just,
suggestion
you
that
know,
you
it's
popped out.
other thing.
Trying to find the loophole.
Yeah.
The
What about
loophole.
angel food cake?
Oh, definitely
There you
not.
go.
Oh.
Okay, we're honing in on something.
What about like banana bread?
Oh.
There you go.
You know,
Yeah, she
only
makes
really
banana bread.
Dense banana bread.
She makes banana bread.
Now I would root
But
your
if
golf
you
club.
notice, I don't eat most of it. It is our
You keep
children.
ends.
I only like the ends
Yeah.
because it's denser.
You open your jaws and rotate clockwise.
What about meatloaf?
Nice. 2 points. Well, she's vegetarian. We have beyond meatloaf.
Okay, I
Oh, so
made a
the-
very
so
good,
it's the texture of meat. Meatloaf she was.
root, like vegetarian yogurt meat.
Really?
Yeah, the very, you had creamed
Vegetarian gluten meatloaf?
No. It's in Target usually, but we're on the cake, there's no Target.
No, there's no Target.
I'm so
Garcia.
excited. It should be.
Thank you guys.
Hmm.
Muffin data and sweeter data and
You wanna
sample.
open
I
And
know.
sample.
one, or-
And you said there was another one you're gonna grab, right? Another.
I was just thinking that because that one's already
Oh.
broken off, we could,
Yeah,
I could.
that
use
one
it
right
on
behind
there.
the
Yeah, why
behind the
not?
arm right there.
Is that a different sponge?
It's a different type of sponge, yeah.
Where do you want it
Which
to
one's
go?
open?
Where's it going?
It's pretty large.
Quivers 7 and 7 to 12 are open.
I don't know that that's gonna fit.
Okay.
7 to 16 are open.
It's a little bit bigger than a quiver.
That's very realistic.
Yeah. Pink and-
Yeah.
Is that
Okay.
actually? Is that actually?
That's a Bob box.
Or meatballs.
Is that actually a nice Muta?
What was it?
No, it's
Yeah.
something else because it has the Zoanthelli.
Yeah. The Zoids.
Zoanthelli.
Do you want
That's
in a
why
bow
I'm
box?
not on the mic.
Well, I don't know about bio boxes, Sam.
I
Oh, is
prefer to save the bio boxes
Yeah.
for corals because I think
To
I
me
never
and
want
meatloaf
corals
just a
for
complicated
experiments.
cake.
Yeah, can you separate chunk it off of that and just put it in a box?
There's a whole discussion on cake over there.
There's a whole discussion.
There's a whole discussion on
Will
cake
the,
Rhubarb
and rhubarb pie and meatloaf.
Oh, no pie. No
Isn't
pie.
isn't vegan meatloaf
Do you
just
wanna
a
do one of these
complicated
ones
cake?
or that one?
I don't care.
I know. I should see what the girls
Yeah.
are made of.
That really looks like
Texture
cake.
of uranium cake.
Oh, you're rating a rating cake.
What,
Can we bring torch up more than that?
What is it called? Yellow? Yellow cake uranium.
You do it 8.
That's what it's called.
Yeah.
Bow box.
I'll write it down.
Yeah, one of the grossest
After you
things
pop it
in
out,
the
I'll
world
open
is
bow
apple
box
pie.
too.
Like,
Okay,
Yeah,
cook
so
for you
weird.
don't like textures.
Yeah, but
Because that's a weird one.
It is a weird one.
Yeah.
No, I
What's a weird
mean,
one?
no, the apple pie is a weird texture.
And I
That you
agree.
I
gotta-
don't like apple pie.
I don't. No, I agree. But I've had very good apple pie. Is good. Most apple pie I don't like.
I love
But
pie.
but a very good apple pie.
That's like
Pie
a
in the sky
tautology.
apple
A very good anything is good.
Rotate counterclockwise. There you go.
Touche.
Soy cream. Yeah, so I guess
It's
just
like a peach.
complicated
It's like
soy
peaches.
oh,
Most peaches I don't really like, but when a peach is good, It's really good.
So do you like your
Peaches and
peaches
plums,
fully
I feel the
ripe
same way.
or?
Peaches and plums.
Really
You
good
should
plums
talk
are
to
basic.
Luke.
But most
He
of them
might
are
have
not
a
very good.
similar
Yeah, and you do the first
situation,
bite,
really.
no?
He doesn't
Nope,
like
exactly.
straight
Yeah.
peaches,
Oh,
he don't
shoot.
like that
Would
Did
you
you
not
guys
like
do
ripe
the
peaches?
lasers on that sponge for size? I forgot to do that.
Well,
We did not.
we have a manipulator on.
At least half of it, you can still use the height.
Blasted.
Yeah, I think what we have, maybe we can do a little laser image. Still
Shoot
no
his
luck.
top right off. You guys can be more powerful lasers on this.
Yeah, I'd say
That's
the,
perfect.
the orifice there is
Six,
6, 7, 8.
Got it? Thank
There you go.
That's perfect, thank you.
Saying you're gonna burn holes in it?
Yes. Just carve the top off the lasers and then
Just pick it up.
Pick it
Not a
up.
bad idea, actually.
Okay.
Now that you mention it, we might need to look into that.
So do we know where this sample's going?
Yes.
Yes, a quiver.
Quiver 8.
Or I can disconnect from here.
This is called an accidental sponge.
This one's got an SH number.
Happy birthday.
Happy birthday, Colleen. The chorus left. Just saying happy birthday, Colleen.
You only have about 2.5 more hours of.
I sort of think it could be a funny joke for the rest of the cruise.
Oh my gosh. Too early in this cruise.
We'll just leave it on the wild.
Leave on the bar every day, every time for the rest of the cruise. Happy birthday, Colleen. Even when it's Sam's birthday, everybody says happy birthday, Colleen. Yeah.
Have more birthdays?
Yeah, he's got one coming up in a couple of days.
Can you image it?
Yeah.
It's your birthday.
Good job, Sam. Did you tell him you like ice cream?
What kind of cake did they make you?
Because they did
Ice cream
Maybe
cake.
a,
ice cream.
Baked
baked
Alaska
Alaska cake.
Whoa.
So nice.
Yeah, you never told me what kind of
It did make me realize that they do have ice cream cones and
They had a
wrappers
little
that
skit.
just are not
Yeah.
out.
They dressed up in
That was like a
wigs and maybe
The ones that were in the cones and
Yeah.
those were homemade.
I think so, because it had the ice cream that was like in there,
Yeah,
inside.
it was like covered in chocolate. It was like a drumstick. I thought it was just drumstick.
Oh, I don't know.
Maybe
I
it was.
saved one, put it in the freezer for later. So I will be able to tell.
Good call.
I was like, please. I couldn't eat anymore of the ice cream. I was gonna get sick. It was so good.
Yeah, homemade ice cream, you must want all that.
Didn't ask me anything yet.
Nothing.
My
Yeah,
birthday
it's
comes
not
up.
a shit. This is kind of weird.
That's awesome.
I'm
Great
so
job.
glad there's not the
it
ice
comes
cream
up.
freezer. That's just
Yeah.
bad.
Oh, yeah.
I,
so much
I found out that the cupboards above, like the sink and stuff, there's chips in
Oh.
there. And I found some Lay's.
Yeah, we discovered that a
Sam
couple
and I
days ago.
had some Pringles.
Oh yeah, does it
Yeah.
have those tastes like combo pizza flavored pretzels?
Sorry, I got this part right here.
Yeah. It was like some pizza.
It's like I went right back to like a childhood memory of eating those on the beach. Some doggy hands going to the Pringles.
Soggy hands.
You know, salt
Yeah, it's the
buttery.
Sand.
Yes.
Sand. Like,
You're wrapped up in your towel. Eating pizza Pringles.
When we have a chance, can we parse and look at?
I was gonna remind as soon as we were done with the
Hard off.
harvesting.
Thank you.
When I have my birthday in a few days, I'll spend
Let me know when you want power
an eighth
back on.
of my birthday at sea.
Really?
Oh, wow. Eight?
an eighth.
An eighth.
Damn.
Of how many birthdays is that?
Two last week. It'll be 4.
Wait, you'll be 33, right?
No.
32?
Yeah.
Sorry.
4 birthdays?
He was like,
at sea? That's a lot.
Now, when I say 4, that doesn't sound like many.
Right.
When I say an eighth, it sounds like a lot.
Yeah. An eighth.
Yeah,
one.
you tricked me. I was
At
thinking
sea?
more
That
8.
seems like a lot. Maybe it's just
Were they
me.
all on research vessels
Yeah,
or?
yeah, all the research vessels. Nautilus, the first Falcor, this Falcor.
And
Those
Rebel.
are maybe the best.
Although the Rebel was kind of more like a.
Oh yeah, the Roger Rebel.
I wouldn't call that an 8. That's more of like a 1/64 because we dipped
I was-
in the morning. We were
I was
like,
given a cake.
everybody was like, let's get
I was
this.
on the Rebel for my birthday last year
We were supposed
Oh.
to stay one more night. Yeah,
in November.
November.
Actually, I was on the dock,
just way too long.
Marfac,
Woo!
in COVID, in
Yeah.
COVID quarantine.
A little piece of sponge still hanging on.
Any champagne for a birthday toast after shift? No, no champagne.
There's wine.
There might be a little Sauvignon
There's
Blanc.
wine and we have soda water.
Soda water and wine.
Oh, there you go.
A wine Shorla. Wine Shorla.
Wine short.
It's good. It's good. It's quite good. Very refreshing.
I love it by Charlotte.
Wine Shorla is
I've never
sin
had
sin
this.
good eigentlich, ne?
Sick.
Yeah.
Oh.
Wait, what's it called?
Wine Shorla, which is like wine with,
It's good in the summer.
yeah,
Ooh, yum.
half sparkling water, half white wine. It's good. Lekker.
It's a spritzer.
No, but sparkling water and wine. Jack, can we get the power
You need
back
to
on?
treat this sash a little better.
What are we doing after this?
Oh, all right. We've got our accidental one.
Or you
Now we are
need to
gonna
put it
come
on.
up off bottom and start moving around.
Wait, there's an
got-
octocoral.
Oh wait, we need, that's right, SH18685
That's
Yes.
correct.
needs to be sampled. Is this SH18686?
What
The
do we
accident-
say?
the accidental sponge?
Yes, yes. The accidental sponge.
That we all can get.
Trying to knock it off. I was like, you're actually not awfully fast.
Well, thanks, J-Rod and Chris. Sponge
I thought those
Heroes.
scratches on the top of the bottle
Uh-oh.
were black coral versus.
Uh-oh.
No, that's totally
Maybe you have to wear it in Colleen's honor,
I
Scott.
think Colleen, I'll wear it. You didn't wear it.
You can wear it.
Yeah, I'll wear it. It's not,
It
it's not
reminds
my
me of
birthday.
those those Arctic
birthday is
polychaetes,
it?
you know, the big ones.
Oh, that's
Scott,
why.
whose birthday is it?
I don't see the bio box. I need to see there's a sign that always gives me a cue that reminds me of whose
Yeah, just
birthday
like
it
on
is.
Facebook.
Oh, happy birthday, Colleen! Happy birthday, Colleen!
Every girl that turns 21 does wear a birthday sash.
No luck.
I didn't.
I did, and a crown.
Alright, so what else are we doing?
Sampling that, there was that coral. There was a
This
coral.
yellow?
No, the one in the
It's
coral
like a
there.
meter ahead or
Oh,
something.
that big yellow one?
Aren't you getting cold? Would you like your fuzzy blanket?
I don't think we're gonna be able to see that. It's
Yeah.
that octocoral.
Yeah.
That
Yeah,
brownish
great.
one.
Dope.
I had to like crack into your computer
Yeah.
and find that file. I was like, you didn't even know, like,
How did you know where to look for it?
It's in PowerPoint. It was like right after you did these.
Oh, is that right after I made it?
It was
Oh,
like a PowerPoint slide. It was like PowerPoint file. I was like, oh, there it is. But the funny thing is, I had asked all them, and Eric sent me. He's like, here, I have a JPEG version of it or whatever, and it was. What does it say? It's a geochemistry of.
Oh, it was one of the earlier versions.
One of the other versions, and I was like, oh great, and then I was like, wait, that's not right.
Yeah.
'Cause it's, what is it?
Ecological chemistry.
Ecological chemistry of ocean health. Something like, yeah.
Yeah.
So it's slightly different. I thought, oh, wouldn't that be ironic or funny if I printed the wrong
The wrong
version?
one.
But I think it's the right one, right? Okay.
Sampling this guy, right?
Yes. Yeah, and I couldn't even remember what
Where are you gonna put it, Chris?
How much are they on?
7's open.
What? Oh, yeah, so you want to equip it?
9, yes, equivalent.
I'll put it inside.
Side?
Yeah. Just want to
Like
put it over there.
On like that,
Did you mark?
to the right of the arm.
I was gonna do it, but then I realized it doesn't. I was having- So we just used 8.
And 4.
You wanna go to 20 or 21?
Yeah, Paris.
The labels are mixed, the 20's on 19.
No, they didn't fix it.
Celine, is it like the lab's logo on the?
That's all the way out.
Alright.
On the blanket?
Yeah.
Yes.
That's the wrong arm.
Yep. I didn't like go too far.
That's good.
Alright, so which one's 20?
One second row on the right, or
Zero outward?
yeah, if you wanna go that way.
Official hands.
What's the number on the lid of that one?
It's gonna be 20. I imagine.
17, 18, 20, 19?
Oh yeah, 19. Yeah.
Yeah, 19.
Oh, I see, yeah.
That's a. What's up, buddy?
Oh, big crab.
Oh, wow. Oh, that's a good one. What do you think? Should we try cycling again? Or do you think it's... So when did it happen?
It was like, I don't know, like a minute pretty fast. Yeah, cause I was like, yeah, then I went on a 5-minute break, it was fine, and then it wasn't fine, so it was like 2 minutes of break. Yeah, but I was periodically, and it was fine, so we got at least 30 days. Not just a little.
So what happened?
just
It
wanna,
just went the...
like, right at the joint there?
But not on the readout, just the number, it was like.
Totally expect you to
A couple million,
right there where the branches
and
off.
it's stuck. And this exactly
Go back there.
happened, like.
Yeah, the rightmost piece is
Okay.
not dead at the end,
Testing,
so not.
and they were able to power cycle a few times, and then it just came back, but now that's not.
It's a lot longer than they look.
Yeah, that.
I tried to grab one earlier and it was all.
We got like on that one that's coming down, sticking up, like this. Yeah, like that first one.
Yeah, right there.
Alright.
Stabbed into the TV screen. I thought I could
How do you
pierce the
look
claw
at it?
and pick up the
Oh, with the
Yeah,
camera?
very impressive.
What do you think you'll see? Like maybe it's poured out or something, or, okay.
Oh, it's a chunk.
No, I think it's a good idea to look at it just in case like maybe
Or like, I do it with my friend, and I lip sync, but she's like singing, and the pig came up in the background, like, told us we did good, and I was like, yeah, I was like singing. But you get to go Rihanna song, and I was like, I can't do that. It's not good enough for Rihanna. Yeah.
Yeah, that's fine. I think it might be a little hard to get it into the quiver. Yeah, there. Oh, that's fine. That's good.
So what is the name of this one, Sam?
We're not sure. I'm just calling it floppy Flexorid.
Floppy.
Flexorid, yeah. Plex AURID.
AURID.
That's a
Okay.
cool name, floppy flexor.
So this is the third that we've measured of that.
It's the fourth and
Fourth? Okay.
final for that.
Do I get to fly after this? Do we get to go somewhere?
Could we try to cycle Solaris again? It's still not, and we were wondering if we could actually try to take a look at the wand just to see if there's anything obvious.
Oh, I missed it. Did it go in?
It did, both of them.
We decided that the signal we were seeing from this guy was just totally over his background, right?
I think so. That's really good. Was that? Is that my fault? No. Just got caught.
I don't want to sit back down.
Nicole, I think this is everything, but it should be pretty accurate, except for the spicer.
Twice.
Usually peppers, brown. Like cuttlefish, but he likes your Solaris.
Oh, yeah.
Yeah.
There's two of them.
There's probably another one.
Cuttlefish are back.
Snap, crackle, and pop.
I love how they're just checking us out.
They probably think the world is like ending. If I live in the dark and then suddenly like a huge ball of light comes, I'd be like, oh my god.
I think it's a UFO.
We're probably aliens to them.
Yeah, all three of them are still there. 1, 2, 3.
All three are there?
Yeah. 1, 2.
Yeah, look at them. They're just loitering.
Get them in this
Look at him.
It's funny how that one's facing a different way now.
Oh, yeah.
They all definitely have different personalities.
They definitely do. Coloring. One of them's kind of loud and obnoxious. One of them talks too much.
All right, Lena, there's the wand. Can we see anything obvious?
Can we go towards the base of the silver tube?
Tube, yeah.
The other side.
Oh, this side?
Yeah, thank you.
Or?
What are you looking for?
Well, it's the cycling is not working.
So the PhD had like a really, really high count all of a sudden.
Right.
It stalled out and now we can't get it on yet. It won't initialize.
Oh, crap.
So it's unclear to me what could have happened.
Yeah, I'm not sure we're gonna see anything there. I'm not sure we're gonna see the problem. Unless it unplugged. And I don't think, there wasn't anything we were doing that was straining, right?
I got it.
Okay. I mean, can't really see a loose
Well, I
one.
mean, if it was unplugged
Ouch,
was what we
yeah.
were looking for. Yeah.
But if it were unplugged, we'd get a hard ground fault, right?
Maybe on that side here. I can try to swing it out real quick.
Does that ring out?
It looks like it.
That ring?
I can't see what you're saying.
Here, I'll just come back around.
Okay. Yeah, so oh, yeah, that's, wait,
Yeah, it looks like it got pulled out.
that ring.
Does it
came-
look like it's out a little? Yeah.
That ring pulled out, which is this connection.
Oh, yeah, it was inside, so,
Uh-oh.
Okay, well, that's the problem.
yeah. We'll step it.
Oh.
Yeah,
You want me to stop it?
Yeah, so we'll keep the power off.
okay. Can you turn the car off there?
It is off.
Oh, sorry. Thanks. Is it
All
something
right.
you guys can fix?
Well, Solaris has come unplugged, so
Well, okay, that's still good, at least.
So do we want to have a look
those
around?
spin-
I think it's worth kind of just still cruising around and looking at things, and maybe we can find some varieties that we can come back to.
Yeah. Okay.
Look for some good targets. Any collections?
Fly around, third year.
We got,
We can just go on a joyride.
yeah,
Joyride,
we've got one
windows
squeezer
down.
left, we've got dye, we've got plenty of coral quivers, we got
Yeah.
bio boxes.
Yep.
You were cruising yesterday.
Yeah, finding a varieties colony would be
I would
fun.
love to find some varieties. That would be amazing. The cuttlefish are going to be sad to see us go.
Pick it up just a touch.
Oh, they'll probably come with us.
Y'all to the,
Oh, that.
either pitch in or y'all to right just a touch. No, close. Set that down. Just a little
Well,
bit.
the Solaris people can just,
There you go.
Yeah.
go take a coffee break, a nap.
No, yeah, Susan.
So, should we still prep for the next dive as per normal? Question mark?
What up, dudes?
I think prepping sots would be good, and then, you know, you could hold off on everything else just to make sure. Seems like that could be I don't wanna say quick fix, cause I feel like I'd be jinxing myself.
Do you still plan on coming up around 2 or 3?
I think we'd probably come up earlier than that now.
Right. So we could probably cruise around for.
A couple hours, but then maybe come on up and try to fix the situation and go back down.
That sounds good to me. We can easily. She's thriving, obviously. What did you say?
All right, we're gonna keep our eyes out for some future targets, since Solaris has become unplugged one of the cables to the photomultiplier tube, which is the detector for the instrument. So we'll need to bring up the ROV in a while and fix it, and then we can send her back down. But in the meantime, we're just gonna cruise around. Look at this beautiful reef. Definitely like a coral down there, something. Oh, it's okay. We can just keep going. That's a cool fan. What? Do you know what that is?
Probably Gorgonia.
Let's see the purple. Let's see the.
Which fan are you talking about?
Are you talking about this one, Sam? This guy?
Oh, down to the right.
To the right,
Oh,
it's smaller
that's
fan.
not open yet.
Up
In the center.
Up straight to the right of
To the
that
right.
blue.
I got you.
Up there.
That guy.
Ooh, yeah.
Oh, nice.
See that? I didn't even see that.
But the Agaricea here and the other.
Agaricea, yeah.
Floats on surface are just skipping to the bottom of that top elevation.
Alright.
I think I identified the floppy plexura.
Okay, let's go.
Keep going, or?
Pseudoplexura.
Oh.
Sure. Let's just keep looking around, yeah.
Pseudoplexura porosa.
Four also?
Look at those cuttlefish.
Pseudoplexura.
They're my friends.
I think they're gonna come back to the ship with us.
Ah.
If I could turn these on.
They're right on the very bottom, like, hello.
Yeah.
Ooh, look at those Agaricea here, those beautiful plates. Oh, there's another fan. This purple one here.
Oh, yeah.
What is that one?
That is the wide. Let me get the name.
Look, that fan is just all up in
It's
my
the wide mesh sea fan.
Oh, cool. So this dark purple is something else, right? Is this or is that a sponge? Or is that a coral? No, there's a little orifice thingy here.
Gorgonia Mari.
Keep going?
Sure, yeah. So yeah, we are, what are we at? 31 meters. Whew. Pretty soon we're gonna start seeing some scuba divers.
There he is.
Oh my gosh.
Both of them photobombing.
There are a couple, the cuttlefish
They're
just
getting a little
right
too
in
close
one of the
now.
cameras. Oh, we can see her eyeballs. Oh, you can see it now. That's hilarious.
They could, if they're not careful, they could become chum.
Yeah.
No, they've been with me for a while. I think they've, ooh, look at that lobster. Little lobster.
Oh yeah. Wow.
Oh, wow, right there.
Yeah.
Yeah. Ah, very cool.
29 meters.
Is the captain freaking out at this point?
Oh, we can show
Yeah, we're gonna start seeing scuba
them. Here's
divers.
the big fans over here.
Oh, here we go.
28 meters.
Big fan.
Oh, wow, look at that. Oh, my goodness, look at that.
We got a lot of fans.
We have a lot of fans. Oh, look at this. Look at the cuttlefish.
Are they
cracking.
cuttlefish?
Aren't they cuttlefish?
No, they're reef squid.
Oh, they're
Squid,
reef
yeah.
squid.
Do
All
you wanna
right.
collect some of these for experiments?
The
Is it
squid?
Solaris? Is that-
Which one? Which one?
Well, this one might be good for experiments. The sea plum.
Y'all let me know. If you want to keep going up, have a look. Come back
Yeah,
down.
we,
I guess.
You want to have a look?
we could,
Santiago's
Can I keep
probably
him? I
cool.
promise I'll hug him and pet him and hold
Ask if
forever.
they're ready for.
Oh, imagine that was Santiago.
do you want to ask if they're ready?
I see
These
away.
are pretty shallow.
Ready?
Um,
Where are we going?
27 meters.
What were they aiming for then? For
26.9.
I guess deeper.
26.9 meters. 6.9. 26. We are.
Come back, Squid.
No, they're still with me. They're up in my headache rack right now. They're right up here. They have not left me.
Oh yeah, they're there.
I got another got big bag.
Three of them. All three of them. Look at that. All
On the
three of
left,
them.
Chris?
Yeah, look at them. Right, just hanging out there.
Us on Ben, let's give him an update. I'll squeeze it through
Number 6 is still
Oh,
open.
could we look at this one down
Yeah,
here?
give me a second. Just trying to get
All but one.
Front right, number 6 is still available. Has the 90-degree 3 printed nozzle on it.
which one again, Paulette?
Oh, I just wanted to check this one out here. That looks like a mounding coral.
It's like it's praised as that, doesn't it?
Yeah, so it's
Ooh.
it's not really
I need more than mine, Chris.
it's not really yellow, but but here
I'll just take mine.
It's praised.
Yeah.
and this one over here, Nicole, right? Like this too.
Yeah, those are two.
Oh my God! And this one right here.
Yeah.
But that right there, yeah.
This and then there's a yes, there it is. That looks like Preiades
Gonna
asteroides.
drop a target.
Go for it.
We don't
Get
have
out of
Solaris
the-
working.
Oh, there's a little.
Shoes.
That we saw yesterday.
Today, 2 of them on the dive.
Turning into Sam.
Ugh, and
Am I good?
that's great. Could we look up a little bit? I think there was maybe another one, like right up.
All right, Chris got a marker drop for this. So I think, right? Yeah.
Yep.
That might be OFAV there.
It's right there.
And there's the
There's
other.
another
Oh, that's a
Preiades.
beautiful one.
Oh.
Yes.
Look at that bright green one. We used to see that one up in Australia. The
Which one?
to the left. Touch. Just behind that spiny coral or the stalk coral.
15 meters to the north.
Where,
To
where?
the
this
right.
one.
Oh yeah, that's Siderastria, right?
Yeah. I don't know
Is
what
it-
it's called. But sampled that before.
Is that a Gorgonian?
All right, I'm getting
This?
No, just up there on the left. Top,
Oh,
top
look
left.
at this.
That one up there behind the
Oh
squid.
yeah.
Hold on, guys. Give me a second. I'll show you.
Sea fans.
I'm up in the waves
Are
up
we
here.
diving right now?
Yeah.
22 meters.
Look at those fans.
Oh, we're
Oh
up
my
in the,
goodness.
surge.
Yes.
The surface surge, wave surge zone. Which might make it hard to sample.
See the. Top left.
Wow. Holy cow. This is amazing.
This is what I wanted to show Luke. You know, he's getting all excited about sea fans and stuff.
Oh man.
So there's another Preiades right here.
Yep. Sorry.
So we've been looking for if everybody's looking on the
Yeah.
on the
21
screen.
point.
These small yellow kind of bumpy corals. Those are Preiades asteroides.
20 meters. Oh man.
Wow. 19 meters.
So there's a bunch of Preiades asteroides up here. There's another one here.
Yep.
Here's another one.
So those would be our positive controls for Solaris. If those aren't giving a signal, something's wrong.
Wow.
According to experience.
So this is a coral I've sampled a lot on scuba, not on a sub at this depth. And
Oh, look at that giant sponge right there.
yeah. Preiades is a species of, or genus of corals that's very, very hardy.
How do you spell it?
P-O-R.
Is that another big one?
Nope, no, I don't think
That
so.
one, that one?
Yeah, that might be. Yeah.
No.
I can see myself.
That is not what I thought.
Appreciate you though. Thank you for letting me know. I would be amazed if USBO was tracking me right now.
It's such a steep angle.
Look at these fans. It's just amazing.
177 meters away from the at 10 meters.
Photo bombing. Photo
Take my
bombing.
mouse and
Wow. Holy cow.
We put my dials up.
So we've been trying to find parietes since through the cruise so far, we've been getting really low to no signals on our Solaris sensor for reactive oxygen. And that could be real. It could be that for whatever reason in these waters, the corals are not making it. But it would be pretty unusual. At least we haven't seen that so far. We've done work in Hawaii and off in Monterey Bay.
Hi! Happy birthday! Happy birthday, Colleen!
And Cuba, lots of different places, and we typically see that at least some species are making it, and it tends to be the really hardy species. So finding Parietes asteroides is going to be a nice positive check for us. This guy's just showing off, yeah. He's like, look at me.
I think he's posturing. He's like,
Is that
you want
squid as
to
well?
fight? You want to fight?
Look at that.
I think maybe he sees the reflection of himself in the globe of the camera.
He's like, who is this other squid?
Giant squid.
So unfortunately, we can't actually test Parietes right now because our Solaris isn't working. We need to get it back on deck, replug it in. But now we know where to come, and we can come back
We know that we
on our
come
next
17
dive.
meters depth.
Yeah, we are. Are we at 17? Oh my gosh, we're at 17 meters.
barrel.
Just jump on it.
Oh wow, that's a huge barrel sponge there. Oh, and there's a Parietes right in the bottom center of the screen.
Oh my
You can see the yellow mounding coral. It's typically called the mustard coral. It's kind of a mustardy yellow color. Very, very, very
Kind of a gray
resilient.
Poupon, right? Kind of,
It's-
it's not really French's.
It's not French's, no. It's more of a gray poupon color.
16 meters.
Can we look over the edge to see if we can see it?
You can see
Yeah,
my
you
lights
can
right
see
there.
I mean,
Oh my gosh.
oh my gosh.
And there's the floats. Whoops.
The floats have been at the surface since dinner.
Damn, that's
Yeah,
crazy.
cool.
Jeez.
That's okay. Tight quarters here.
Let me get a picture of this one.
Wow.
This is like a night dive.
This is a night dive.
Night dive.
Could you free dive to this stuff?
30.
Not anymore. Once upon a time, maybe. It's like 50. It's like 50
I know
feet.
Juliana probably could.
In a sec.
Yeah, someone says that someone we could just dive in and replug it in.
That's great. That's a great idea. Somebody should just go jump in. Colleen, you brought your mask. You got your snorkel and mask.
Somebody.
Yeah, the vehicle's only 480 volts, right? Yeah. Just don't. 4,500 volts.
Yeah, it's pretty
starting-
funny.
That's a little mound and it's starting to go downhill, so I have to come off to my left a little bit. I can't go.
We could
down.
go for a little swim with those squid.
This would be a great dive spot.
This is a beautiful dive spot. We'll have to come back on scuba one of these days. Revisit our sites.
This is not me.
This is full photo.
This is full photic.
Yeah.
Like all these gorgonians are probably symbiotic in the hard corals.
What if we were here right now during the day? Would the RV have any more lights? No, it would be fine, right?
Yeah, we normally still have to turn the lights on a little bit because it looks a little funny.
Yeah.
But, yeah, no, I mean, done it before. We can't stay here for all too long. And then
We get too hot.
Yeah, we just have to drop down just a little bit, cool off.
Oh, that the gorgonia's got a disease.
How's our tent now?
I'm sitting right about 60. I'm gonna have to go.
Where is Janelle? She would be loving this. I don't know.
I don't know.
What am I
Bit.
at? 17? Yeah, without
I'm gonna-
a little
we are
bit
at
more line,
16
I can't
meters.
go anymore. Oh, here's a couple cardiac.
Can you use me up?
Yeah.
What? At 16 meters?
I just have to turn everything down to ultra low.
Also, you know how Luke's always said, I want to see Dr. Cole in real life. Let's go.
Yeah.
I'm gonna start back up, unless there's something y'all wanna see.
No, this is great. I mean, I think this is It's wonderful to see this. Thank you.
Don't have to suck in too much, but.
And we could easily
All right.
Yeah.
Is there anything that you want to look at down
Yeah,
there?
like he could entry look, you know.
Yeah, if we could just keep, get down to maybe a more comfortable depth, and we can just keep cruising around. It would be nice to see how deep we, like at what point do we start seeing parietes?
Okay.
So if we can just kind of keep our eye out for those.
Dyer operated in situ chemiluminescent centaur. That's what it's like, yeah.
Just me, just.
It's just Nicole and her water bottle.
My emotional support water bottle.
Speaking of emotional support animals, we lost our squid, didn't we?
I didn't get a comment. Look.
Wait, an emotional support squid comment?
Hello, fish. Some bunch of little fish swimming
But
around
not
there.
many fish, right? For a coral reef, right?
Well, I mean, you can see one.
I know. I'm just saying, like in general.
Well, yeah.
Probably because of subs-scaring water.
Yeah,
Probably,
they like
yeah.
lights.
And noise.
Oh, there's a.
They were great squid, someone commented. They were.
I guarantee you.
They're
they
very
will
well
come back.
behaved. They kept their distance. They didn't speak up. They raised their hand when they knew the question.
Someone said can't be worse than sharks.
So that looks like maybe that's still a parietes right
yeah.
there.
And down to the left.
And that's at 18.
And down to the left.
Oh
those
yeah,
guys.
there's a little bunch of patch of them.
And over there.
Yep. Oh, a nice little
Oh,
crab here.
Yeah.
Yeah, there's a bunch of parietes in here.
Yes.
And maybe a Siderastria right there. Oh, another barrel sponge, Scott.
Wow.
What? Where?
Another barrel sponge
yeah.
tucked in there.
Oh, neat.
Or nurse shark.
Where? You see one?
No, there should be nurse sharks.
Oh.
Are we scared away a lot of fish?
Flamingo
There's
tongue.
another Siderastria probably here. There's a.
Flamingo tongues.
There is so much here.
Yeah.
Oh, more. Isn't that one?
There's an Agaricea.
What is that right there?
This black here.
No, it's a little thing.
All the other kinds of
It's just a sponge.
Yeah, a little more.
Can we see what this is right here? That light brown one? I'm not sure. Oh, that's a sponge.
Oh, there's your. Is that another Montastria?
Now where there's so few
So
sponges.
no, that's
Is
not
that OFAB?
Montastria.
The
That's
gift that just keeps on giving.
Oh yeah, here's
Is that Agaricea?
this is Agaricea.
Wow, cool.
Oh.
What was that?
That is like Fabiolata.
Faviolata. Oh, okay. Okay.
Looks like a parrotfish.
It was this one. Lyra, is it this one?
That was Rory.
Did you see we were up at 16 meters?
We're at 18 now, only.
Oh, there's a Parietes.
Yeah.
Yay!
Yeah, we got loads of prizes this day.
This will be good. This is gonna make Lena very happy. What is that? An OFAV?
Is this O5?
Is this-
O5
Ofaviolata here?
Yeah.
Oh, this will be great to come here. So OFAV sometimes gives me a signal, sometimes doesn't.
Ah, cutie.
This is a good birthday gift.
This is a great birthday gift.
Basket star.
What?
Oh, neat.
I didn't know they were at this dip.
Oh, and look how he's opened up.
Yeah.
Yeah, he's moving.
Oh,
Trying
wow.
to catch this stuff we're bringing.
He's like, I gotta go.
Wow, he's really moving.
He is moving fast.
So fast.
I haven't either. I didn't even realize that they move that much.
Wow.
Maybe spooked by our lights or something.
Well, when
I don't know.
I was drowning.
Oh wait,
There's two.
there's two.
Oh, is there one in the back?
Oh yeah, whoa.
Okay, can you
Wow.
step that in? Give me, like, 5 meters out, please.
And this maintaining 5 meters more launch to
Yeah, right.
What's that stringy thing?
It's a basket star.
No, like
Oh.
there's a stringy animal.
So if I see a question, it's a basket star. Yeah. It's you can see it moving. It's really cool.
Yeah, that's the
it's got pretty deep.
We saw some of those earlier when we were at. Are we okay?
Yeah, I was just fighting the umbilical for a while, and then just gave me the leash.
Right.
Okay. That wasn't.
So we
surge.
saw some of those earlier, a couple dives ago, but they were all curled up. So this is the first time we've seen it open, a basket star open on this cruise. You can see them crawling around. Really neat. Look at this cover.
This looks amazing.
Lyra, is this all Orbitella over here?
Yeah.
Sorry,
Oh, I think that was all.
You want me to go back to it?
No, I was just wondering if that's Orbitella.
Looks like it.
Yeah.
Darren, ¿esto parece Orbicella? Era chassis.
Orbicella?
Orbicella Franci.
Yeah, Francis. I was wondering where you were. I was like, she's gonna love seeing
I was
this.
watching YouTube.
Oh, okay.
I read to her, run, come here.
Run! I know.
Sergeant Major.
Oh, this is just
Have you ever
amazing.
seen so many diversity in Mexico or things like that? In the Caribbean, like this? Or no?
What's that?
Have you seen in Mexico this diversity in the...
No, this is amazing. Yeah, this is great. This is
Never
a
seen-
wonderful site.
Yeah, I've definitely never seen this kind of diversity of a shallow water coral from an
You got it.
ROV.
If you have a lady who visit Isla Maguilla,
Oh yeah.
that's our department
Another
for science.
barrel.
We have all the piers,
Yeah.
everything over there. It will be...
That would be fun. We'll have to come back and visit.
You can bring the kids too.
Yeah, they would love that.
No, just that crazy
Sometimes we
colors.
prepare like school things, like mini puzzles.
Oh yeah.
This is just amazing.
Does he know how shallow we are?
I just texted him and he said 17 meters.
Wow, there's another basket star on this one
Oh,
here
yeah.
up on the top.
So they climb out and then at night climb up onto those things and.
And feed. Oh, here's another one here
Oh yeah.
on the top.
Cool.
Yeah.
Oh, I see.
Oh my gosh. Where's Luke?
Sleeping.
Oh, look at those sponges, Scott. Those
Yeah.
are really cool. Here's another basket star here.
Oh yeah. Good eye. Another
Porites.
another barrel there.
Oh, look at little, what is that, an angelfish here?
Yeah, four eye butterfly fish.
Shiny.
That little fish thing. That
Look at that fan. That fan is beautiful.
Is Lena put that as her new desktop?
William did that.
Oh, William.
Yeah. I think.
Okay, that's the best. You guys have the best people. This one's like a little faded. There's these three down too. It's like everyone has a different setting just in case they want to see something.
We're just having fun.
Okay, so RV's
Well,
next one.
That is sponge? Yeah.
Solaris got partially unplugged, so we stopped using it, but now we found a bunch of variety.
There's another Porites. And another Porites. Yeah, they're everywhere
Back
up
to
here.
16 meters.
So is the RV next one?
We are up.
What? Yeah.
We are up.
What, are we gonna take it out of the water?
We're
You can see
exploring.
it over the sign too, right?
Enjoy the view for the water.
See how shallow it is?
Wow, look at these. What are these?
Gorgonians?
Gorgonians, yeah.
Sea rods?
Sea rods.
Oh, wow, that's a great shot.
Except that the Gorgonian was in the way.
I think these are Orbicella, right?
Okay.
Are they? Yeah. That fan looks chewed up.
Yeah, quite a few of the fans have looked chewed up a little bit.
Yeah. There's also some
What would do that?
I don't know. Could that be the?
Looks like
herbivory,
Yeah, it looks like grazing. Somebody coming by and cheer on.
There's a Porites. Another big barrel spun chair. There's a Porites. Yay!
It really stinks we can't test it right
I
now.
know.
It really
So actually, that's a good point.
So close.
How would you set down in here to work?
It's tough, but we could do it.
You think so? Yeah? Alright.
We found some cheaper.
Well, we could take a glamour shot of a sponge puzzle.
What is this guy? That bushy one right
Check
there.
out a sponge.
This one looks different from what we did see.
Oh, look, they could
Which
be
one?
on the land
That one
bar.
right there, center stage.
Oh, this guy? Yeah.
Big seafood.
Sam, do you know what this is, Nicole?
This one
Like
we've
sea
seen.
plums.
Still an
There's
octopus.
something, a dead one there. Ooh, look at that barrel.
Well, that's a big barrel.
Oh, yeah.
Legs stuck in out. Break your ankle.
That's
break
a
your knee off.
huge barrel.
Pick your knee off. Straight off.
You step in that without looking.
That was a great time site.
Another little barrel.
So do you guys die here for
16 meters.
That's a lot. I can, you can see it.
16 meters.
Oh my god. That's crazy.
Is that Siderastria maybe there? I think that might be OFAV.
So many octopus.
As soon as we saw Porites.
Yeah, I'll also make sure I have Porites.
If you'd like to start moving deep.
You wanna move deep?
Yeah, I mean, I don't want to. Oh, there's another Porites.
So you wanna go down?
Yeah, that's fine with me. Or we could, yeah.
You know,
I
enjoy
think
the
so,
birthday
that's
guy?
fine. Well, I'm loving this. This is great, but I don't want to put us. I'm sure it's kind of stressful for you guys.
I'm fine. My temperature went down and I came back up. I'm just back up to 61.
Here, right there. Here. Friday
Or
says
do you have to come up near the ship?
Oh, lionfish.
Lionfish.
What was the question about the RV?
Does the RV need to come up near the ship, or can we like pull it back to the ship?
What do you mean?
Like at the surface, can we swim to the ship or does it have to come back underwater?
It doesn't have to, but yeah, I would probably go down just to cool off the music before we do the recovery.
Yeah,
Let me just go check out this fan and then I'll head down.
Oh, there's a.
Is this a madrasis?
Oh.
Is that?
Tennessee the bag. Tennessee in the goal.
Does anybody know?
I've ever seen a plate.
Is this a madrasis?
Oh, that's it.
De Janet, ¿eso fue?
I think that's an Agaricia that makes plates going up.
This one?
Yeah. That's Agaricia. You can see the part that is
On the
flat
bottom.
On the bottom has the ridges like
Okay.
Yeah.
Interesting.
I haven't seen them.
There's some weird ones that stand up.
Cool. All right.
Angelfish.
Angelfish.
Oh, big angelfish. Beautiful.
Squirrel fish.
This is a birthday joyride.
Yeah, birthday joyride.
Yeah.
16,
Down.
15.5 meters. There we go.
Alright, you ready? You ready for this?
I guess I could do an EDA call here.
You haven't? Yes.
We just hit this place.
Start
Yeah,
pulling.
go go.
Come fast.
So many octopus here.
We're swimming south now. Back off to deeper water.
So if we can just try to keep our eyes out for Porites, it would be nice to know like how deep we can find it.
This is high speed.
Doesn't get shallower than this.
Start rolling up a little bit to
Big barrel.
That was a huge barrel.
go out to see it. Sebastian just like crawls up to the beach. Pulls it.
Y la pared, por ejemplo, una pega en la pared. Y ven.
No, I didn't. Which side?
En Buena Vieja.
You want the lights off? You want the lights off, you said?
Up higher.
Well, yeah, hold on, let me just turn back around.
Yeah.
Hold on, back like that for a second. Alright, there we go, down, mid tilt down. Slide back around.
Siderastria, I think there.
Desastres esperar, Tony. Desastres esperar.
Un poquito.
Es Buddy, ¿cierto?
Sí, sí, es como, tiene como mucho puntito. Suele ser amarillo, mostaza,
What depth are we at now? 19. There's still parietes here. There's one here.
Sergeant Major.
There's another one here, here.
The Fridays, yeah.
Yeah, there's quite a few here. I saw a Siderastria earlier too.
Little purple sponge.
There's a Parides, possibly. There's one.
Yeah.
Yeah, that pretty purple sponge there.
Yeah.
Oh, there's one.
So are we going deeper now?
Yeah, 24 meters. Is that a brain? Cool, no. I haven't seen any Diploria.
I think that's an Agaricea there.
Okay. I haven't
It's just
seen
got
any
a,
Diploria, have you?
no, I don't think you will here.
Which design is a on? Let's see if it's good. That's muy bien.
What are we at? All right, we're back to
27,
26
27.4.
meters here. Here's a Parides, I think, there. Some pretty purple sponges there.
As far back anymore, I'm just going down now.
Those are really crazy looking.
Alright, how deep did you want to go? Till you started seeing a certain something, right?
Yeah, I was just trying to see if I could still.
Seeing.
Well, there's a Parides there, so we're still seeing it, and it's
34 meters.
34 meters. Where are we at? I was wondering. Yeah, we tagged that where we were earlier, right? Yeah.
Well, see
The target.
the squiggle that goes up, that's where we went up real shallow.
Yeah.
You know, it's up
Right.
here. That target that we dropped was.
Well, and y'all took photos, right?
Yep.
So you'll have a waypoint of it.
Okay. You were taking screenshots, right? Yeah.
Let me go get them.
Nice.
Some kind of jacks, yeah, they do the best ones.
Criminal jack.
What are those two green targets that are on there right now? Up on the thing?
Those were earlier in the dive. These were at around 100 meters. Those were there's a giant yellow sponge.
Oh.
There's a big barrel.
But directly up from that was kind of where we're seeing.
Yeah.
You want to come back to those spots or go somewhere different? I can split the gap or.
Oh, maybe we could just kind of try to cruise around at this depth a little bit. What are we at here?
40 meters
We're at
now.
40?
Yeah. I can go back up as well, whatever you want.
No, let's try to stay here a little bit and then,
Okay, so right around 40?
yeah, maybe 40, 35
You
around
wanna
this
go
area.
East or west?
I don't think they, was east, we weren't allowed to do east, right, because of the buoy?
Well, we're like splitting it.
Oh,
Yeah.
okay. Well, if we can go east, that would be great. I think we already did west.
You mean, go west?
Oh, I mean, sorry, go west. Yes, I'm sorry.
Big old bushes.
See, mañana. Oh, yeah. Whoa. Big, tall, big, huge one.
Whoa! What is this?
Look at that.
Whoa.
It's huge.
It's like an easy package. It's a black coral, right? Yeah. Probably easy package.
We look like those around here.
Yeah.
Yeah. We're down a bit, yeah. Acá ven a pintura. Pero, está asustado. Tiburones hay de. Ah, sí.
Keep looking at that one, keep going.
I think we can keep going.
On the move.
What's the one we haven't found yet?
The yellow thing?
No, we got that. The madrasas, we still haven't found
What
one.
does it look like?
So, let's see.
It's like thumbing fingers.
Like this?
Yeah. Okay.
Yeah.
Oh, yeah.
I thought it was yellow. Is it yellow or not?
Yeah, it looks kind of light brown. Yeah.
That little grouper.
Looks like a grouper.
Some kind of little grouper.
The one on the right.
Yeah.
The other one. on the left.
Bonita de corazón.
Jeremy wants to know at least.
See?
Is it a sponge? What is that thing? Is it unfolded?
You talking about the green thing?
Sí, sí, pero fue como a 900 sponges oriented. Is it man or channel?
Es que hay uno de los meros, una de las veces que le decimos cherno.
Ah, okay. Just another sponge was oriented to thing.
It has a lot of holes in it.
Wow, look at that. Huh? Loretta's telling to have us look out for a Strangia.
A stranger here?
Yeah, I don't know.
It's a, before you get up by Boston.
If you're still there, Loretta, do we expect it here? Have you seen it in Puerto Rico? Another couple barrel sponges there.
Crazy black clothes right up here. That might be that octocore, huh?
This one here?
Yeah.
Yeah. Is this the octocoral?
Squid? Yeah, that's the one we sampled.
That octocoral?
Yeah,
Yeah.
this was
Okay.
reviewed. Is that what Philanthropic Barge? No, that plume is, but there's a different one that we had sampled. My best guess is Pseudoplexora porosa.
Do you want to look at it, Santiago? We can zoom in on it.
We can, sure.
Yeah.
I don't know much.
Yeah.
Species.
Is that the one that we got a superoxide signal on?
Yeah, but we thought it was fake because the.
It was like a background.
Oh, okay.
Shifted up, but the Plumapathies we got a signal from too.
Oh, right, the plumapathies.
Do you need to see it better? And more focus?
Is that good or, yeah? I think that's good. It's got nice big polyps. Look at those orange, like orange and purple sponges.
Yeah.
Just crazy.
Those are like ones that I'm customized, like just seeing on scuba diving. Trip like, oh yeah, the orange sponge. But those giant barrels are crazy.
Well, for guys Santiago, we don't want to
I have a question for you, Colleen, while you're looking.
Oh, yes.
If we ended the dive earlier than expected, if we go shallow again and pop two miskins right before recovery, or just shallower.
Yeah. J-Rod, would that be okay if we go a little shallow and then pop a couple of miskins?
But just before recovery for eDNA, or else it will cook at that temp.
Oh,
So
okay.
the closest to recovery as possible.
Oh, okay.
So we'll do it right before we recover. Is that right there?
The red branchy thing?
Yeah.
Yeah, that was fine.
Yeah.
37 meters.
It was just one because there were a whole bunch of fangs.
Bermuda. Another one there. 30 meters.
Wow, that's a big whippoorwill.
Yeah, it's huge, right?
Super long, so 3 meters long.
Have you gone scuba diving? Have you gone scuba diving?
Not here.
We definitely need
I've done a lot
to.
in the Caribbean, like in the Bahamas and stuff.
This is beautiful.
Here?
Why? What happened?
Is scuba diving better than this?
No, absolutely not. This is pretty amazing.
The waves were what? The waves were so big.
Oh, look at this. Look at the
You're trying to back out and do it.
crab up there. Oh, look at parrotfish.
Yeah.
Is that a parrotfish in there? Oh.
Oh, he's got his little.
He's got his cocoon.
Yeah, he's got his
He's got his,
mucus
little, his
cocoon.
mucus cocoon.
Oh, I love it.
They do it so you can't. What's a mucus shark? They nuzzle in and they make this mucus cocoons. The sharks can't get to them.
Did you
Well, I guess
screen
that
grab?
can't smell them. No, it's too exciting.
Wow, that's
Oh my gosh, so cute.
You're not gonna like a good mucus cocoon.
There's a quote for
I
the
didn't know
book.
that. But
Nothing like a good mucus cocoon.
nothing like going to sleep in a mucus
I
cocoon.
can't wait to
Amazing.
Mucus cocoon.
Because I remember when I sleep, I'm like,
He is smiling at us, I agree. Oh, that's a big sponge.
Look at that sponge.
Or Serapathies. What? Or Serapathies. This stuff they could be out of. Doing this Serapathies. But that's
time the tide is supposed to change around here? Y'all could look it up.
You want me to look?
Account for when we go back up there to take the listings. 'Cause right now it's good weather to get up there and do listing. But I don't know how much
Are
longer
you
we'll
looking
stay
at the
down.
tide? Yep.
Yeah, 'cause the conditions are perfect to be up there.
Okay, okay.
I'm just moving real slow, Ben. Can you have one of the guys look at the fishing, the buoys out there, see how close we are to the buoy? And text, Mike? I think we're still a ways away. Okay. What's that? Just a big dent up?
Interesting that it's getting a little more like macro owl kind of cover over this direction. Like we had that one patch where it was like really corally. That's a cool spot.
Hold on, I got a gigantic one out here. This thing is massive. This thing-
Oh wow. You see that, Sam?
That's like the bed I was talking about.
This is
So there's like thousands of these. They form like forests.
Is this a black coral?
Yeah.
Wow. That is crazy.
Can we zoom in on its polyps? It looks like
We
there's
know
a lot
so
of
much
stuff
about
in the
these.
water.
You know a lot? Yeah, because they're easy to get.
Oh well, no, nothing.
And you said you want to have a zoom in on the polyps?
I'd like to see them. I don't know. That's one thought. Because it looks like there's a lot of food in the water right now.
Not go crazy.
So pretty.
So it just was high tide. It's not a very large tidal range here.
Okay, cool.
It could, but I don't know if
Half a
there's
meter.
other species that will.
Okay, then we're good.
Brandis or breeding.
Wow, beautiful. Thanks, Gerard.
No worries.
That's so cool to see the little food around. So do they fire the way
Do you remember when I said we were still seeing varieties? It was like 30 meters?
I think we saw the last one at like 38.
38 meters? Yeah.
Is that a spike right there?
Where?
That big yellow thing? That big knobby thing?
I think so, yeah.
Okay.
It's not from that traces.
No, I don't think so.
What's this? Oh, it's a falling plant.
Not that much garbage here either.
There's a big arrow.
We didn't move me.
You having fun, son?
Hey, Colleen, what are we gonna do for the rest of the diet?
Well, I mean, we can't Solaris, so we can zoom around if we want to collect anything, or we could just come up and fix Solaris and then go back down.
Did close to the surface, I'm waiting for Luke to update, giving him the lowdown.
Well, we can grab a few sponges for the fungal cultivation.
Yeah, if you see something you like.
Okay.
Can we collect a piece of that anti-bad piece, that big black coral?
The
The big bushy one. Did Santiago say anything about collecting any animals or
No, I
anything?
couldn't find it. I ran
I
out.
just saw it. He was going upstairs.
You want
Go back to bed.
this one right here?
Yeah.
Do you want that purple sponge?
Is it going in a bow
Yeah,
box?
I was thinking that one.
You're gonna tell me if
Go in, you can go to the quiver.
Sponge.
you ready for Black Coral Olympics again, Ben?
These guys won't be so bad. They're not so
That
sticky.
coral you guys were
I don't
after was
think.
terrible. It like sticks to my forceps, like my, just like metal, just like grab onto metal somehow.
Like these lines.
Yeah, that's-
All right, well, we've been just cruising around,
The parrotfish woke up.
having a birthday romp here, which was amazing. Oh, there's a parrotfish. No, it's okay. Oh, so cute.
Get in your cocoon. What are you doing?
Go back to your mucus.
They can do it 2 times. So.
Oh.
Okay, so you said we're putting this in a quiver, right?
Yeah.
So how old do you think that black coral is that is that huge?
Not so old. These guys grow fast.
They grow fast?
Relatively fast.
Like,
Unless than, I mean, we don't know the ages so well. I wouldn't say it's
just get
hundreds of years old. It's not maybe tens.
Somewhat far back from it, so we can get a.
Tens of years old.
Into this thing.
Just guessing.
What's that? No, no, you're fine.
These are much nicer, warmer waters up here than
How
the
you wanna go? How you-
deep sea corals that grow much slower. It's colder, less nutrients.
Try to get like right down here.
The little parrotfish is trying to get back in his little cocoon.
No, this big bush one thing up here, yeah. I'm gonna come around here. I think this might be where we
Right,
can
this is-
that's not the madrasis? That's a sponge, huh?
That's the one I thought. You told me I was wrong.
Oh, you think that's a coral?
On the left, is that a madrasis? Or is that sponge?
I don't see olive.
Sponge, okay.
Really sponge.
Let's see if I still currently uploading that. Or I'll get you some minerals quickly for you.
Yeah, it's a sponge.
Sponge, yeah. A sponge.
So,
You say a sponge.
A sponge.
let's see if you'll be able to reach this thing.
It's spongy.
I think you will. I think I might need to move over. I think I got you too far away from the thing. Yeah, I got you too far away, right? I don't think so. Yeah, but I can maybe get you in position. Yeah, I'm gonna come back over here. A little bit more. Something like this.
Wow.
Let's see. Where do you want to put it at? You want to put it on outboard or inside? He said it's not going to. What do you have open? Seven's open. All these. Oh, that porch is coming up?
Well, this is independent technology.
It could be one Ricky or Brent. They're kind of hard to separate.
Oh, stop that camera's coming back.
If you know what you're looking for,
So it's coming down.
it's got lower branches.
Excuse me.
Happy birthday.
Happy birthday, Colleen.
Well, we only have like, how many more minutes of that do we have?
An hour.
2 more weeks.
Hour and 12 minutes.
Do you think you have the species name for the Adresia?
You think you got a name?
No, I asked him for one.
Which Agricia?
The one, oh wait,
The ones we sampled or the ones we saw?
the ones we saw, do you think there's multiple?
There's a lot of them now.
Okay.
There was, remember the Agricia that had the pieces that stuck up? As a lobe?
Hey, Colleen, I'm wondering if you want to do a CTD here somehow.
Luke definitely won't say no to that.
Oh, yeah.
Well, a CTD. I kind of feel like we could do that tomorrow.
Whatever you want to get, man.
You can take that little bit that's lit up right there.
Question for you guys: if we're operating in 40 meters of water, well, yeah, 40 meters of water.
Oh, there's a little salt there.
Would you be less or more or less comfortable putting this a CTD down? More comfortable? Okay.
Yeah, because like worst case scenario, we need to be in the shallower water,
Right.
and if something bad happened, the vessel had to move away.
Yeah.
Like, they're going to deeper waters.
For you, yeah. So we could potentially do that during the day tomorrow, Colleen.
Yep, yep. I think that would be a good
It wouldn't be
idea.
a 400-meter cast.
Right.
Well, it's actually,
That won't fit in the quiver, it's definitely one of our box.
Stuff in it.
Do you want to try to stuff it in, or do you just want to go straight to bottom?
I think you just go straight in the bio box.
Yeah, I mean, we're not staying in all that much longer, right?
Right.
And one is open?
The only thing with anything in it is 1A is the only thing that's open.
So do you want to go
1B
one
is
in,
open.
or do you want to go to one of the back ones? That's good. Roger that. Gonna open up 3?
We're gonna pop this piece once we're done here at this step.
at this depth? Okay.
Yeah, but closest to recovery as possible.
Number 5 and 6, right?
Yes.
And how shallow do you want to be for that?
We'll keep
Yeah.
at this depth because I think we want to keep more towards most mesophotic.
Okay. Did you get 75 liters pumped?
Not yet. I still have 2 more pumps to go.
Okay.
So I didn't want to do shallow either because I would have messed up the triplex through. It's a little joyride, you know. On a 75-liter joyride.
The amount of diet, you're gonna be up for a while.
I guess this is the shallowest buck or 2 is dropped in our river.
Mostly I wanted to ask that rest in the water because I didn't really think of the space.
We actually saw,
I think we made it to 10 last time.
I saw 15.0.
I think we made it to 10 in Australia.
Really?
Somebody on the chat said they breached the surface.
We did, well, that was just plain, you know, but yes.
That was what playing on
Yeah,
ours.
we did. But we were definitely up in it, just doing this,
Yeah, okay.
like how we were just about to get there again.
You know, survey reaction, well, it's out of the water already.
You know, when we recovered a Century, we had to hand it off to a trader. So we grabbed Century, brought it up to the surface, and
That's 3.
You had to do that?
yeah,
What happened since we lost
Was that bio box
Yeah.
3A?
Lost propulsion?
3A, yes, yeah. Well, it got stuck on the bottom, and they couldn't come up, but they had all kinds of issues. So we went
Where was that?
Australia.
Oh.
I have a really good photo. I put a sticker on it. And it says, Sebastian touched me here.
All right.
With
Am I-
permission?
Yeah.
I mean,
There was consent.
is it because it's dark out that it's not dangerous to break it up so shallow to
All right.
heat up the urban?
I was wondering that
Axe
too.
what I said. I'd like to pump this. I would like to pop 2 bottles right now.
Right now.
Thank you.
Before you forget.
No, he lots of things, lots
Okay.
of decision making.
Lower's coming off, open up tires for you.
Popping 5 and 6, Colleen.
Okay.
Shit coming down.
Excuse me.
5 and 6.
Also, just wanted to wish you a happy birthday.
Of course.
It's Colleen's birthday.
Yeah.
So we're all getting a little loopy over here.
Alright, that's you lined up with 5 then?
I always forget that there's a sign on the other end that tells me. Oh, yeah.
That's what we all need.
Reminds me.
Good fire.
All right. That is going to be SH13 566. Bottom 5.
Another good fire?
Awesome. Thank you. SH13
And then,
5 6 7.
what would you like to do now?
I think there was a purple sponge Kate was asking if we could grab.
I'm gonna start pump 4.
Like right around here?
Yeah.
Behind the big coral frog.
So if, I think it might have been up a little bit,
3 5
Back around that way.
perhaps. Yeah.
Let me fly up.
What was the name of that black coral we just sampled?
Antipathes grandis.
Antipathes grandis.
I was gonna say, oh, I think I know they call it grandis.
Which is absurd.
So good with naming things.
First species, because we're doing species limitations on this and related species.
Oh, hey, where's my buddy? Where you at?
My colleague is.
Come here.
Oh, yeah, there he is.
Oh, he's back.
There he is.
There's the. Oh, we missed you.
Can we get another shot of that big black coral so we get the whole thing in view?
I just want to
Okay,
say, my
he's
friend
That's the way of saying no.
Hi.
my buddy.
Hey, buddy.
Oh, I think he's waving at you. He's waving.
He
Look
is,
at
he
that.
said, tell you guys.
He says, don't
Oh
hold
my
it.
god.
The boat's that way.
That way. Bye.
Oh, there's the purple
There's a sponge.
Oh, okay, that guy. All right. You said you wanted to overview this one?
Picture of the video.
Yeah,
Yes,
the
the
grandis.
this guy.
All right, let me try to turn these lowers off and see if I can
And those spongy.
maybe get rid of some of these strippies.
Depth 37 meters.
There's that.
That was good.
Oh, that's nice.
I'm gonna come
Look at
around.
that.
So many scraps.
You saw the joyride, right?
Joyride.
Shower.
Where's Erica?
The shower water.
This is going in the quiver, the purple thing?
Yes.
Yeah.
We're just gonna trim off this.
You're like, oh, she's on the deck.
Eric, Kay, we're just gonna trim off the end of it.
Okay, turn off the end is fine, food is fine.
Yeah, I think that's good. Maybe even off the left end, so
That-
that,
the
that
smaller
little
one?
one, so that we don't take the beautiful big one.
Alright. It looks like a oboe.
Looks like a didgeridoo.
I was gonna say it looks like a
Like a spongery-doo.
didgeridoo, yeah.
Yeah, for the fungal microbiome sponge. They're understudied, and often in sponges they're really low abundance. So if you sequence, you won't necessarily see them, but if you cultivate them, you will see these cool fungi. Curious about them. They're known to produce like bioactive molecules interact with each other. They also don't know if the fungi and sponges are like, if they're symbionts.
You're seeing anything weird?
Sometimes they probably are, or if they're.
We started at 400.
Alright, I'm just trying to stay away from that.
Like if there's those spores that just like
That's
get
That's
stuck
an
in the
interesting depth
tissue
too.
and hang out there, and then you grow it. That's
80
interesting.
meters is pretty interesting. It's kind of like
Can you start working your way out there?
You don't dive, it's really shallow.
I'm gonna try to get
Pretty
stable.
low abundance,
I just really don't wanna mess up that thing.
What kind of sponge is this? Is this the Elysium?
It's not white, but
Is that
yeah, I
that
think it might be. It's the same.
Like bacteria.
I looked at a sponge guide and I just lost all confidence.
No, bacteria is
It's
very
like
high.
some species that have like all the
Fungi.
shapes, all the colors. and I was like,
Okay. Also,
Yeah.
sponges are one of the few organisms on Earth that have an archaeal symbiont.
Really?
Very
no, my
cool.
mom does.
Has that
Yeah,
archaeal
they're-
My mom does, yeah.
You're good, babe, go ahead.
They're super
That's
ancient.
it?
I'm gonna be rotating a little bit.
So I don't need the whole one. How big is that?
Probably 10 inches tall.
Yeah, the claws.
I would just take half of it if we think it could live after
There you go.
maybe
He's very-
7
very
hours.
deep.
You like your sponges, huh?
You know,
Yeah,
cup
I do. I don't
top.
want to hurt it.
I think sponges could grow.
Yeah, stable, right?
Wouldn't just
Stable
like a
enough.
small nubbin of it regrow anyways?
Really, I love it.
Like even like
Yeah, usually you can cut them up and then they regrow. Yeah, so maybe just half of that.
Okay. So it's top half of this guy. Okay.
Very weird, like, you can hear it from the surface. So dollar.
So you're gonna take like, just, you're just gonna use a small piece.
I'm probably going to use
You gotta cut more.
Should we walk around tomorrow grind the sponge on her wrist. Like,
Oh, that's a big
We could
piece.
leave behind to the Puerto Rico, to their institution, leave behind the bit of the sponge, the top hat, if you're not,
What do you mean to like dry it out?
for identification.
just like for education, just general display purposes.
Yeah, that's a good idea. Let me see how much, once it's like out, how big it is.
One
'cause I
sponge,
use
one black roll, one salt roll.
I'll use a fair amount of it on the cultivation.
Whatever you guys.
I like
Primarily your research, secondarily.
Would we put it in ethanol?
You want aim it for quiver 7?
That's correct. It'll lose its color probably, a little bit. That's definitely.
You know,
When they're paint to.
the crinoid
Say-
like dyed the ethanol like black.
I saw that.
Yeah.
Rhinoid did?
Yeah.
Yeah, they
We
can
don't wanna
miss
waste
crazy colors.
Just dry it. You're good.
You could just strap the sponge to the side of the basket as a noise for stick a support on the pole.
Whip curl. Get out one more time.
Perfect.
Only
Thank
black
you
pearls
guys.
ready, isn't
Great
it?
job.
Other than sponge screws, you guys just sticking sponges all day.
Yeah, there are. There was one, the one on the Nautilus that they were watching videos from actually. So I went, oh, how nice.
How would they plug in sponges like this? Like trash? They sponges all over, push them up.
I think they have different clippers on the
What are we doing next?
Coral lenses.
Is there any other collections we want to do right here? Did we already do the Niskin bottles? Yeah. All right.
We can always get more sponges.
Did you see one?
I'm sure we are.
We could do a glam shot of the dye in a barrel sponge.
Oh yeah. We could get like position it in such a way where the arm is
How to do.
out of the way.
Position it in such a way that the arm looks like it's the sponge's arm.
Sorry. What do we have for process? You guys got a lot of samples to process, right?
Not too many actually.
Okay.
Only like 12 or something.
We do have limited time here though, so we could get up on deck and fix Solaris and then get back in.
I gotta turn squeeze it around.
Turn this, yeah. We've got some squeezer processing to do.
For 25 more minutes, I'm not gonna say no.
What's that?
So if you wanna stay for 25 more minutes, I'm not gonna say
Oh, are you
no.
doing eDNA right now?
Yeah.
Oh, okay.
If I could get a full suite, that'd be great, but
Do we need to stay at this depth?
ideally.
Can
Or
we,
deeper.
can we zoom around a little bit at this depth?
Oh, yeah, of
Okay.
course.
All right. So we'll do 25 more minutes.
But we're not gonna let their stuff on. Well, there's the pirate rules of spot ones, or is it coral? Speaks to coral.
Do you want to go back east or?
No, it's assisted asexual.
Where are we gonna need to be to come out?
Well, technically east, but not by much.
Okay. All right.
I can keep going west if you want to come west a little bit.
Sure. We could go west a little bit more. That would be great.
And just staying around 40, correct?
Yes.
For eDNA?
Yes.
They pack you.
No worries.
This will be really informative.
Similar sites. They go-
Oh, never mind, we're gonna plug it
No one knows they actually do it.
Yeah, exactly. So much luck to that.
I feel like we've been to this rock before.
Well, yeah, we went past it and then we came back.
Okay.
There's not that much CCA.
You're right.
There's too much.
There's a lot of green.
Too much algae.
Green
Wow,
algae.
what's that red?
Is that another anti-bad piece?
Sponge, yeah. Grandis.
They don't have very, they don't travel very far, they just land and settle. So they go in the fields.
You don't say.
Is it?
There's another one.
Oh, wow, yeah.
Oh my gosh.
The only thing that's done is.
Forest.
Oh, yeah. AutoCast. AutoCast with the internet.
It was very high biodiversity.
I thought we died there.
And another one. God.
It's the only way I'm finding something around.
Lionfish.
Lionfish.
Boo. Pew, 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 pew.
I didn't want to say that.
Oh.
But you wanted to say that. What are those things? Sponges? They look like mushrooms.
Purple?
Should I take offense that you always leave every time I come?
No, I'm legally not allowed to be here before.
I get it.
I've been yelled at. Alright.
What's our altitude right now?
Like you're not allowed to be in this.
About
4 meters.
Well, that's the back of the ROV, but I'm skirting the bottom, so.
But the back is where the,
Okay.
where the United States is.
Okay, true. 4. That's okay.
4 meters.
Where are you finding it?
See the right below the circle, out.
Oh my, this whole
Listen, I
time.
tried.
This
I
whole.
tried that, but WordPress is like, no.
Yep.
You're like red.
Now you know.
Because I'm shifting
A little painful. I stared at the screen for so long and I just didn't think.
Oh, wow, this is a nice.
And right next to it is other.
I know,
4
well, I
meters.
always look at.
Oh, that's fine. You always look at depth.
Yeah.
Right next to it is out.
Trunkfish? Cowfish? Trunkfish?
Oh, look at this guy. Look at this guy. Where'd he go? Oh, there's a Asteroides. Oh, look at him.
You found parades here?
Yeah, there's one right here.
We'll circle up in it.
Cowfish, trunkfish.
Turn around. Turn around. Come on.
Turn around.
We don't yet know.
I see you. Look at you. Oh my gosh, so cute.
There's one person in the world working on the whip.
He's giving us the stink eye. Look at that. I love it.
I don't deal with it. You have to focus on it or
That was awesome.
Jeremy, when I come to the Smithsonian, do you see all of your collections?
They regularly do tours in the museum tour center with collections. You can like open up the door like, ooh, an elephant.
I've been there. I used to work at Cal Academy. And whenever I would like run an incubation experiment, I would like run over and talk to someone.
Ooh, Russell area.
Wanted to show me their collection, and I was.
This guy right here, to the left, this white thing.
It's so.
Which one?
Trying to get
If I
there.
get the funding for the arm, then I can.
Ooh, this white guy.
What about that? Yeah, what about
Yeah,
that
we
white
can make that
sponge?
happen.
Is that a sponge?
Or the
Yeah,
red
that's a
sponge?
sponge.
They're gonna make me.
Yeah, I'm gonna be
Oh.
there second, third.
Is that white one a sponge or a coral?
Yeah, it's a sponge.
That's a sponge.
Yeah,
Why don't we take a piece of it?
You want it?
15th through the 19th.
It's a short trip.
I know.
I'm
But I'll
not
have to
taking
book
it.
an
It's a
appointment.
coral.
Coral.
Oh, lionfish.
If we have to
Yes.
run calling, we could also do the red
Right, so
stocky one on the
he's
right.
next door to my son.
This
Oh, nice.
this guy?
Yeah. Just an idea. I mean, if we're gonna hang around for a while, it's just like, hey, can I
Yeah, if you want to do the white one and the red one.
Yeah.
Yeah, that'd be great.
Doesn't travel often.
Can we do more collections?
Colleen, one of Colleen's former students, Jeremy, is the curator for the mineral collection.
Yes.
Oh, that's a really cool collection.
Yeah.
Do you know Gabriella Farfan?
No, they did a tour for all the postdocs.
Okay.
The mineral collection, and I couldn't be there for it, but I wanted to. And everyone said it was super cool.
Yeah. She's awesome. I can hook you up if you want to go check it out. She's really, really nice.
Yeah, no, they regularly do these tours. You know, they get like a curator, and they're like, all right, let's check it out. And it's always so cool, all the behind the scenes
That's
stuff.
really nice.
We went there one April for March for Science with our kids, and her predecessor was gave us a tour behind the scenes. He was giant crystals.
Yeah, like what's behind the scenes is like what's out.
Yeah, I mean, it's just so much fun.
And it's amazing.
The museum support center is the best place to go. You just like pick a random floor. The whole building is just like specimens.
Yeah.
Random, like you just opening up drawers. It's crazy. It's so cool.
Yeah.
Types everywhere. Types everywhere.
Yeah, very neat. So what depth are we at? We're at 35. And I definitely saw Parites down here.
I don't know if you're gonna be able to get the microphone first, but I got you set up for red, and then I can maybe bring in the rails to get you to that one.
Okay.
You wanna do these guys, or do you wanna do 9 and 12? Porch is coming up.
Jeremy, are you reading another paper? And like in the citation, there's just like stuff about did you like it? Because they seem to have a lot of worlds there.
They did. Historically, they did.
That's it, all the way out.
Recently,
Run in the last
they
pump.
work with the museum.
Should be quick.
Yeah, they're really good. And fish. They do a lot of like really good fisheries.
I don't know how it works in Australia, but we're usually funded.
Yeah. Yeah, you don't pay for it.
Well, I don't know how it works.
You should have lots of fish.
Let us know.
I ate them all.
Good job.
I helped.
Sorry. They're so good. It's really bad to have a big bunch just sitting there because it's just easy to just keep popping them.
But there's reasons. Bird Islands.
Take it down in the Baba?
Lizard Islands. Orpheus Island. Those both fishes.
I'm trying to just get some support while you do this here.
Red Swan is something called Red
We're
Swan.
picking 2.
You could work on it next year.
Go back then?
Yeah. Well, it's a little far out, but
But I mean, like, well, like, if you're studying there, you could volunteer at the museum or curation. I guess it'd be fast.
do they have a connection?
We collaborate together. There's one, it's just one big group of connections. Santiago. Specifically people that are creating.
Yeah. I know.
Is this 86 for a collection?
This is 1868,
8, 9?
8, 9. That's a good group of people.
I'm off. All
What's the
right.
last one you got?
8-5 was.
Okay.
Coral.
8-6 was the.
Oh wait, 8-6 is the last
8-6
one.
was the accidental sponge. Yeah.
Okay.
8-7 was the
That's
antipathies.
this red one
Ah.
right here, right? Hey?
Yes, please.
Okay. Oh, that's 8-7.
That's 8-7.
Okay.
8-8
Okay.
is purple sponge for
Oh,
Kate.
the
That's
purple sponge.
Aplysia.
Gotcha. And then this is the 8-9. And it's a red sponge. All right. Thank you. This is all at the same time.
How much you want?
How about the whole thing?
Whole thing, please.
The supervisors. Do this, and if they say yes, they're usually
Like before you're accepted?
Yeah.
You're on the
You
rock.
could just have 1 or 2 supervisors, and then
Turn your elbow
once you
in.
start, you can
There you
close
go.
the door.
Guess I'm supposed to do it differently.
If you just a little bit.
It's different. I
It'd
mean,
be better
you
all
won't
day.
do a master's first.
Now you're not gonna hit the rock. You're going like that.
You can't just do a PhD without a master's.
You might just be able to get it with a red.
In the States. But you're doing both at the same time. It's like 5 years, you're doing coursework, so we work for taxes and masters. And then, then you go overseas.
In Australia?
Australia, you would even do research, you should have either a master's degree or a research component from
Do your soft grab that you did before?
master's
Without the,
degree or
without the
undergrad. And then you go apply
That's
for-
perfect.
But yeah, you want to do a master's anyways. It's a little faster than doing a PhD in the States. Master's is 1.5 years. Yeah, and then back to back.
Okay.
And they can be related, so like, if you do your master's in the same area, you've already got a paper out. So you already do 3 papers for a PhD.
If it's easier to chop it higher up, that's okay.
Okay. So you can just drop it.
Well, his first, he was in the lab for 2 years, his first 2 years, and it failed, and then he switched.
There's no rush.
There it is.
I think that's one
Very
thing.
good. That's
Take
amazing.
time off, like, pretty young. Go to another country.
You should just cut that in half and use it as a.
Then when you're ready, you want it. You don't want to do it before you're ready.
No, I want to add it to the collection.
There's lots of people who go to Australia and work in the forest.
I'm gonna.
Like, pick fruit and stuff.
You know what I mean?
And you get that in, and you get to live in
Get that
Australia for free.
blended and then rotate
And
it
there's
out.
a piece that you can do specifically for that. So Australians don't want to do that work. But like foreigners come.
It's pretty long to do that.
Australia
But
for a
I mean,
while.
and usually you just happen to have one thing, so if you bend it right now, it'll start going. Yeah.
You're fed.
Actually, you can get rid of them.
You're fed.
What do you do? You get fed. Okay. You get fed.
Brilliant. Those are pros.
I think it's too long to fit in.
I'm just worried that it is, but it should fit in like this.
Oh, nice.
Brilliant.
Here, over it, push, come down. Shoulder left. Alright, y'all, y'all right? Wait, y'all right? That's good, pop out. You gotta rotate left. Try to go. I don't know what it is. Yeah, just like that, now keep rotating that. Go. Keep turning up. So I need to start rotating, pull it down, so you need to pull it down. Bring y'all to the right. There we go. Straight y'all to the right. Y'all to the right. Y'all to the right. Okay, shoulder left. Shoulder left, y'all to the right. Just sharpen it in there.
It fits perfect.
Yeah, nice.
It fits me the perfectly.
Brilliant.
All right. How are we with time on the
I
email?
have to wait until this is
Are we taking?
over to keep going.
Are we taking any more?
So
time?
the sample will,
Okay.
but it's okay, I'm on the fast pump, so it should only, it should be less, should be around 12 minutes.
Okay.
For me?
Once
Four?
that quiver goes on, I'll press it once. 10 minutes.
So now there's five
Just
spots.
shoulder left. Four. You got it. Okay. Oh, you got an elbow out.
This one,
Elbow out.
this one,
Alright. You off to the right, please.
Did they do the white one?
Four right over more. Alright, now just shoulder left. Just shoulder left.
Get the chin, elbow in, yeah, elbow in.
Oh, you gotta bring your yaw to the left a little bit. Bring your yaw to this. You got something there.
Get ready for
Your
some
yaw to
sleep.
the left. Elbow out first. Yeah, turn it right. Yaw to the right. Now elbow in. And yaw to the right. Yaw to the right. And yaw to the right. There you go. Sit down. Too much pitch out. Don't come in anymore. Shoulder right. Shoulder right. Pitch in. Pull a little tighter if you're hot. You're too low. So you bring your arm up. There you go. Now you can shoulder left. Shoulder right.
oh, yeah, there is a little squish.
All you gotta do is elbow out. Elbow.
Oh, look, we got a machine. Look at the baby squid.
Where? Where? Oh, don't get in the quiver.
It's crying. Oh, it's really in
Honestly,
the,
looks like the other.
it's really living life on the edge.
That is so cute.
That was really cute.
Baby squid sighting.
Sponges, guys. I really appreciate it.
All right. Number 12.
How many sponge cultures have you prepared to make this whole bird? How many culture plates and how
I
many?
made 375 plates.
How many plates do you make per coral? I mean, per sponge?
When I take it up, I have 5 per sponge.
Five, so there's how many plates do you-
That's all. Yeah, so we're ready for sponge singing.
You could get 75.
But I might use some for subculturing.
Ooh, there's a little something here. Look at this.
So, see if culture,
They've been all over the
So
place.
like,
whatever.
like
Really?
separate
Yeah, in
colors,
this camera, you
like,
see 'em
I
coming
put
in and
them
out
together,
all the
like,
time.
between 3 and 50 cents.
30 what?
It's amazing how much is going on right
So much
here.
going on.
They're just.
We have like 6 now.
We need more.
We need
Just
more.
go down.
Definitely.
Go down.
How much money do you have for each?
I'm not sure which ones I'm most interested in sequencing. The one little sequence are the ones that are
Pitch
the
up.
water and the ROS and the pumping.
Ready to ground it
What's that?
before?
Freeze and come on.
Plates, do you have those?
Fumble. But yeah, I mean, yes, you could. I think it would be that interesting.
I love it.
Why? Why not cool?
Not wrong.
Maybe
That
when
story is
It's
true.
long.
Does it have a fun?
All right.
Just a little work on
You think you can reach that white one there?
If you want to try it, we can try it. They're not filter feeders, so I think the filtering in the sponge is part of what makes it interesting.
passive filter feeder. What,
Yeah, let's look it up.
what's the medium? What's your culture medium?
Container
Below
blisters,
what?
agar.
What, grounded bologna? Just grounded bologna with peanut butter?
Ground up what?
That's what I grew up stuff on, grounded bologna and peanut butter.
In your fridge, you mean?
Or
Or in your closet, all these samples you
all
have.
my electron microscopy lines. I got all kinds of corals in my closet.
Do you?
That I processed for electron microscopy, but they didn't section more images then, but
My
I
understanding is
box
that
that I took my.
in corals, the microbiome is more associated
What's that?
with disease, right?
Well, the oh.
Oh,
You get
people
that
are
one for me?
bullshit.
I'll take that. Thank you very much.
Nice.
Nice. That one snapped off easily.
It's fairly wide.
Look at this cake.
And by months.
Oh,
Up to them.
beautiful.
Can it go in the
Can you
bottle?
hold for 1 sec? I'm gonna get a picture of that. Can we get a snapshot?
Did
Two.
you get a week before?
Nice, thank you.
Thank you,
Not so
guys.
bad. That's
Is that in a bio box?
That's a coral.
Is
That's
that a
hand.
coral?
That's a hand. That's a coral.
Okay, Mr. Coral, give me an message number, please.
Wait, what is it?
I don't know. I gotta focus on that.
Working on
give me a number.
This is Blur Gorgia.
Blur Gorgia. I like it.
Where are the solids?
SH18690.
Thank you.
Blur
Well, we could
Gorgia.
culture its fungal microbiome if we wanted to too.
They're deep. They retracted really far. Oh, that looks like.
That would be cool to look at it actually, Kate.
Actually, I don't know
Look,
what it is.
might as well look at a coral. That would be cool.
What bio box can we put this in?
It can go in either compartment, BioBox 2, or 1B or 3B.
Which one you want? You want 2B?
I really
call
love
it
getting
blur
to
To
see
be
the
or
different
not to be?
propulsions of these small larvae and stuff. They've got so many different ways of spinning around.
Yeah, it's just a little guy, little baby fish.
Yeah, the baby larvae are fascinating and they're amazing.
All right, it's 3:26 UTC. Collecting another specimen that we thought was a sponge.
We should give some to Jeremy was suggesting that.
Wait, you see my sponge today?
Yeah, and we could
Well, to identify what species the sponge is when we get it up.
We have a Seagram chapter.
A what?
A Seagram chapter.
What does that mean?
The organization. Seagram.
Yeah. Does that have a tag on it?
Isn't that just an alcohol?
Yeah.
That we actually have a lobby, so
Oh,
we
good
have
job.
Yeah.
bacteria.
Yeah. I'm trying to rip one. Oh,
Or
hey.
samples.
Oh.
We're outreach mainly.
Where did that go? BioBox 2?
Bio box 2B.
2B.
Yes.
Or not to be.
Happy birthday, Colleen.
Everybody Colleen?
Finish the sample.
Oh, you guys only have 33 more minutes.
Zooming in on it.
Are you just making up for the fact that Scott's not here right now? Partners in crime.
Cool.
Very
Flying
interesting.
around a little bit more?
Yeah, let's fly around.
Yeah,
We can keep
I
doing
have
it.
20 more liters.
All right.
This should only be a couple minutes.
Stay at this depth, a little bit deeper.
I think we want to stay at this depth, right?
Yeah,
For
but if you want to go just a little bit deeper, that's fine too.
Okay.
What do you want? Stay at this depth?
Yeah, why don't we just stay at this depth?
What
Maybe
about
kind
that
of.
great yellow sponge?
I think we're gonna just
Move on.
move. Yeah, we gotta get up and we gotta be off the ground to do the eDNA.
All good. Okay. It's beautiful.
Squid. Squid and fish.
Oh, I love all the squids.
Why are you so angry, buddy?
I know, he's kind of upset.
Not into the light. Oh, he's trying to get away, but he's next to the wall.
Oh.
Let's leave him alone.
I'll leave
Oh, okay, he's disoriented. I don't like this.
Hi, buddy.
Oh, is that a? That might be a Montastria there.
Yeah, Montastria cardinals.
Yeah, the bubble gum or the gumdrop
What is that?
Montastria.
The one we couldn't see for the fly.
Yeah, so that would be a good one we could come back to.
The ROS in.
Yeah, and we couldn't. Ooh, there's a squid again.
I left you alone.
It's the same
Yeah.
one. It's just
Go away, dude.
This is his fault now.
This is his fault.
That's what happens when you stare at the light for too long.
It's
It's
just
all backwards.
It's just really confused by the light. There we go. Got out.
Do you you guys try any octocorals? Check them for ROS when you dove and
That a sponge or a coral?
Ooh, what is that one? Oh, that's a sponge, I think, and it's got the zoanthids on it.
Wow. You don't think that's coral?
No, no, it's got the osculum there.
This is a good spot right here.
Nicole, do you need to stop
Oh,
at
how
home
pretty
for
is
a
this?
sample something?
Yeah.
Oh, I didn't
Yeah,
realize
left
that.
on- How much time do you have left on pumping?
I have 15 liters to go, so maybe 5 minutes.
Ooh, can we see what this is over here? Is that a
I'm
sponge?
just gonna give you this cold view.
This is amazing.
This little spot is really nice.
This is, we should.
Oh, it's a, oh, forget it, it's a big barrel.
Can we mark this spot?
I just took a screenshot.
Oh, there's more of the Montastro over there.
Over here.
Can we just get an overview real quick? Give me a sec.
This is beautiful. Wow, where are we?
Canyon too. This is cool.
What if we zoom down into the giant barrel swirl?
And you keep zooming more. Then we're in
And
another.
we just realized Puerto Rico is just a big barrel swirl.
Where'd you want to look at?
Oh,
That one there to the left?
Well, this one, there's quite a bit. I'm actually curious about.
Oh, there's that sponge.
Is that a
green
sponge?
zoanthids. This thing's a sponge.
Oh, that's a sponge,
See,
yeah.
this thing's a sponge
The
right
green
here.
one?
And this looks like maybe that's a
Yeah, that
Montastria.
one's pretty common. I don't remember.
Oh yeah, that's a
But it does have
Montastria.
yellow zoanthids,
Yeah,
yeah.
it's got the different.
We saw a lot of that in the Virgin Islands.
Okay, what do you wanna call this? This area?
Beautiful.
Coral Canyon.
10 liters.
Sponge king.
Thank you.
Ooh, this is gonna look cool over here. There's like a nice little.
There's a coral
Oh,
here.
look, hiding down there.
Yeah.
There's a little bit glow patch right there.
Oh, it's a tree.
Wow.
Their shark might be around.
Yeah.
Wow. So beautiful.
Yeah, this is habitat.
What depth are we at here? 33? Yeah.
So
We
it's
saw,
33 still.
parrotfish, the mucus.
Come back
See
down.
that?
Yeah,
It
it's
was
not-
so
Go down?
cool.
No, I was saying when we come back down, it'd be cooler to check out those big ink path. You see if they make a signal like those fluent path use.
So is 33 considered mesophotic or?
Do you need me to go
Depends
back?
on the region. If there's,
Right. How turbid the water is?
Do I need to go back down
30 liters is 90.
for the E EDNA?
You're fine here.
We'll have to-
5 more liters.
Yeah, there's a really nice thing up there. I'll go down here and then I'll come up where we end.
Can I poke a peek at that really quick?
Yes, ma'am.
I think that might be a coral here. Maybe a. Sit.
We saw a bunch of rules that so ours was slightly input.
Yeah, I think that might be Psittastria. What do you think?
I think.
much coral in
What
the
about
face.
Isia?
Yeah.
Oh, which one?
It's kind of aggravating.
Oh, you think it's an Agrisa? Yeah, yeah.
The ridges.
Yeah, you're right.
Good.
I wanted to find the varieties yesterday. We used to measure it like yeah, we found tons, but it was after solar, the solar was set up like, but that's when we were up at like 15 meters. We saw so many corals in there. It was just forest of octopus, just all thing like a dozen species. So many of them.
There's a lot of Agaricea here. Nice plates. Little tiny barrel sponges. Itty bitties.
Alright. EDNA sampling complete.
Noise.
So they did one.
Oh, lionfish down there.
Mm-hmm. Being all creepy.
Being all creepy.
Yeah, chubby. Boxfish. Hey,
Look like a whole plate that just fell off.
Yeah, this is just amazing.
And look at all those black whips, black whip coral. Bright green.
It
Like
looks like
snorkel.
sideways.
I've seen those corals in Curacao with snorkeling.
I'm down now to like 43 meters.
the one that looks like
43, okay.
Yeah, the ones that like.
Yeah.
Oh,
Is that your
line?
Oh, there's a little Montastria right here.
There's one up to the left, top left screen.
Yep, and a little one there. Yeah, little guys. Feel like we might have lost the Porites at this depth. It's surprising we still have Montastria though.
Pumping done?
Yep.
Are we coming up now that she's done pumping, or do you want to keep coming up?
I think we could probably come up. I mean, we could sit here all day, really. So beautiful. Somebody's got to make the call. But yeah, we got a lot to process.
Gotta be the party pooper.
Ooh, what's this thing?
Cool, you got an idea?
That's a
of
big
which
old
things-
sponge.
Oh my good. Look at that sponge.
I think the, whoa,
Spiking.
Imagine you put the flowers in your bag.
Holy cow.
Is there anybody
I
inside?
think that's probably the only one.
Let's have to look inside.
Worthy. They'll probably have his polyps in.
Oh, is that a little sea cucumber there?
That's a good sponge.
Look at that one.
Perfect
Do
timing.
you want to try to put squeezer number 6 in there and fire it?
You want?
man, that big.
Wait. Yeah.
Do we want to?
If we,
No.
are you done with your eating?
Yes,
yet?
I am,
Oh,
yeah.
are we
Drop
coming
the
up?
Go to town.
No, don't worry about it. Don't worry about it. Come back and get it. That's an amazing sponge.
That it's just. Yeah.
Yeah, that's what I was saying. I think we should.
Yeah, it's good.
What?
Squirt that pink stuff all over it.
In this one.
About, doing the dye?
Yeah.
Well, we could drop a target here.
Yeah, we could come back tomorrow. This is a beautiful area right over here.
Why don't we drop a target here, yeah?
Maybe more for all coming today.
Yeah.
Sponge garden, yeah.
Yeah.
Oh, that's crazy. Very cool. Something else.
Oh, something digging.
No,
Are we coming up?
maybe the only thing using.
Yeah, we figured we should probably get
Sponges.
everything processed and then try to get Solaris plugged back in and troubleshoot. You know.
Both these colonies.
Lionfish.
We're about to start recovering here in a little bit.
That's what I had
Just gonna come over the top of this and
exactly like normal.
thank you.
Oh, look at the eel.
Eel, more.
I wasn't sure. I didn't get a chance to ask.
Oh, that's amazing.
I didn't get a chance to ask something.
Wow. Look at you, little guy.
We're planning on just doing a quick turnaround and get Solaris.
But you're gonna pull samples out, right?
Wow, look at that.
First time on live experiment.
He is not happy with you.
He just has resting grumpy face.
I would
There you
never.
Huh?
I never get this close when I'm in the water because they freak me out. So this is nice.
No, my husband also can't look at them.
No, they totally freak me out.
Don't make eye contact.
Yeah. Oh, he's coming out. Is he coming out?
We don't plan on collecting any more samples before we recover, do we?
I don't think so. No, did you? Unless you want something more. Wow. That.
Fair amount. I haven't done it like not in years.
Which guy? Which guy?
It was at who gets scientific
Oh, arrow crab. Oh,
No, it's like
oh, okay. Bye bye.
Oh, he's going back in, huh?
Okay.
Yeah.
I don't think this light is good for these stories.
Yeah.
I'm gonna leave them open.
Yeah.
Yeah.
There's another big sponge.
Giant one. Yeah, these are great.
So a bunch of Agaricea.
Are you gonna come back here after you
Another
drop me off, or-
Agaricea. Yeah, I mean, I think.
And you said you wanted to troubleshoot Solaris. You just need to plug it back in. And make
Well, we
sure
need
it's
to plug it in and then make sure it's working, right?
working. But you don't want to necessarily tear it apart.
Oh no.
Okay.
No, just plug it back in and then flush the lines. Maybe. I mean, we thought we maybe got a little gunk in there.
Lena and Erica can do that, right?
Yes.
Okay. Yeah, I'm gonna turn around squeezers and get.
How many squeezes would we have?
3 samples and 1 biscuit.
Oh.
So you ready? Yeah. All right.
You ready?
I'm ready. I labeled the bottles.
Can we look at that just really quick? I just
Oh, I see.
wondering if we're. No, that's not.
Happy birthday, Colin.
Happy birthday, Colleen.
Oh, there's a Parites right there.
Oh yeah.
A little guy, but
Feliz
what are we at here? We're
cumpleaños.
at 29. Yeah. Oh, and this might be one here too, actually.
Yeah, they have to zoom in on?
Sure, if you don't mind. Yep. That's a bigger one.
Plenty of Parites targets for us to ground truth. So what happens? What happens when we put Solaris on and there's nothing?
Then I'm very, very sad.
Well, yeah, then we got a problem to figure out.
And then, yeah, so then there's a problem.
Colleen, do you have an estimated turnaround time between ducks?
Well, we gotta process squeezers and
I can probably get the squeezer turned around in an hour.
hour and a half, I would probably say is more realistic, right? Yeah, something like that, assuming that there's nothing wrong with the Solaris. Ooh, look at, is that growing out of the sponge? Or is that an illusion?
Might be an outlet, yeah, I don't know.
Out of the sponge.
Out of the sponge. Nice.
It really is.
This house one at Deep too.
That's cool. Yeah. It's like a tree that grows in
Out of
like
out of glass corals or like glass sponges.
That's cool. It is giant barrel sponges.
I can see like how that happens like once they're adults, but I just don't see how like a larva settles and manages to survive.
Wow, this is funky looking.
Oh, this is like
Woo.
a lesion.
Or maybe they both grow up great.
That might be a little Parites over there. Little mound.
If you turn it upside down, it looks like Star Trek.
They're best friends. They're best friends in a while. Oh, that's cool sponge. That one?
Oh, yeah.
Then you want to go start getting ready? You want to start getting ready? Okay. Just, yeah, release command in here.
Oh, here's some big Parites.
Yeah.
Parites, Parites, Parites.
Yeah.
Parites.
Yep.
And then y'all just
Bunch right there. And there's another one.
See, so, so when I see that, it's like the living part is really just this cap.
Yeah.
And that may have been Parites below it.
It might have
but
been,
that
yeah.
died and then it resettled. Is that kind of
Yeah.
how that works?
Yeah.
Just one time.
Ugh. Who's that?
Oh, I don't know.
Grossness.
Blob. The blob. An estimated turnaround time. Yeah, probably 90 minutes. We got a team. Everybody's awake. We're gonna be trying to turn this around quick. That's amore. Oh, we got some comedians online. Yeah. Huh?
They're always there.
They're always there. That's amore. That's a dad joke. See, I would get criticized for.
This place is pretty
Oh, there's a parrotfish down here.
Yeah.
Yeah.
And a sergeant major. Ooh, a damsel.
Actually, they don't say that's what I like.
Oh, look, that fan that we're going by looks actually doesn't look grazed. It looks diseased.
Yes, it does. Yeah. Yeah.
And look at this one. That was a fan.
Yeah.
There you go. I made it back.
We got now, we're at 19.
I'm in the washing machine where I'm at.
There's the Paredes. Ooh, what is this guy? Can we look at that really quick? That did not look like a. Oh, and down there. Ooh, that might be. What is that?
Madrasas?
Is that Madrasas?
Yeah, maybe.
Is there a choral person
Let me see.
still here?
It looks fingery like that, but or would it be a type of, like a crop rub?
There's a whole bunch of those other dudes you like right there as well.
The Paredes asteroides here, and
Oh,
then
and there's the lettuce coral, right? This one down there.
yeah, the
Is
Agaricea.
that, oh, that's right.
But this one might be.
Take a screenshot of it?
Yeah, I took a picture. That might be.
You want a better image of it?
You've got one.
I think it's okay.
Niskin
Okay.
4.
Oh, here you are.
I was curious, do you have plans for the other three?
Oh, and up here too. There's.
There, it's not fired.
You see that
Yeah,
up
I was
there?
wondering if you
You
guys
want
have
to fire
plans
them right now?
I just want to
Yeah.
fire 2 more.
Go for it.
This is
Tell
a
'em.
very good spot.
I think that
Yeah,
might be
so I
Madrasas.
I'll just take 2 liters from. Okay.
Tell J-Rod.
Yeah, J-Rod, when you get a chance, can we fire Niskins 1 and 2, please?
Yeah, right.
Thanks.
That looks like a can.
Good deal. This is good. This is a
Yeah,
good
no,
one.
this is really good for
Is that
octocorals.
a can?
This is very exciting.
That's a sponge.
The microbes be cool too.
Right, yeah.
We'll have like a whole duck range for microbes.
Well, that's neat. So that was at 19 meters. I think we saw Madrasas.
Cool. Yeah, I'll just take. I don't need. Oh yeah, if you, Scott, if you guys want any water, I can only take like 2 liters. So just let me know.
Okay.
I'll leave like 2 liters. It's 2 or 3, I guess, if they're 5 of them.
Oh yeah, and there's some more over there. I think that's what they kind of look like little fingers. I think.
Trying to find a good one.
No, I'm good. It's okay. I'm awake.
Alright. Ooh, I'm excited about this one. Look.
Hey, Scott?
Do you filter that water?
Yeah, I do.
2.2?
Yes.
Yep.
Could I give you a bottle and you could save some of that 0.2 filtered water?
Absolutely. Absolutely. Yeah.
You filter it for us.
Yeah.
You scratch your back, my
Oh, there's
back,
some-
you'll scratch yours. No way, that's not how it works.
Yeah, I'll do that for sure.
Yes.
Yeah, just give me the thing and I'll make sure it's clean in there. Otherwise, I just dump it in a bucket. So.
Yeah, right, okay.
That looks like it was a Montastria, but it doesn't look very happy.
Pretty sure you are right, my friend.
Oh,
Where?
is this? Yes. Awesome. That would be neat because we weren't seeing them.
Yes.
What,
Oh, wonderful.
What Niskin do you want fired?
Cool. Thank you, Ann.
I believe 1 and 2 still haven't been triggered. So
Yeah, it's a
those would be great.
With the crane. I think, I don't know. Did I tell you I got a plane ticket? Sorry, yeah, sorry. You were asleep. Yeah, you got a plane ride, so you're good. We're gonna fly from Mayagüez to San Juan at 4 o'clock.
It's better than driving.
37 minutes.
You're gonna have to pay on the
$80,
kids.
it's like $80, $70.
It's really it's cheap. You want to get the kids,
Right,
the twins.
in San Juan. Exactly.
No, what is it?
Yeah,
Some boy and a girl or
no, all girls. Three girls.
they're the Bobby from the they're the Bobby side, that side,
What?
like the girls.
Are they daddy's girls?
Yeah.
Oh, yeah, of course.
I don't want to be.
What's wrong right there?
Oh,
Oh, click that green, the green button. It's off for some
I
reason.
don't want to give him
See it?
Yeah.
Not for me.
Give me a second,
Well, I
Ben.
didn't have to do that. She did.
It was planned or was that?
Nope, we're just firing these two.
For the twins?
Those things still working good?
Yeah, they're working amazing.
Great, check. Awesome, thanks, Gerard.
Ben,
It's called Bridge.
I got you, Ben.
We were aiming for
Wait
two
just a
pins.
second, buddy.
Overshot. With 3. So we said we got a 2-for-1 deal.
I'm not old. My parents were divorcing
Yeah.
and my mom found out and the boys have like almost like
Oh wow.
they still do that. I don't know why, how.
What's that?
Anything. I just keep in the middle.
Okay.
Mine is
All right, we are good. That's it?
Yes, I think we're good.
years older than
10 years older? Yeah. Yeah.
I think it was.
Well, yeah, it happens. Sure. You give me bottles.
Hey, look, we're gonna be coming off bottom. Tell them, yeah. Hey, Luke, you got me?
Alright,
We're coming off bottom right now.
Well, 1 or 2.
Ben, I'm trying to talk to the Ben. Yeah. Look, we're coming off bottom now. If you can send some guys out, slope.
They're all here.
Okay. Cool.
I'm fine, thank you.
Yeah, we'll do.
All right. Well, that was exciting. So we are going to recover the ROV now and pull off all the exciting samples. We got corals and sponges and process water samples in the lab. And we're gonna plug Solaris back in and fire it up, make sure everything's running okay. So we should be back in the water in a hour and a half or 2. And I think we're gonna come back to this area and do some more measurements and collections. It's a beautiful area here and a lot of corals to see and to study. So thanks for tuning in.